Good morning, everyone. I hope you are enjoying Bangalore and the 9th Annual International Conference of BGCI. So without wasting much time, let me introduce our chairperson, Mr. Prasanna Sirol. He is a dedicated father of a child affected by rare diseases and has been trailblazer in rare disease advocacy in India since 2007. He is the co-founder and director of Rare Disease India, and he has played a pivotal role in raising awareness.
And Vice President of the BGCI. After completing her BSc and MSc, Ma'am went on to complete her PhD in Genetics and Biochemistry from IIT Chennai. She has served both national and international corporate world till 2005, and after 2005, she has been passionate to serve society through science and education. She was instrumental in starting the MDF, which is a journey in the area of rare genetic disorders. She established MDCRC in 2011 and has been serving patients with Duchenne muscular dystrophy since then. Her adept knowledge of the subject and interpersonal skills have grown the institution to an international level. Dr. Lakshmi is also the principal investigator for many national and international projects. She has presented her work in many national and international conferences. She has published research papers in national and international journals of great repute, and she's a member of the International Ethics Board of AVP. She has been awarded with many awards which are extremely precious to us as a community, but the one worth mentioning and which we are indeed honored that she has won is the prestigious Community Empowerment Award in 2024 to raising public awareness of genetic disorders by UAE Genetic Diseases Association, Dubai. Heroes Award in 2015, Healthcare Achievement Award, Yuva Shakti Award. The list is endless. We're indeed proud to be associated with ma'am and learn from her every day. We welcome you to the stage, ma'am. Now the chairpersons will take over and invite the first speaker, please.
So I think today's session will start with Dr. K. Tangaraj. I don't think he needs any introduction because he's a familiar face and known to everyone. So Dr. Tangaraj is presently a JC Bose Fellow at CSIR, Center for Cellular and Molecular Biology, CCMB, Hyderabad. He was also earlier a director of the Center for DNA Fingerprint and Diagnostics, that is CDFT. Then additional charge of Institute for Stem Cell Science and Regenerative Medicine, that is INSTEM in Bangalore. Last 3 decades, he has been, in research area in the field of population and medical genomics, and he has published over 300 research articles. He is an elected fellow of Indian National Science Academy, Indian Academy of Science, and National Academy of Science. He is a recipient of several awards like JC Bose Fellowship, Sun Pharma Research Award, in Medical Sciences, Raman Research Fellowship, Lifetime Achievement Award, Excellence in Science Award, Distinguished Scientist Award, Sir C.V. Raman Memorial Award, Sir Dr. U.N. Brahmachari Award, and delivered several orations. So I request Dr. K. Tangaraj to come over and take over the session.
Could you please change the slide? A very good morning to all of you. It's my pleasure to be here. And before that, I must thank the chairs for the kind introduction. Also thank Annie and Willu and other organizers for inviting me, particularly to talk about genetic variations. When we talk about genetic variations, there's no other country than India because of so much of diversity that exists in the Indian subcontinent. Let's see, that was close. No, it will be closed. Yeah, so because of the huge amount of diversity which you have in India, if you look at this slide, we have people typically look like Africans, the Andaman tribes. We have people who with very light skin color typically look like Europeans. There are some people, particularly the Northeast, their phenotypic features are like Southeast Asians. And there are people with very unique features are unique to India. So when you ask me that what are the reason for such a huge diversity in terms of phenotypic diversity and genetic diversity, so we have the very first modern human who migrated out of Africa, and subsequently there are several migration into India until very recent past, and people have admixed without any restriction until maybe last 2000 years. And now people practice endogamy. And we also have population which are living in very isolated place. So that makes every individual or every population acquiring genetic mutation. There are a lot of genetic environmental influences that that's mainly because of natural selection and so on. Although we have evidence, genetic evidence for most of it, but I will be highlighting some of the very important aspects because of a very time limit. I mentioned that we have population which migrated for the very first time from out of Africa. That's none other than the populations of Andaman and Nicobar Islands. Subsequently, we looked at population around India to understand population history. So in this study, what we have shown was that in the prehistoric India, there were two founding populations. So one, we call them as ancestral South Indians. They are part of the early human migration, while the Andamanis were migrating through southern coastal routes. Some groups stayed in southern part of India, whom we call as ancestral South Indian. And there was second wave of migration again from Africa, and somewhere in the middle, people have diverged. One group has come to northern part of India, other group has gone to Europe. Still, we have a lot of genetic evidence, particularly the Y chromosome haplogroup R1A1, which both people from North and Europe share that. The skin color mutation in the SLC24-5A gene, the lactate persistence gene mutation. So all suggest that this ancestral North Indian and Europeans have some kind of genetic affinity. So these are the two founding group in India. On the other hand, if you look at the PCA, the principal component analysis, while these three HAP map population, the European, African, and Southeast Asians, although several individuals are there, they are in tight clusters, suggesting that they are genetically more homogeneous. But look at Indians, although several populations, so they're all very unique. Also, there's a kind of genetic affinities between the group. That's why there's a gradient of clusters. They are not clusters somewhere here and there. So although every population is unique, but we all share some kind of genetic affinity. To understand this part, the ancestral South Indian and ancestral North Indians, we have actually looked at these two groups to understand when the admixture between these two groups have taken place. So for that, we took another study where we have taken large number of samples from South, large number of samples from the North, and try to understand when this admixture might have taken place. Initially, what we have shown was these two founding groups initially gave rise to many populations. So you can imagine that the first out of Africa human, when they migrated, they must have settled somewhere in the jungle. And after completing food, whatever was available there, they must have, one group must have migrated to other place in search for food. From there to another place, then they must have started agriculture and so on. So every time there was a separation of groups. That's what is shown here. Both ancestral South Indian and ancestral North Indian. But these two groups have admixed during the last 2,000 to 4,000 years. Okay. So after that, during the last 2,000 years, every single population started maintaining the endogamy, means marrying within the group. So this tells that about 65,000 years back, there was a migration. There was two groups, and those two groups have mixed during 2,000 to 4,000 years. Then last 2,000 years, every population started maintaining the endogamy. And this happened much before the caste system came into existence. Okay? Probably the caste system must have put more pressure on every single population to maintain the endogamy. So now let's look at, so what is the impact of the endogamy on our health and disease? To understand this, we took another study, and this time we don't want to actually restrict only within India, wanted to expand this to other South Asian countries. Because when you expand this, in India actually we have 4,635 anthropologically well-defined population groups. But if we expand to South Asia, we have 5,000 population, which nowhere in the world has such a huge diversity. There was a couple of reasons why we want to go to other South Asian countries. One, at least at population level, which I am not showing here, we share at least 50% of the genetic component has gone from India to other South Asian countries. The second one, in terms of the disease, so one of the mutations which we found in myosin binding protein C3 gene, it's a 25 base pair deletion. We extended the study to worldwide population, and we found that mutation is restricted only in South Asia. Just one example. There are several such examples we have. So that means whatever we study in India, it's also relevant to other South Asian countries. Right? So that's why we wanted to expand it. So then we went on to collect samples from 2,800 individuals belonging to 275 endogamous group. So that, as I mentioned, that all the South Asian countries have been included, including Sri Lanka, Pakistan, Bangladesh, Nepal, and so on. So in this study has revealed two different information. One of the, of course, understanding the population history, which I'm not talking here. So mostly, the important findings of this study is that there's a lot more opportunity for us to understand and map the mutations which are causing the population specific disease, particularly population specific recessive disease. So I come to that. This is called IBD, identity by descent. And every single line represents one population. There are 275 lines here. We also included Ashkenazi Jewish and Finnish here to compare, because these two populations are well studied. Lot of disease genes have been mapped. Therefore, we wanted to see where they stand compared to rest of the Indian population. You can see they have very tiny IBD score. IBD means it's calculated depending upon, for example, we are comparing individuals from one particular population. And most of the individuals, for example, if you are taking 100 individuals, for example, 10 of them, 50 of them, 50% of them have the same haplotype. That means there's no difference in the DNA sequence. Today they say that they are unrelated, but actually the cells that they must have come from a common ancestor, therefore they are sharing the haplotype. So based on that, this is calculated. Now we look at that most of the Indian populations are with the high IBD score. That means possibility of coming from a common founder is much, much higher compared to the Ashkenazi Jewish and Finnish population. Now I'll show you the disease which was found in Ashkenazi Jewish and Finnish population. There are several diseases have been identified. So these are the genes, and most of them are autosomal recessive. As I don't have to explain about these two. So autosomal dominant, obviously whosoever carry the mutation, so they obviously show the disease. Whereas the recessive mutations are the one which you don't see in the carriers. Therefore, there's no diagnostic there. Until otherwise, the person who carry the mutation, the heterozygous condition, marries other person with heterozygous mutation, there's not going to be much of a problem in the family. But when they contribute the mutation to the next generation, both the parents, therefore there's a recessive disease. So both Ashkenazi Jewish and Finnish have such a huge problem. Now we look at that when with the very tiny IBD itself, there are several disease and disease-causing mutations have been identified. Now you imagine that almost 1/3 of the populations are with even much higher IBD. So what kind of problem most of the Indian populations are with? And most of the informations are not even in the record anywhere, even ICMR. Right? But we predict that lot more population in India are going to have a problem. So I'll show you to explain, because I don't have to explain about the pedigree, but explain about the problem with recessive disease. For example, here there's a mutation in heterozygous condition. That because it's a recessive mutation, no problem at all. She married somebody from outside without mutation, therefore no issue. The mutation again going to the next generation in a heterozygous condition. Then the next generation, these two are going parallelly. But after a few generations, now you see in the same community, right? But now they might have lost their relationship after several generations. But you can imagine somebody who come from a village. There's a village, some 100 families, and you expect it all 100, although some of them they say that they are unrelated. You can actually trace back, and they must have come from a common ancestor, right? So this is what happened. So now they show that we are not related, but both of them are carrying the mutation, girl and boy. If they marry, there's a 1/3 of chance that they contribute the abnormal chromosome to the next generation. Therefore, there's a disease. So we have several examples. This is one of the examples I'll show you here. There are a couple of families here. There's a gene called polymerase gamma. This is a nuclear gene, but this is essential for mitochondrial DNA replication. There is a single base mutation in the gene. This is a wild type, the mutant, the heterozygous. You can see both C and T existing together. The parents, this is a consanguineous family where you see both the parents 45 years, 39 years, absolutely fine, but they are heterozygous. But unfortunately, two of their children carry a homozygous mutation. Therefore, there's a disease with encephalopathy, seizures, stroke-like episode. Most importantly, they die at very, very early age. You can see that these two with the homozygous mutation, this boy has already died, but we don't know the fate of this girl because this we have analyzed long back. I'm sure that unfortunately he also may not be exist. This is this consanguineous family, but here you see the non-consanguineous family, because that mutation is well spread in heterozygous condition, although they are unrelated, they married again. It's unfortunate both the children have died. And this causes huge amount, as I mentioned, that brain MRI shows a lot of lesions on the brain. And this is the intact 10 kb mitochondrial DNA. But with the mutation, patient shows multiple mitochondrial DNA. Single base deletion, single base mutation in the nuclear gene, which causes mitochondrial DNA deletions. And these are, of course, some of the histopathology studies shows this accumulation of abnormal mitochondria in subsarcomal level and the COX negative 5. But these are some of the diagnostic markers. So this is just one example. There are several such examples. Now I am going back to give you the track of a study. Now we know that there are a large number of populations are expected to have population specific disease. But we don't know any phenotype at all about these populations. So we selected 4 populations. One is Reddy, Color, and Yadav, and one more. So we went on to do exome sequencing. So we went back to the population. Of course, we had already DNA. So these are the population, including Kalinga. We collected these many samples. And while doing that, we came to know that the Reddy population from Anupati, they have a type of spondylitis. But the Color is the one who has disease called junctional epidermolysis bullosa near Madurai. There are 8 such families. You can see multiple children are affected. That means parents are heterozygous, no issue at all. But unfortunately, there are some families where all the 3 children acquired mutation from both the parents and they are normal. So we identified 8 such families. And this is a phenotype. What you see here is we went on to do the exome sequencing. We identified 11 base pair deletion, a gene called LAMB3, and a lot of lesions, including congenital heart disease. The children die within 2 months after the birth. Then we went on to see how this mutation is existing in our own sample. We had 9,256 samples. None of them carry the mutation, even the heterozygous condition. Then we looked at the database of worldwide whole genome sequence database of 8,62,000 individuals. When it converted to chromosomes, 17 lakh chromosomes. Only 2 individuals with heterozygous mutation. We don't know which part they have come from, but they probably say that they are South Asian, probably from India, from that region. But otherwise, this mutation is very, very rare. When you talk about rare disease, this is different type of rare disease. There are common rare disease, which everybody knows. This is the rarest of the rare disease. But when you go to the population, the Color, what is common? Right? So we have to look at different. Then we went on to, so we have 2 different aspects. One is our curiosity to look at basic science. Other one is how we can help the population. Then we went on to see whether all the deleted chromosomes. So we, at the population level, we identified 10 heterozygous individuals. Wanted to see whether all the 10 have come from a common ancestor, whether they all have same haplotype or different haplotypes. So the deleted chromosome, they are the same. It means there's a single mutation which arose in the population some time ago, has been circulated in the population. Whereas the other chromosome, we see that they are different. So we looked at 2.64 megabase region to identify whether the deleted chromosome has a common founder. Then we went on to see, because this mutation is a novel mutation, so we don't know whether really this mutation is the real cause for the disease or not. We looked at, and this is where there's a deletion. This is the position. The wild type gives 1172 amino acid, whereas the mutation introduced truncation. It's reduced to 799, nearly 370 amino acid missing. Then we went on to create an animal model using CRISPR-Cas, and we got two lines of deletion. So one line with 45 base pair deletion, but that is non-frame shift deletions, delete only 15 amino acid. Then the animal which we developed are actually showing the phenotype which we have seen in the patient. And also internal organs are, there's a kind of inflammation and so on, and we are looking at it now in detail. The other line precisely deleted 11 base pair, and that, because of truncation, deletes large number of amino acids. Therefore, there's a truncation of protein, and hence it leads to the pups are dying within 2 days after the birth. So this impact is very severe, although it's 11 base pair deletion, because it's truncating. Whereas the 45 base deletion, it's not truncating, but it's reducing only 15 amino acids. So this is our science curiosity part. Now, how can we help the society? In the meantime, we came to know that there are a couple of mothers who have got pregnant, and we try to collect amniotic fluid, and in Madurai, first time we could not able to find a place. Then we brought the patient to the parent, this mother, to Chennai, and we collected the amino acid. We collected the amniotic fluid, and we just did very simple PCR based method, because the 11 base pair deletion we could able to detect by simple PCR and electrophoresis. Of course, we confirmed by sequence. This homozygous, heterozygous, and the amniotic fluid shows the heterozygous condition. Therefore, we advise the mother, although we are not fully trained counselor like you, but whatever the genetic information which I have from my MPhil PhD days, so we did, and it's our joy to see the child born with the proper phenotype. Okay, and the second child also was with heterozygous condition, fortunately, and so we have satisfaction that we could help the family. Then in the Genome India project, some of you may know that DBT has funded sequencing of 10,000 individuals. That's also very important for you to look at the frequency of the variants which you might find in the patient. So we had a camp near Madurai. We invited the people, we collected the sample, and we screened. So this is what the results of the 10 individuals who carry the heterozygous. And to give you some perspective about the Genome India project, we just completed sequencing of 10,000 individuals. You can see all of this from 99 ethnic groups that represent all the 4 major linguistic groups like Dravidians, Indo-European, Austroasiatic, and Tibeto-Burman. This is the Tibeto-Burman, Austroasiatic, and this line actually is the Dravidian and the Indo-European. So this we sequence and identified now, just give you the account of what we found is 54 million variants. Okay, single nucleotide polymer. Of course, we are doing it. So this actually DBT considered as one of the important projects, not only for the investigators and also for scientific community as a whole. As I mentioned that if anybody want to look at the allele frequency of any mutations, one can use it. But right now it's not available. Of course, maybe next year beginning, January onwards, will be available. So this was actually on 27th February, the Honorable Minister of Science and Technology has announced the sequencing of 10,000 individuals in Delhi. So this is one information which we, myself and Professor Naragari was coordinating. I also want to highlight about the another program which I initiated when I was director of CDFP.
Excuse me, doctor. 2 more minutes.
This is the last slide. Last but one slide. So we initiated a program called Pediatric Rare Disease with the background which I was telling, understanding the issue. And this allows, and this is of course, the Secretary DBT was launching this program. This has many components. You can see that institute all over India is part of this. And initially they do screening, then if you don't find anything, go for exome or whole genome sequencing of 5,000 trios. And of course, prediction, making animal model, as I was saying, and the part of treatment. And we made a video in 8 languages. You can look at, you can type this pediatric mission program on pediatric radies, just to make the awareness of this program. I'm sure that this is going to be very, very useful. CDFD is leading this program. So with that, I thank all those who have contributed to this study, and of course funding has come from CSIR, UKRI, DBT, DST, and MRC. So thank you very much.
Thank you, Dr. Kangaraj. I think you gave us a very bigger perspective of genetics, because when you talk about genetic counseling is one-to-one, but you brought the concept of probably Vasudeva Kutumbakam, that whole word in this family is what I understand.
Just few seconds I want to tell you, because there's not much time, I didn't elaborate. So now what we have approached the Tamil Nadu state government to give us the permission to screen the entire individual, at least people with reproductive age, children, leaving the elder one, to understand the frequency of the mutation and also subsequent counseling to the individual. So that's where the TICs also will be part of it, to going there, giving them appropriate counseling. So if we do that, we wanted to create this as model for the rest of the study. We can screen and give appropriate advice for at least next 3 generation. We can able to eliminate the mutation from the population. So Ashkenazi Jewish have shown this. They have organization called Doreshim. So they have done and eliminated a couple of mutations from the population. So as a scientist, as a head of this organization, whatever the best I could do during my tenures, I have done, and this gives me more satisfaction. Thank you.
Dr. Kangaraj, just one question for the genetic counselors here. As you know, in our community, we sort of in India, lots of marriages within the community as such. And again, we see even in muscular dystrophy, so much of autosomal recessive. So is there any key points you would like the genetic counselors to have in mind during the genetic counseling? Because it's generally the program which comes, and of course then we take it to the triome for the parents, and of course the pedigree is completely drawn.
Correct.
From your experience for the genetic counselors, is there something that they should keep in mind while doing the pedigree and then counseling after that?
So that's the reason I was telling the population history is one of the important point to understand the population history. And one important point about mitochondrial disorders are now mitochondrial disorders are not only caused by mitochondrial DNA or mitochondrial dysfunctions, also caused by the nuclear genes. So if you have the perspective of the mitochondrial disease, how come only from the mother, so that's changed now. So father also can contribute the mitochondrial disease. So keep it very broad idea of all these. And maybe wherever there's a doubt, you can always discuss among yourself and discuss with others.
One question from the audience. We'll do it again in the panel? Okay. Okay then. Sure. Yes. Thank you so much, Dr. Tangaraj. Let's give a nice round of applause to Dr. Tangaraj. Thank you. Next, we'll be having Dr. Maya Tangavelu, and she's not new. And to say, as Dr. Ami rightly mentioned yesterday, she has been such a motivating factor for the startup of BGCI in India. And I still remember the first day I met her in Chennai. And now we are going to have her talk. And she basically had a graduation from Delhi, and then did her PhD and ABMG and FECMG, and medical director of cytogenetic services and technical director for fish services. So she has studied and expanded on human genetics and cytology in USA, Canada, and Denmark. She has been a professor of several universities in the USA and currently volunteering clinical faculty. She has been with the Department of Pediatrics, Genetics in University of California. And she has a membership of American Society of Human Genetics, American Board of Medical Genetics, and American Society of Hematology. She has been a reviewer of the following journals: Gene Chromosomes and Cancer, International Journal of Laboratory Hematology, Obstetric Gynecology International, and several more. And she has more than 100 publications, abstracts, and chapter in books. More than 40 years of experience in cytology, microarray molecular techniques, carcinogenicity of drugs, and again in teaching. Has collaborated on international stage for research activities and academic activities, and more for us. She's so special because she has been instrumental in starting BGCI in India. Welcome, Dr. Maya.
would like to thank Dr. Hassan for giving me this opportunity to speak from California. This year's topic, genomic variants in personalized healthcare, genetic counselors as facilitators, is an important topic in the practice of clinical genetics and genetic counseling in the era of genomic medicine. As you are all aware, genetic counselors play an important role in the field of clinical genetics. The role of genetic counselors has evolved over the years with evolution of technology used in the diagnosis of genetic disorders and disorders with a genetic component. The various phases in this evolution may be described as those in which most disorders had a cytogenetic basis. Then came the phase during which single gene abnormalities prior to the identification of the genes involved and was based entirely on family history. Then came the phase of single gene abnormalities post-identification of the specific genes involved, and finally the phase after gene mapping and sequencing of the human genome. Some tasks have remained the same through this evolution. Tasks like gathering family history, recommending appropriate testing, explaining the clinical and laboratory findings, calculating and conveying recurrence risks and risks for other family members, and explaining limitations of the diagnostic assays. Since the mapping and sequencing of the human genome, information from sequencing is used for evaluating cases suspected of having a genetic disorder. In the early days of clinical genetics, if the available diagnostic testing did not offer an answer, many families had to wait years before receiving the diagnosis. The average period of wait was between 5 to 6 years. With mapping of the human genome in the early 2000s, sequencing was introduced as a diagnostic tool. Sequencing has reduced the wait time for families to receive a diagnosis. This is especially critical in cases in the neonatal intensive care unit where critical decisions have to be made. Genome sequencing is now recommended as the first line of testing for a variety of conditions like intellectual disability and developmental disability. Because many features are common to many genetic disorders, sequencing is more efficient compared to tiered or sequential testing. Some of the assays used in gene-level testing are microarray, genome sequencing, exome sequencing, and multi-gene panels. Multi-gene panels are based on phenotype. For instance, panels to evaluate congenital heart defect will have genes known for syndromes for which congenital heart defect is one feature. This is a very efficient process to come to a diagnosis. Genome sequencing assays are incorporated into diagnosis across many specialties, including prenatal, pediatric, hereditary cancer, cardiovascular, and other subspecialties. This approach has improved the diagnostic process by reducing time to diagnosis, eliminating the tiered testing process, and avoiding phenotype-based ascertainment bias. You all may have yourself ordered many of these studies. The human genome contains approximately 4 million variants. When the genome is evaluated at this high resolution offered by sequencing, a number of variants will be detected. These variants are either structural, single nucleotide polymorphisms, insertions and deletions, copy number variants, and translocations and inversions. In cytogenetics, when one encounters a potential variant, like an apparently balanced translocation or inversion, parental karyotypes are useful to some extent in determining its clinical significance. At the nucleotide level, when one comes across a de novo variant, its significance will depend on if it has been seen before in a normal individual or an individual with a clinical phenotype. Based on this kind of information, variants are classified into pathogenic, likely pathogenic, variant of uncertain significance, likely benign, or benign. A significant number of genomic variants cannot be easily interpreted with regard to clinical significance. Variant interpretation guidelines, databases, and other resources have been developed to help with the interpretation process. Expertise and time are required for interpreting the variants that one encounters. One tool used to interpret the significance of variants are reference population databases. They aid in the identification of pathogenic variants and benign variation present in every human genome and supports the discovery of new disease-gene relationships. Because some variants can be ethnic-specific and benign, with movement of population across the globe, reference population databases are essential for appropriate interpretation. A variety of databases are available to aid in the interpretation of variants. One kind of database are the population databases. Some of them are shown here. In 2010, the 1,000 Genomes Project data was published. In 2013, the Exome Sequencing Project, which was made up of data from 6,500 European and African American individuals, was published. And then in 2014, the first large-scale aggregation of existing sequence data from 60,000 individuals was released. This particular database, which is known as NOMAD, has data from more than 195,000 individuals and is the most widely accessed reference population dataset. There are some databases that are more disease-specific, like the one from the National Heart, Lung, and Blood Institute, that the aim of which is to improve scientific understanding of the fundamental biological processes that underlie heart, lung, blood, and sleep disorders. And then there are other databases that are healthcare system specific, like the Dysinger system, healthcare system. They have their own database, which is made up of information, including sequences from participants from their population of patients that they provide services to. INVAR is a freely accessible public archive of reports of human variations, classified the diseases and drug responses with supporting evidence. And INVAR collects data about genotype-phenotype relationships from many contributors. And this is one of the databases that many clinical entities use for interpreting the data that the variants that they come across. Currently, there is a database that is being developed by the National Institutes of Health. The aim is to enroll 1 million participants. And in this, they are not only collecting the whole genome sequence of these participants, but also the health data that is continuously collected from the electronic medical record system, and also looking at environmental data and physical measures, etc. So this is like an ongoing project which involves not only the genome, but also the different healthcare, the different health issues that the participants may encounter over a long period. Moving to the role of genetic counselors in variant interpretation. Genetic counselors have been involved in interpreting variants in labs that do the sequencing. But since there are established principles for variant data curation, patient-facing genetic counselors can use these principles to interpret the variants themselves if they wanted to. Now, why would genetic counselors want to be involved, that is, why would patient-facing genetic counselors want to be involved in variant interpretation? One of the reasons is that discrepancies have been encountered, and these discrepancies are sometimes between labs. There will be differences in how the interpretation is, how the variants are being interpreted, and also between the lab and the clinical team. For example, in the cardiovascular genetic system, discrepancies in variant interpretation between the genetic counselor-led clinical team and the lab has been estimated to occur in 18 to 19% of the reported variants. So it's important for genetic counselors to have some amount of skill and expertise in evaluating the results that they get from the labs that provide the data from sequencing. There is also a new profession known as variant scientists, and many genetic counselors who have developed the expertise and skills in interpreting variants are becoming what is known as variant scientists. There are also some certification programs in which provides certification not only for genetic counseling, but also for variant interpretation. So genome sequencing will become part of the diagnostic process in the many cases of rare diseases as well as disorders with a genetic component. So genetic counselors have to be equipped to evaluate and understand the information in the reports so that they can discuss discrepancies with the testing lab, and they can also appropriately convey the information to patients and families, which also involves reclassification of the significance of the variants. That is, sometimes with accumulation of additional data, the significance of a variant can change from, for example, saying likely pathogenic to maybe becoming a variant of uncertain significance. So, and because of these reasons, variant interpretation should be a part of training of genetic counselors. Thank you for your attention, and once again, thank you, Dr. Hassan, for giving me this opportunity.
Thank you, Dr. Maya, for the overview. Because of the time constraint, I call upon our next speaker, Dr. Srinivas Murki. He is the chief neonatologist at Paramita Children's Hospital, Kotapet. He is a DM neonatology from PGIMR, Chandigarh. He has more than 150 Indian and international publications. He is an MD and DM gold medal, past president of NNF Telangana, joint secretary NNF India. His major areas of interest and research focus involves teaching, innovator of MURCs, TPs, cost-effective care, CPAP, mother's own milk, etc. Thank you, doctor.
Thank you.
Hello. Yeah. So this is a real pleasure to be among a different group of delegates. I happen to be mostly among pediatricians, neonatologists, and obstetricians. Today I'm among a different breed, and I find myself isolated. age, he was admitted previously to a different hospital. He was still then exclusively breastfed. He was diagnosed to be GERD's reflex disease, and they thought it is the failure is because of reflex disease. Yeah, thank you. They thought the failure is because of reflex disease, and they treated for with anti-reflex medicine. But again, at 2 months, he went to another hospital where he was treated for severe anemia. He received blood transfusions. That time he was noted to have jaundice and pale color stools. So meaning that he was having a cholestatic jaundice, and he also had a liver which was significant. Sorry. Okay. He weighed less than 1 centile, head circumference low, length is low, so he was definitely failing to thrive. He had pallor, icterus. There was no dysmorphism and no cataract, meaning that there was no sign of galactosemia. Liver was enlarged. Hemoglobin was 8 grams, less than for that age. Retic count is high, that means there is some amount of bone marrow activity going on. There is no hemolysis or immune hemolysis showing that DCT is negative. The blood counts were normal, platelets were normal. Bilirubin was elevated, which was conjugated, so this was a cholestatic jaundice, and the enzymes were elevated, meaning that there was hepatitis. There was also prolonged PT-APTT, meaning there was liver cell failure. So this child actually had liver cell failure with anemia at 2 months of age. So this impression was failure to thrive, anemia, blood transfusion, cholestasis, hepatitis, and no liver cell failure. So we thought of infections, because in our country infections are the commonest reasons for this, but bacterial infections, screen was negative, blood culture was sterile. Malaria can present, but smear was negative. This baby didn't have any other manifestations. Pallor, cholestasis, anemia, we think of malaria. Extrahepatic biliary atresia is a commonest cause of cholestasis. Ultrasound was not suggestive. Galactosemia, which is relevant to most of the audience here. Urine reducing substance were positive. So if your urine reducing substance are positive in a galactosemia, in a cholestatic, we think it's galactosemia. But blood sugars were normal, there was no hypoglycemia. The other thing which was considered was secondary HLH. The ferritin was very high, fibrinogen was very low, and triglycerides also were very high. So this was telling that there could be galactosemia, there could be an infection. So thinking galactosemia, secondary infection, and probably secondary HLH, this baby was started on antibiotic, lactose-free milk. IVIG was given considering secondary HLH. Baby continued to worsen over the next 2 hours. To our surprise, he was not responding to galactose-free diet as well as for. So our diagnosis of galactosemia didn't look appealing for the baby, and IVIG did not work. So HLH also didn't work. The baby went on to develop seizures, encephalopathy. Then he started having increasing liver spleen size. He had multi-organ dysfunction, edema of feet, cardiac dysfunction, and then pulmonary hemorrhage, and succumbed. Patients required for withdrawal of support, and the baby succumbed. They did not accept for a liver biopsy while the child was alive. The parents refused liver biopsy, so we couldn't get a liver biopsy, but they agreed for exome sequencing, and then we could get the exome sequencing. Liver biopsy postmortem has shown macrovesicular steatosis with cholestasis and grade 1 iron stores. There is no evidence of cirrhosis. IM was suspected with these possibilities: galactosemia, tyrosinemia, and cystic fibrosis. So 3 possibilities there from the liver biopsy. This is what the report came as to be. No pathogenic or likely pathogenic variant. Summary of findings: variant potentially relevant to index. The index patient is negative for the disease. Secondary finding: no pathogenic variant, but findings unrelated to the phenotype. We were surprised why they give this, because the phenotype was cystic fibrosis. One of the differential is cystic fibrosis. Cystic fibrosis can present as liver cell failure in the newborns, but it's a rare presentation. But it helped us because we could go back to the lab and said that this will fit into cystic fibrosis because the genotype, liver biopsy, and the genotype are fitting. So the parents were very happy to know that at least they know the cause. We told them galactosemia, we told them HLH, we gave them treatment saying it will definitely improve. The baby didn't improve. So, but once they heard this, once we showed this, we went back to the lab and they said, okay, now it is turned out to be only a cystic fibrosis, which can be diagnosed prenatally. At least for the next pregnancy, they can save the child. So what I said was, why it came out to be negative for pathogenic variant, I don't know. Maybe you can let me know why it has happened that way. I give you liver cell failure. So, yeah. So again, liver cell failure in a newborn because of cystic fibrosis is rare. So maybe that is why they would have thought, because cystic fibrosis causes more of lung related issues or failure to thrive leading to steatoria. So that might be the reason. This is another child, 6-month-old girl child. Again, I am taking mostly jaundice cases because this is the time where we send most of the genetic alleles. Jaundice and rash all over the body. This is born to consanguineous parents. Term baby, exclusive breastfed, no encephalopathy, no bleeding tendency. Rash was started at 3 months of age. The rash progressed from face to trunk and limbs. Infant was always irritable, was noted to have jaundice. Noticed 1 week ago, but the rash was there for 3 months. Stools were acholic. When you direct question the parents, they're saying that the stool were always white. So vitals were normal, there was no dysmorphism. Weight, you can see, is not gaining so much of weight. Head circumference length looks reasonably okay. He had scratch marks all over the body and multiple ulcers. There was significant pallor and icterus. Liver was enlarged. This is the baby. You can see the rash and the cholestasis in this baby. Since the cholestasis was very mild, the parents didn't realize that there was problem related to the liver. The bilirubin was elevated. Liver enzymes were not much elevated. GGT was low. This is a diagnostic clue for us. And albumin was normal. That means there is no low chronic liver disease. This was cholestasis with low GGT. Serum bile acids were elevated. Urine reducing substance were all again positive, thinking that maybe it is galactosemia, but galactosemia will not present like cholestasis like this. Ultrasound abdomen did not show any cholestatic or obstructive cause. So we could make a clinical diagnosis that this was a classical case of PFIC, progressive familial intrahepatic cholestasis. And thyroid was done as a routine. So cholestatic jaundice, no liver cell failure, age of onset rash, which was clinically confirmative of a progressive familial intrahepatic cholestasis. So this, for confirmation, you have sent the exome sequencing, and the child has responded to therapy which is given for cholestasis and PFIC. He responded dramatically. Next 2-3 months later, the parents come, the cholestasis improved, the rashes have gone off, everything has become normal. So this is the genetic report. It says variant in gene known to be associated with provided phenotype, but what we have is heterozygous, and it's given as variant of unknown significance, and it's an autosomal recessive disorder. So I don't know how this baby can have PFIC and heterozygous mutation. And read the line below. The second heterozygous variant is not identified in the gene for an autosomal recessive disorder due to the sensitivity of NGS testing. So hence detection by the second heterozygous variant by alternative method is suggested. So most likely they wanted us to do a Sanger or repeat the test. Deletion of this. So maybe for us it is again convincing the patient to get another exam sequencing because child has responded. And clinically we are saying it is PFIC. So I don't know why it was missed on the first attempt looking at the second allele, why they could not look at the second thing on the single investigation itself. Additional finding, there is congenital disorder of glycosylation which doesn't fit into because it was heterozygous anyway. This is the third case. So first case, we couldn't make a diagnosis. Genes made the diagnosis, but confusion created. The second, we made the diagnosis. Genetic made us tell you need to further do something else to confirm the diagnosis. This is the third case. This is the baby, 2.5 month old boy, born to primary mother, non-consanguineous parents. Vaginal delivery, normal Apgars, birth weight is 2.25, IUGR baby. On breastfeeds from birth, both the parents had low IQ, they were not evaluated. 1 month baby gets admitted for poor weight gain and hypoglycemia. At 2 months he gets admitted for dull activity, anemia gain, jaundice significant, cholestatic jaundice, seizures due to hypoglycemia, currently referred for persistent cholestatic jaundice. So the baby was lethargic. He had decreased activity, signs of severe dehydration. You can see the baby there. He's lost a lot of his weight. Pallor was there, icterus was there. He had relatively normal vitals. Blood pressures were normal. Liver was enlarged. There was no other organomegaly. On investigations, we can see that hemoglobin was low. He had already received one transfusion before admission. Infection workup was positive. Jaundice was saying that there was a liver disorder, hepatitis, enzymes were elevated, and there was no liver cell failure. GGT was normal, electrolytes were normal, thyroid was normal, ammonia was normal, lactate was normal, urine reducing substance was again positive in this baby. And most of the time, if you have liver disease, urine reducing substance can become as false positive. So, however, because treatable condition should be always be considered, this baby was started on lactose-free diet. ENBS was sent, and IRT was normal, meaning that at least in the screening, cystic fibrosis was negative. Serum bile acids were normal, elevated. Ultrasound was normal. So this was a child again with failure to thrive, severe dehydration, cholestatic jaundice, probably an infection, anemia, urine reducing substance were positive, and this child responded to galactose-free diet. He's improved dramatically after starting in. All the things come out saying that probably he had a liver disease related to lactose. So the baby improved, and we were almost sure that it will turn out to be galactosemia. Now the genetic came out like this. So what the gen says, it's a heterozygous autosomal dominant and intellectual developmental disorder. So I feel maybe this is something to relate it to the parents. Now there may be a need for evaluating the parents to look at this, because both the parents must be harboring this variant that is causing intellectual developmental disorder, and probably that helped us to make another diagnosis in the baby. Although the child has improved out of cholestasis, but he needs evaluation for the IQ because he's too young to be evaluated for development or IQ. So, but I feel this has helped us to tell that, okay, you have a child who can be affected, maybe we can work on the child from early, and they can also plan their next pregnancy appropriately because we know that this child has given them a diagnosis. This is another child, 36-day-old baby, born to non-consanguineous couple. There was oligohydramnios for which they did a section. No significant perinatal events, breastfeeding from birth, and was referred to us for disorder of sexual differentiation. And whenever disorder of sexual differentiation is there, we are worried. And these children come, this child came to us with vomiting, decreased oral intake. On examination, the child was irritable, there was clitoromegaly, there was no palpable gonads, and the baby lost weight. There was tachycardia, temperature was normal, blood sugars were normal. You can see classical clitoromegaly, and the gonads are not palpable. If a child presents like that with failure and with vomitings and dehydration and clitoromegaly, we are almost sure it turns out to be a classical case of congenital hyperplasia. So that's what we thought, and then we evaluated for that. Admission, hemoglobin was normal. There was no infection. As expected, the electrolytes were suggesting that this is a case of CAH because hyponatremia, hyperkalemia, and the other things also thought that it is a case of CAH. Now, blood culture came sterile, blood gas, metabolic acidosis. Unfortunately, the cortisol and aldosterone came as to be very high, which is not tenable with a diagnosis of CAH. In CAH, cortisol levels are low, aldosterone is low, and hypoglycemia, and 17 OHP should be high. But here you have 17 OHP high and cortisol and aldosterone are high. So we thought another differential of pseudo hypoaldosteronism, which also have a genetic association, but we cannot explain the phenotype. We can explain the shock and vomiting, but we cannot explain the genitals. If it is not CAH, how can we have the genitals like that? So pseudo hypoaldosteronism does not have ambiguous genitalia or DSD. So what we thought, maybe genetics will give us a clue, and then we send a genetic evaluation. The management was fluid with hydrocortisone fluid. The child improved with that because he had definitely hormonal imbalance, and with therapy he improved. Ultrasound has shown visible uterus, saying that it was a girl and a female pseudo hermaphrodite or XX DSD. The baby was discharged on day 5 on medications, normal electrolytes and normal weight gain. So our differentials were XXDSD or XXDSD with pseudohypoaldosterone because the aldosterone and the cortisol were very high. So this is the chromosome which again said that it's XX. So probably we were thinking that okay, it will turn out to be CH. Now what came out was no pathogenic or likely pathogenic variant causative to the reportive phenotype is what came out to be. And this was a shock to us because we thought it is going to be 100% CH, but genetics said that it's not so. Then we got opinion from our genetist, and what they said is we need to go ahead with a targeted sequencing after selective amplification of CYP21A2 gene with MLPA. So we have ordered for this. Maybe we'll get the report of the CAH on the subsequent reports. We are not sure. We have still not got the reports, but I hope that some diagnosis will come out of this child. So this is the last case. Baby K, 19-day-old, born to second degree consanguineous parents, normal vaginal delivery, and last, I'll finish. Yeah. So on day 7, the baby was admitted with cholestatic jaundice. He also had bleeding. So there was liver cell failure, hepatitis. So liver cell failure, bleeding, cholestatic jaundice was referred to us on day 19 for persistent bleeding and worsening of cholestasis. The child looked active, and there was no dysmorphism. Liver was enlarged, and investigation showed anemia, cholestasis, and liver cell failure. Urine reducing substance was again positive, and ferritin was high, but other features of HLH were not there. So, sorry. So the girl child, cholestatic jaundice, acute liver cell failure, urine reducing substance positive. So child was put on lactose-free diet. He's improved because the lactose-free diet. Again, we thought it is galactosemia, classical galactosemia, because he had bleeding, he had liver cell failure. Everything improved with the therapy. So for diagnosis, nowadays they're not doing GALT. I don't know, for some reason. We can't get a gene for GALT, but they'll do genome sequencing, but not GALT. So they did a genome. I don't know, maybe you can let me know why they're not doing GALT in the labs. So this was the genotype. They said it is homozygous for galactosemia. Yes, at least now the last case, the phenotype matched with the genotype, and definitely it is going to help the baby because the outcome is going to be reasonably good. Early diagnosis, galactose-free diet, these children do very well. So just to summarize, case 1, liver cell failure. We didn't think of clinically cystic fibrosis, but genetic has helped us to make a diagnosis and then comfort the parents at least for the next pregnancy. Second case, PFIC. We are sure it is PFIC, but probably that heterozygous mutation is worrying us. Maybe subsequent investigation can help us to confirm. The third case is a clinical mismatch because clinically we couldn't make a diagnosis proper, and then genetic also didn't help us. Maybe subsequent evaluation is going to help us. Case 4, case 3, that clinical CAH, genetic mismatch, where the parents had IQ problems, and then we got a new diagnosis of related to IQ disorder in the baby. And case 5, this is the perfect match, but they've given variant of unknown significance. That is what I wanted to tell you, that they've given us variants of unknown significance where clinically it is galactosemia, and they said it is galactosemia. Probably the variant is not diagnosed previously, that's why they gave variants of unknown significance. But maybe next time this will be put in the database and it will come as pathogenic. Thank you. Yeah, so now we did tell them that it is matching, so most likely they'll include the database, and then probably next child will benefit out of it. Yeah. Thank you.
Thank you, Dr. Srinivas. Because of the time, we may not take any questions. Panel discussion will take it at the later. Yeah.
Thank you, Dr. Srinivas. Next, we'll have Professor Dr. Zulfalil, and I am very happy to introduce him because I share the board of Asia Pacific Society of Human Genetics, where Dr. Zulfalil is there. It's the second time we are together. Welcome, Doctor. And to introduce him, he's one of the members of the WHO Technical Advisory Group on Genomics. He serves as a consultant pediatrician, clinical geneticist, and lecturer at the School of Medical Sciences at the University Science Malaysia. He's trained as a pediatrician and holds M.Med in pediatrics from the University Science Malaysia, MSc in medical genetics from the University of Glasgow, and PhD in pharmacogenetics from the University of Aston, UK. Professor Zulfaril has decades of experience and has greatly contributed to the study of rare diseases among the Malay population and includes his participation in the Human Variant Project and the Global Network. As a testament to his contributions to the field, he was also awarded the UNESCO Chair on Human Genetics of Thalassemia in 2022. Welcome, Dr. Zulfaril.
First of all, I'd like to thank the organizing committee for inviting me to give this lecture, especially to my dear friend, Dr. Amy Hassan. Now, the topic of my lecture is Global Building Network, and I will share with you the work that I've done on hemoglobinopathies, especially on thalassemia, over the last 10 years. This is my disclaimer. Now, I would like to begin my lecture by highlighting the amazing ability of our human DNA to change, to adapt, and to evolve in order for humans to survive. Now, if we look at the global distribution of malaria and we compare that to the global distribution of hemoglobinopathies, we will see a similarity. In order for humans to survive the invasion of the malaria parasite into their red blood cell, it changes the structure of the red blood cells to make it less favorable for the malaria parasite to thrive and to multiply within the RBC. Now, while humans survive malaria, they have created a new disease, which we now know as hemoglobinopathy, including thalassemia. Now, if we look at the countries where malaria, hemoglobinopathies are endemic, we will notice that these are also countries in which the World Bank categorized as low and middle income countries. And these are also countries where the health systems are weak and are unable to cope with the economic burden of the disease. Now, the global burden of the disease is huge. More than 300,000 affected infants are born annually, and up to 100,000 children with thalassemia major die each year in the LMICs. Now, hemoglobin disorders contribute the equivalent of 3.4% of mortality in children under the age of 5 years worldwide, and more than 6% in Africa. The economic burden too is huge. Malaysia, for example, spends about 100,000 ringgit per year to treat one thalassemia major patient. Or, for the average lifespan of 50 years, the Malaysian government spends about 5 million ringgit, or about 1.2 million US dollar to treat one thalassemia major patient over his or her lifespan. Similarly, in the United Kingdom, the estimated total healthcare expenditure for the management of thalassemia is about 720,000 US dollar for the first 50 years of a patient's life. So the economic burden is huge. So these low and middle income countries where thalassemia and other hemoglobinopathies forms a major health problem, they find it very difficult to cope with the economic burden of the disease. Now against this background, the Global Globin Network was established about 10 years ago. It was officially launched in 2015 and currently it has more than 80 members from 52 countries. The focus is on hemoglobinopathies in low and middle income countries by strengthening education and research in the human genetics of thalassemia. And the focus is on building capacity for genomic diagnosis and clinical services and research, and to share all the relevant genetic data between the member countries. For better planning and progress among these 32 GGN member countries, a stratified roadmap for improved care and prevention was established, and this was based on the following 9 areas. First, a prevention program where the disease is available in the country, blood transfusion availability, dedicated treatment centers, iron chelation, hemoglobinopathies patient registry, patients association, prenatal screening, antenatal screening, and genetic counseling, whether all these are available in the country. Now, based on the 9 areas, a stratified roadmap for improved thalassemia care and prevention for the 52 GGN member countries were established. And these 52 countries were divided into 4 groups. Group A are countries where services are well established with a national system for prevention and control. 6 countries are categorized into Group A. And Group B are countries where efforts of a partial or fragmented national control program are in place, but with limited accessibility. 16 countries are categorized into Group B, including India. And Group C are countries where expertise on hemoglobinopathy data collection and management exists, but it's not part of a sustainable national control program. And 7 countries are categorized into Group C. And Group D are countries where expertise and infrastructure for hemoglobinopathy data collection and management are limited, and 3 countries are categorized into Group D. If you are interested to know more about this stratified roadmap and the work on GGN, you are most welcome to read our paper, which we published about 2 years ago. Now, this is just to highlight the activities that we have organized over the years. And the last activity that we organized was in 2019, where we held our annual meeting at the UNESCO headquarters in Paris. Now, the Thalassemia International Federation, or TIF, they have recommended the prevention policies, which includes the following key components. First, a national strategy on prevention, which suggests government approval, control, and support. 2, public awareness program. 3, screening program to identify carriers. 4, genetic counseling services. 5, prenatal diagnosis as a choice for at-risk couples. 6, pre-implantation genetic diagnosis. And 7, the application of new emerging technologies in the management of the disease. Now, if you look at those policies recommended by TIF and compare that to those recommended by the Global Globin Network, we will see similarities. And this includes the prevention program, the screening program, genetic counseling services, prenatal antenatal diagnostics. Now, one of the policies recommended by TIF as well as by the Global Global Network is on genetic counseling. I believe genetic counseling should be made available in countries where thalassemia and other hemoglobinopathies are endemic. Genetic counseling should be made available before marriage, after marriage, or when patient is already pregnant. And patients should be given a choice either to avoid marrying another carrier or to separate from a relationship that puts their future children at risk, and to marry their chosen partner with the knowledge of the risk involved. And couples should also be given choices either to proceed with the pregnancy, accepting the risk of possibly bearing an affected child, or to avoid having any children, and to undergo prenatal diagnosis or antenatal diagnosis, as well as pre-implantation genetic diagnosis. Now, all patients and their families should have equal access to quality health care and ultimately contribute to reducing the thalassemia burden. Now, with a good, effective prevention program in place, we can see the number of new births or new cases being reduced. Cyprus and Singapore have shown that with a good, effective prevention program in their country, they were able to reduce the number of new births or new cases in their country. Malaysia, too, from the year 2015, have seen a reduction in the number of birth of new cases. At present, the mainstay in the treatment of thalassemia includes regular blood transfusion followed by iron chelation therapy. And in some centers, bone marrow transplantation are being offered. But I would just like to highlight the availability of new modalities of management of thalassemia, and this includes gene therapy and also the pre-implantation genetic testing. Pre-implantation genetic testing, or PGT, offers several advantages for couples who are carriers or inherited diseases such as thalassemia. It allows couples to make informed decisions about their pregnancy. It allows them to select the healthiest embryo free of the disease. It enables couples with an inheritable genetic conditions to avoid passing it on to their children. For couples who are carriers of monogenic diseases such as thalassemia, PGTN is usually offered to them. Now, gene therapy is now available for thalassemia as well as sickle cell disease. There are 3 drugs that have been approved by the U.S. Food and Drug Administration, or the FDA. The first to be approved in 2022 was Zinfaglo, which uses the gene addition method using the lentiviral vectors for the treatment of beta thalassemia. And in 2023, FDA approved another drug, the Lyfgenia, which also uses gene addition method using the lentiviral vectors for the treatment of sickle cell disease. And latest to be approved was Casgevy, which works through gene editing technology using the CRISPR-Cas9 method. Casgevy works by using the gene editing technology, the CRISPR-Cas9. It can be used to treat both beta thalassemia as well as sickle cell disease. Now, using CRISPR-Cas9, it added the BCL11A gene. Now, the BCL11A is a gene that tells the body to make less fetal hemoglobin and more adult hemoglobin. So by reducing the activity of the gene, the body can then make more fetal hemoglobin. So when the BCL11A gene is edited, the body can then make more fetal hemoglobin, and this will help the body to make more working RBC. Now the advantage of CAPGV is that it uses patients' own blood stem cells that are collected and sent for editing. Therefore, no donor is needed. Now, Zinteglo and Lyfgenia rely on gene edition in the red blood cell instead of gene editing. Zinteglo works by adding working copies of the beta-globin gene to the collected stem cell. Now, the modified stem cells that are collected from the patient's body are then returned through an intravenous infusion. So both methods, Zinteglo as well as CAPGV, uses patient's own blood stem cell, and therefore no donor is needed. So genomics is the way forward. Genomics can be used to improve the effectiveness of thalassemia management. Now, for genomic medicine to take place, it requires molecular profiling of all the patients. Molecular profiling lays the foundation of genomic medicine. We know that not all thalassemia patients will require a molecular diagnosis. Thalassemia can be diagnosed by a full blood picture and a HD analysis. Not all patients would have their molecular diagnosis done. However, for genomic medicine to take place, all these patients will require a molecular profile. Now, for genomic medicine in thalassemia to take place, all the variants that are related to the thalassemia gene needs to be annotated. Now, to answer the clinical relevance of variants for its use in precision medicine, clinical annotation is required. Now, we know there are an increasing number of sequence variants which are still of unknown significance, and the process for the clinical annotation of these variants is ongoing. Now, ClinGen, which is an authoritative central resource under the NIH, interprets the genes and its variants for the use in precision medicine. And to do this, it forms the Variant Creation Expert Panel, or the VCEP. And currently, there are more than 60 VCEPs that have been formed to annotate variants for various diseases. For thalassemia, the VCEP has recently been established, and currently the work for the annotation of the thalassemia genes and variants is ongoing. Now, the program for genomic medicine or precision medicine of thalassemia also involves understanding the role of genetic modifiers. In order to understand the role of genetic modifiers, Global Globin Network works with Inherent, which is the International Hemoglobinopathy Research Network based in Cyprus and led by Dr. Petros Fountaris, to study the role of genetic modifiers using large-scale multi-ethnic genome-wide association studies. And the goal is to provide a resource of genomic variance data, which will be the platform for precision medicine in thalassemia. Now, in addition, the Global Globin Network also works with Ethernet, another organization based in Cyprus, in the formation of a database for thalassemia and other hemoglobinopathies. In summary, to prepare the foundation for precision medicine of thalassemia, Global Globin Network works in collaboration with the following three organizations: the Ethernet, ClinGen, and in heaven. So the way forward and the future for thalassemia is in genomics. Now this is my last slide. So the take-home message is genomics is for all and no one should be left behind. And this includes the thalassemia patients in Malaysia as well as in India. Now I'd like to acknowledge the following organizations as well as all the members of the Global Globing Network.
Thank you so much, sir. Can you please stay with us as we continue the panel discussion? May we have Dr. Tangaraj and Dr. Murthy on the stage, please?
I would like to request our volunteers to make the arrangements on stage. And I'll humbly request our chairpersons to stay back on the stage. Yeah.
That's all. We need only 4 chairs. We need only 4 chairs. We don't need to be. So 20 minutes of the speaker's time is never enough, and we always want to learn more from them and know more from them. So this is one of the ways where we break into a conversation as we ask questions. The audience is free to ask questions when we let them. Okay. So we have a question for Dr. Tangaraj. What role can genetic counselors play in public health platforms, sir? Can we have your words, please?
Yeah, genetic counselors always interact with public, so there's a huge potential for the public in terms of conveying the right message to the people.
Public health
And we were actually discussing in the morning, some of our friends, they say that genetic counselors should play the role appropriately, not thinking of anything else. Conveying the right message. So one of the important aspect which they can convey to the public is that understanding the family history and appropriately conveying, whether they take it or not, but give full effort for conveying the information. So that is what is important. And before that, understand as some of the points which I mentioned during my talk, consider those points to think what needs to be conveyed.
Yes, sir.
Thank you.
Thank you so much, sir. So genetic counselors.
Yes.
The project, have you thought of involving genetic counselors in that? Because you are doing the sequencing, you are finding variants, you are collecting samples, and in all this, anywhere else in the world, large population projects have the involvement of genetic counselors.
So this particular project which I mentioned, sequencing up 10,000 individuals, they're all normal, supposed to be normal, right? And so we are not sure that what kind of disease they are going to have, right? That's true. But our part is going to over by sequencing, analyzing, and leave that to the public. Okay, so there's no, although there is a potential point to be conveyed to the public, but we don't have the real problem with the individuals where we collected the sample. Therefore, hiring during that 5-year period, not even 5 years, a 3-year period, of sequencing 10,000 individual. So we did not thought about that. That's how it's going to help them.
So to add on here, you told about the project in Kammanahalli where you're going to eliminate things. So do you have genetic counselors involved in the project?
Yes. So we thought about it, TICS as the genetic counselor. So we are going to go with the genetic counselor.
Thank you so much. Thank you.
I think Usha want to ask.
From such a national institute, okay, even if it is the project for the normal individuals, and that's why you may be thinking the geneticists, genetic counselors are not required, keep it in mind, keep it on the project thing, that genetic counselors as a team of yours. When there will be genetic diseases or when the genetic disorders are found and discovered, and then, to put it on a paper, it becomes very important when it comes from the institution like you.
No, you know very well how the funding agency works, right? Even if you wanted to put it, they will cut it.
Dr. Kangaraj, I think in forums and as this is the 9th year, somewhere I also understand on the other side, just like that we cannot give in for the genetic counselor. There need to be some sort of hand holding when you say normal patients. So genetic counselors have to work hand in hand with people like you who are pioneering those projects and understand their state, but their role is slowly to be established with. But I'm sure people like you who have served and contributed so much, if you put down to say genetic counselors are important, it makes a lot of difference.
So I understood my MPhil degree is in genetics, human genetics, PhD in human genetics, right? So after 35 years of my research, now looking at the population level, we have now reached a stage where we can, include something. Right? Yes, I convey that we are going to include when we need it, right? Yeah, of course. Sorry.
Yes.
Maybe ask the same question to Dr. Zulfalil ma'am.
Yes, please.
So would you like to comment on that?
Must we put it again to him? Must be.
Sir, excuse me, sir.
No, you call him. You ask.
Dr.
Professor Zulfalil, are you there?
Yes, I'm here.
Yes, that's a question.
I can hear you.
Yes, that's a question for you. It's Reena.
Sir, during all your projects, did you ever feel the need to involve genetic counselors during your projects on hemoglobinopathies?
So that's a very good question. Thank you. The project itself, it's more of it's not a particular research project, but more on the advocacy of hemoglobinopathies. Of course, I think when we advocate, thalassemia, sickle cell disease, as I mentioned in my lecture, we would require counseling. I think that is a policy set by the Thalassemia International Federation as well as from our group. Clearly, I think genetic counseling, plays a big role in the prevention of the disease. And I think countries where they have been successful in in combating hemoglobinopathies are also countries where genetic counseling are available. So yes, of course, the role of a genetic counselor in the prevention of thalassemia, in managing thalassemia and sickle cell disease, that's unquestionable. They play an important role. So Malaysia, for example, we are still, we have started training for genetic counselors. We have about, I can't remember the exact number, but more than 10 genetic counselors in the country. And the number is increasing because of, the training program that is being offered in the country. But this is still not enough, to provide adequate counseling for patients with thalassemia in the country. So what the government, the Ministry of Health in Malaysia, what they are doing, they are training the nurses, and providing them, short, courses of training not as a story, in counseling for families with thalassemia. So that is a temporary measure. But of course, I think in the long term, we would require genetic counselors to be available at all the levels in the country to provide a proper genetic counseling to the patients as well as their families. So I hope that answers your question.
Yes, sir. Thank you very much.
Thank you.
We have a question for Dr. Murki. Sir, one recommendation that you can give for a change in healthcare policies. If given a choice, what would you do?
So I think the world is changing, and then we are seeing therapy, gene therapy for thalassemia, sickle cell, and diagnostics have changed. So I think as all of you want it, I think wherever possible, I think all the hospitals which take care of the significant load like pediatric hospitals and also mother-related maternity hospitals should have genetic counselors. So I think that's what we can, at least I'm going to have one genetic counsel for my hospital. So I realize the need.
Teaching medical hospitals.
Medical hospital compulsory. I don't think there is any doubt about it. Medical hospital should not have, medical colleges should not have one, but should have a department of genetic with genetic counseling. But hospitals like ours, at least some 100-bedded hospitals, when they give permissions, so when they give permission for pediatrics and for maternity, I think they should include this as one of the prime requirements for granting permissions. And these permissions are not given from NMC, they are given from local bodies. So you can actually talk to the local bodies, and DMEs, DHEs who give the permissions, they can be influenced to ensure that these are part of the, because if I am starting a specialty hospital, I need to have gynae, I need to have anesthesia, I need to have resident doctors. So if I'm running 100-bedded hospitals which is related to pediatrics, I think genetic counsels can be one part of them. So that can be done. While I answer my question, I had one question for Dr. Tangaraj. It's so you said that there is a lot of population halotype which is similar across the populations, and you said marriages should not happen in the same area, same region, or same population. So do you encourage marriages from different regions, different population is one thing. The other thing is, have you studied, because the horoscopes which they are made, they have something which tells us that, okay, this is a lineage, that is why you have similar Gotram or something like that. So they never allow marriages among them. So could you relate that to whatever variants you are looking for?
Yes, very important. But whatever the findings which comes from our institute, we always try to convey that to public. We have a science communication officer, and that information is put on to all the newspapers, right? So after seeing that, there are many individuals contacted us, right, for more clarity and so on. So we always help them.
I want to add,
Good point.
it will become much more easier if you can relate it to the cultural thing. So if we can prove that whatever marriage counseling happens, they've all called it Brahmin or Pandit to make this thing. So I think at that point of time, there is a need for a genetic counselor to be part of that. And if we can bring that into a system, it is a needed thing. It is a needed thing, because still every marriage, at least in Hindu religion, cannot happen without matching of certain things. And I feel there is some science behind it. And the science could be that
We need to work out what is the science. And for that, I think Dr. Tangaraj, and then when you collect your database, I think you need to look at these things also, because that will help in giving evidence, scientific. You easily, they'll accept you easily. If anything which is culturally accepted, and if you try to promote it, more likely you'll be accepted by the community.
No, people are accepting it. So I can also give you suggestion that, so why don't you write to newspaper as a popular article to No, not necessarily. So if it is useful for the public, I think we should make awareness. So that's one of the point here.
So you should be the one, like Balasubramaniam used to write, articles. So you should, then people will listen.
No, maybe I'll keep this in mind. Whenever we publish next time, we'll add a line to contact.
Article also, they do contact us.
Right.
But I naturally try to look at the cultural sensitivity, but while just listening to you both, I got an idea that now that you get a child with whatever issue, ask them, did they have some Gotram which showed that they would have a good life? And we go back and see what's the story.
No, I think
We could do
Gotra,
that backwards also.
Gotra is followed very well in the northern part of India, where they don't marry within the Gotra.
Yeah.
That's what I heard. But we don't have that kind
No,
of.
but in North India also there's thalassemia, there's cystic fibrosis, there is tyrosinemia, there's galactosemia, all
Depending
different
upon.
things. So that's what, let's show
I
them
feel
that
some-
if, what is the historical
some background. There are certain mutations which are very old. As I mentioned, the myosin binding protein C3 mutation, we established that happened more than 33,000 years ago, before the admixture, right? Therefore, the admixture brought to equally 4.5%. Some mutation, if it arose after the population diverged. So that's what's going to be a population specific. So again, this is the point one has to understand how these mutations have evolved, where it is circulating and so on. So that's also another important point. So we cannot actually generalize any mutations. We have to look at the origin of the mutation, where it's prevalent. Accordingly, one has to give the counseling. Correct.
I have something to continue. My father had, have 6 sisters, and all of them have daughters. So everybody was saying that you should marry within the family. And coming from MBA, I said I'll not marry within the families, and went out comparing all the, horoscope, gotras, everything, but still encountered a problem. Okay.
That is maybe because of
Yeah.
maybe they have avoided something which has come after the hemoglobin happened after the selection of malaria. So you may have avoided many other.
Now all of them, they say, what the hell did you do? Okay.
So we
I have one more question. I think since we are talking about the policy, so whether any of these findings, studies, is it getting into a policy level with the government rather than discussing generally in public, directly to the public?
So maybe we are trying our level best to take it to the government, and there are some officials whom I know, they read our paper, interact. So one of the economic advisor to Prime Minister's office, he said that it was probably about a year ago. He said that I'll invite you one day, you make a presentation to Prime Minister, and we see how best we can do. But did not happen. I don't know. There are many restrictions within himself also. So I hope that it's slowly happening. And once the information is well known to the common people and might reach to the next level, and I'm sure that there could be some policy.
So we have one last question from Dr. Deepak, and that will be the end of the session.
I mean, we talk about consanguinity as one of the prime factors of inherited disease. But looking at the data that we have from India, the random chance of marrying somebody with a genetic mutation seems to be fairly high. Do we have an estimate of the load of genetic mutations, let's say autosomal recessive, which would occur just by a random chance, like as Sir discussed it, even when we land up outside the families? What is the chance that we would have? And do we have an estimate of that?
So right now we don't have any estimate except for the LAMB3 11 base pair deletion, because we looked at it because it's not available anywhere in India, anywhere outside. So it's restricted only that particular population. So we are going to estimate the frequency of that. But unless otherwise we study large number of population, because the complexity exists here because of the endogamy practice. So every population is going to have population specific mutation. Again, to find out whether that mutation, when it happens in a homozygous condition, only then we come to know that's the disease.
But there's no ways we could model this factor. So this number becomes crucial in counseling.
Yes. So the number is very crucial. Most of them are population specific. Unless otherwise you analyze the population, you are not going to get those information.
But from the 10,000 genome which you have now, why don't we look at that and then maybe look at the common things in our country and then make...
Now the 10,000 individual also, we have not analyzed
Yeah. So,
the entire population.
Okay.
So we have analyzed maximum 200 to 250 individuals. And we collected without any known phenotype. They are all supposed to be normal. And we pick up the way we picked up in other study, there could be a recessive mutation in the family because of sharing up haplotype. Then we have to go back the way we have done in the other study and identify which are the population, then precisely look at that mutation, that population, then we will have something. It's happening slowly. We may get it, but not immediately.
So just
Yes,
try to contribute to the conversation and based on what Dr. Deepak just asked, you have shown IBD data, right? How do you know technically which ones are IBD and which ones are IBS? And descent or by state? Because if you can figure out that difference, you can answer his question. There's a random chance of mating with somebody without knowing consanguinity, but I believe some of those IBDs will be IBS as well, identity by state.
Yeah.
So technically, can you distinguish between the two? Then you can actually address that issue.
So this IBD, we have used several mega-base region, right? So not necessarily that individual from the population in a population which is strictly following endogamy, which we approximately expect at least 50%, 60% share a common haplotype. On the other hand, if you go to the next population, which might living in the same area, isolated by genetics, maybe several generations, they're not going to have that haplotype to predict whether anybody else is going to have the disease. I did not have time to explain about the color. They are in 5 different districts. I can tell you and show you the pictures later on. 5 different districts, but only one district we found that mutation in high frequency, not in other district. Again, there's a population level substructure. They're not going to next district to marry. It's so complex. So in this scenario, how we are going to predict?
Sorry, sir, we need to conclude now. We are running out of time.
Yeah.
So thank you very much for this wonderful talk. Can you have a big hand, please? So after the morning tea, we have so energetic talks, and now we feel energetic also. So I'd like to give some token of appreciation as a felicitations. So I'd like to humbly request Dr. Vijayalakshmi ma'am to kindly felicitate Dr. Thangaraj sir. Can you have the, yeah. Can you have the pictures, please? Volunteers? Now I'd like to request Prasanna sir to kindly felicitate Dr. Srinivas Murthy sir. Now it's time to felicitate our chairpersons. So I'd like to request Professor Dr. Vanama sir to please come onto the stage and felicitate Prasanna sir. Now I'd like to humbly request Billu Patil ma'am to kindly felicitate Dr. Priya Lakshmi ma'am. Thank you so much. I'd like to request you to stay in the stage to have the picture. Sir, can you please join us? Yeah. And also Dr. Zulfali to be on the screen for the picture. Just 2 minutes. So we're breaking for the tea. After the tea break, we're going to have a session 2, personalized medicines. I'd like to hand over the mic to Dr. Aruna Priya and Zavia for the next session. Thank you so much, everyone. Within 10 minutes, sir. 10 minutes for tea break, sir. And I would like to convey my deep cordial thanks to Dr. Zulfali for joining us online. Thank you so much, sir, for joining.
Thank you, everyone.
Dr. Prabodha.
Personalized medicine. So I, Arunapriya, a level 2 board certified genetic counselor, and along with
Hello, I'm Sabha Agrawal, board certified genetic counselor. We both are the moderators for the session 2.
So personalized medicine has been an innovative approach for tailoring disease treatment and prevention by unraveling the complex biology of many diseases and applying advanced technologies. Personalized medicine makes it possible for doctors, researchers, and us genetic counselors to more accurately predict which treatments best suit the patient, taking into account individual genetic molecular makeup, environment, and also the lifestyle. So with these few words, I would now welcome the first chairperson of the session, Dr. Tangraj. I think he'll be joining us slightly. Yes. So Sabya would be introducing our second chairperson.
Our second chair is Dr. LBL Prabodha. He has done his MBBS from University of Ruhuna, Sri Lanka. Later, he pursued his MPhil in Anatomy and Genetics and MSc in Clinical Genetics. Currently, he is the senior lecturer in the Department of Anatomy, Faculty of Medicine in the University of Ruhuna. Dr. Prabodha's research interest lies in reproductive genetics, cancer genetics, and epidemiology of cancer genetics. He has received multiple grants and managed to establish and head the molecular genetics laboratory and the genetic clinic at the University of Ruhuna. He has extensive teaching experience in the subject of anatomy and genetics and is actively involved in multiple national and international conferences. With this, I invite Dr. Prabodha.
I now request Dr. Tangraj to introduce our first speaker of the day.
Yeah, good morning, all of you again. So this is another interesting session. There are 5 speakers, and the first speaker is Dr. Radha Venkatesan, my senior, whom I know for last 35 years. and she's going to talk about the. Radha, are you here? Radha? Okay. Good morning, Radha. Welcome to the conference. Yeah, please. You are muted, I think.
Not able to hear me?
Yes.
Can you hear me now?
Yes, Radha.
Oh, yeah.
Okay. So, good morning. Please go ahead. I don't want to cut down your time.
Yeah. Just not this. Just one moment. Just one moment. I'll just get off and. இந்த மாதிரியே கிடையாது அப்படி கிடையாது ஏய். ஆமா. பிடிக்கிறது கிடையாது the term first. Very different title. எப்படி நான் மாற்றுறது டைட்டில் இது மாற்றிக்கணும் give me just 1 minute. That's it. Function pangalaman what kind of windows do you know? Oh, I understand. Okay, right. Yes. Stop sharing. Hide. Hello. Are you able to hear me? Are you able to hear me?
Yes, Radha, please go ahead.
Are you able to see the screen?
Yes.
Yeah, okay, thank you. Hello, good morning. In the beginning, I would like to thank the Board of Genetic Counseling of India and especially Dr. Annie Hassan for giving me this opportunity to present our work on precision medicine. So this has been a very exciting time. I'm very sorry I'm not able to be there, but there are other commitments, both academic and personal, and therefore could not make it there. But however, it's wonderful to be a part of this meeting today. Let's move over to the presentation that I'm thinking of today, precision medicine approach to both polygenic and monogenic diabetes. I am working extensively on monogenic diabetes these days, and now we are also, very recently, we have also shifted to polygenic diabetes. So let me just explain how we are going about the whole thing. So I would like to acknowledge the Indian Council of Medical Research for all their financial support, Dr. Mohan, Dr. Anjana, and Dr. Ranjit for the constant encouragement given to me, and all the clinical collaborators who have given me samples and also shared with me the patients' records and shared with me the experiences of patients, because I'm able to be in touch with all the family members and also with the follow-up. And then finally, the very lovely group of mine, my colleagues, my PhD students, also my colleagues in the form of scientists who are a part of this work as this team, as well as this laboratory. So this is just a map that shows the states of India from which samples are being referred to as. Actually, we are the nodal center of Indian Council of Medical Research for monogenic genetic testing, because I'm also doing all of them free. The monogenic genetic testing is being done free here for the proband as well as for the entire family whenever they give us the samples, and then we give the results to them. So I made the center for India. So this is just to quickly go through the list of collaborators in MODI, NDM, and CHI. So let's just come to precision medicine itself. This is just providing the right medicine to the right patient and the right dose and at the right time. So that is the precise definition of precision medicine. But then what it involves is a number of things. It's very simply put in one line, but then everything is going to be very complicated. It involves tailoring medical treatment. And what is a tailoring medical treatment based on what? Based on various biological data, not just clinical, but putting them together, various other biological parameters, the biomarkers, the genetic markers, etc. And then include the prevention, diagnosis, and treatment. Not just that, you're also going through what we, the continuous, touch with them, because we are looking at how the clinical course runs. Then the prospect of applying the precision medicine that has really been improved. Why? Because of the development of large-scale biological data sets, the availability of them, and also the computational tools for the analysis of large data sets. Very, very happily and surprisingly, the best thing that happened was as the genomic era developed into a larger scale, the next generation sequencing came about, and that opened up, the flood of information into the world. That is because when our thought about genetics was also developing, the computational tools also simultaneously developed and took over, and then came the era of what the mechanization and a number of equipment and that can meet all these challenges. So the marriage between all these
really gave us where we are today. So the personalized or the precision medicine is actually the main thing is here is the use of the genomic information that I'm going to talk about, in addition to the family history, lifestyle, and environmental factors that will customize health management here. So by combining the genomic, chemical, and more accurate predictions can be made about the person's susceptibility of developing a disease, the person's, and also the course of the disease, and as well as the response to treatment. So the promise of medicine and precision medicine is number of them, particularly in the case of pharmacogenomics, in increased drug efficacy and decreased toxicity, and various others in targeted therapy, etc. But the lower cost, etc. But what it really involves is also the challenges. The challenges are also equally many. So first, building knowledge network to store data, building cohorts of patient data for further investigation, and that becomes very important, and updating research infrastructure, and translating basic science into practical guidelines for clinicians, and translating genetic testing results into meaningful patient results. So what really is very, very important here is the last two, translating basic science into practical guidelines for clinicians. Actually, this is what we are doing. Translating genetic testing results into meaningful patient results, because whatever we find as geneticists, and if you start explaining in that genetic jargon, that would be very difficult for the patients, and it would also not really mean, as we go along, I'll tell you where there is really the root block that comes in, the mind block that comes there for the patients. But that has to be treated very well. And the last two aspects of the challenges is what we are trying to meet. The other challenges are also being supported by, throughout the world in various. For example, you have an UK biobank to which you can go back and take it as a reference and compare. But that's not good enough, because you need to have an Indian biobank. So if you have an Indian biobank, and we do have some references internally and externally, and those things are also being done. So there is an infrastructure development as well that's taking place here. So these are the challenges that they have to be met with. So in precise, this precision medicine takes into account individual variability in genes, in environment, and lifestyle for each person. Well, this is mainly with respect to types of diabetes. How can we apply this concept? For example, there are broadly two types of diabetes. One is a polygenic, the other is a monogenic. The polygenic diabetes comprises the type 1 diabetes, the type 2 diabetes. Type 1 diabetes is the juvenile onset diabetes, and then the type 2 diabetes, and also the gestational diabetes. All these polygenic diabetes, as the name says, have a number of multiple genes that are responsible for the susceptibility of the disease. And it is not just the genes, it is also very complex, because various things like the environment also acts. But there is one particular type of diabetes known as monogenic diabetes, which is only the single gene is responsible. Say, for example, if you have something like thalassemia, well, the beta globin is mainly responsible. So like that, here, it is not just susceptibility of the genes, but it is the causation that the genetic variants cause. So that is where the monogenic diabetes really fall, and that is very important. So it is like for me, people like me who are the dreamers and the lovers of genetics, the monogenic diabetes becomes a very good model to work with, and to see what can be done, how we can apply precision medicine concept to monogenic diabetes. In the case of polygenic diabetes, what one really does is, you look at the genomes, or you do the whole genome or whole exome sequencing, and come up with umpteen, hundreds and thousands of various, genetic variation, and then you put them together and form a polygenic risk score, which is polygenic risk scores, or the PRS, which tells you that you are one is at a greater susceptibility for developing the disease. So it is like a prediction model that we can develop. And combining the rest of the things, you can give like the environment, what sort of lifestyle, etc. So we are able to give a prescription for precision medicine. But in the monogenic diabetes, it is not so. This is very different. Here, everything is based on a biomarker, that is the genetic biomarker.
the genes that are responsible for causing it, is able to give you a very good lead, a very good understanding of how we can apply medicine into diabetes practice and into monogenic conditions itself. So this can be broadly applied to, say, for example, monogenic obesity and various other monogenic genes, germline mutations. So the genetic basis, as I said, of various types of diabetes, one is with the type 2 diabetes is a susceptibility, and rare types of diabetes like monogenic is caused due to mutations in single genes. So here we have a number of syndromic forms of diabetes also. So moving on to monogenic diabetes, this is basically a beta cell diabetes. So here, the pancreatic beta cell is very fundamental to maintaining the glucose homeostasis. So which tries to match the insulin secretion to ambient glucose. So here, the glucose is sensed via multiple steps, resulting in ATP generation. So actually, it is the level of ATP which sensitizes the potassium channel that is present in the beta cell membrane. So the ATP-ADP ratio is very critical there, and depending on the ATP-ADP ratio, the whole system, the functioning changes here. That is where the secretion of the synthesized insulin results. So that is, insulin may be synthesized, but insulin will not be secreted. And the insulin will be secreted. So all this depends on the ratio of ATP and ADP. So any single gene defect in any step in this pathway, that results in monogenic beta cell diabetes. So coming to the quick definitions, the monogenic diabetes is a heterogeneous group of single gene disorders, and diagnosed with the diabetes at a very young age because it is very germline, and then it is a pediatric condition, because immediately the results will be felt, and which is unrelated to autoimmunity following a Mendelian inheritance pattern. About 5% of all the cases of diabetes is estimated to be having this monogenic diabetes, and many cases are misdiagnosed as type 1 or early type 2, but for which, and so if you think it is type 1, if the person is diagnosed as type 1 diabetes instead of monogenic diabetes, and the treatment is giving insulin. Insulin is the only method of choice for type 1 diabetes, because although the synthesis of insulin is there, what we say is that there is no secretion of insulin, and therefore when I use that, since there is no secretion of the insulin, there is no insulin itself, and then we start giving insulin therapy, but that is certainly not required, and that is not the right kind of right kind of treatment that should be given. So let's see how it can be done. So the major types of monogenic diabetes are the MODI, that is maturity onset diabetes of the young, the neonatal diabetes mellitus, that is the NDM, syndromes of diabetes, all of which result in hyperglycemic condition, that is glycemia, that is glucose levels much above the normal one. So exactly in contrast, there is also another monogenic disease, which is called as the congenital hyperinsulinism, CHA for short. And this is characterized by overproduction of insulin. And that is by the same pancreatic cells that results in hypoglycemia. Therefore, there is a reduction in the sugar, which is even more, damaging than the high sugar condition. The low sugar is very bad, because the low sugar means there is not enough fuel for the brain, and therefore that is going to make the brain cells permanently damaged. So it is very important to see that there is an ambient glucose level that is being kept in the body. So that's very important here. So now, why is there an extremely opposite things that is taking place, where there is one case where there is overproduction of insulin, the other where there is an underproduction of insulin. Well, so that is exactly because of the ATP-ADP ratio switching, and that is very critical. And that small, beautiful triggering and switching from one to the other, and the ratio completely reverting, is what causes the whole thing. And the reverting of ratio happens because of the genetic mutation, right? So it is very clearly can be traced back. So defining them, I'll just quickly gloss over this. What happens is, as I said, although it is most of them are pediatric condition, there is one condition, the MODI, which is a gradual problem, a progressive
beta cell damaging that takes place. It's a progressive disease, and therefore around 20-24 before the age of 30, this really sets in. And then you know that at that time, that's exactly the time. So it could be anywhere between age 5 to age 20 to 30. So it could, that means it is also the type 1 diabetes, it is also the early type 2 diabetes, or it is also the MODY. So it's very important to differentiate between the three, and very often this is being confused. In fact, about 80% of the MODY is still misdiagnosed as type 1. So that is where we have to really look into various biomarkers, and the GAD65, these are all the pancreatic autoantibodies. The autoantibodies are generally produced in type 1 diabetes because which is immune mediated, but monogenic MODY is not immune mediated. And then we also look at what is known as the C-peptide levels to differentiate between type 1 and type 2 are whether type 1 and MODY can be differentiated at the level of biomarkers. But sometimes there is still a confusion, and therefore what we do is we resort to genetic testing. So it is here that the genetic testing becomes more important than any other biomarker by biochemical testing. So the next is about, just to quickly define the neonatal diabetes. This diabetes takes place within the first 6 to 12 months of age. And usually GAD and IA2 the antibody will be negative. And very early, I have seen patients who are about 1 or 2 days old and then showing diabetes. So for some people what will happen is in the beginning it will show and then it will go off. So that is just a temporary one which is known as a transient neonatal diabetes. But for some it continues to be. So the moment there is the indication that the child, the newborn has this neonatal diabetes, immediately insulin will be given because that is the only thing that will bring, insulin is the only thing that will bring the glucose level down. So it's very important to bring the glucose level down. But that will be taken care of for some time. But after that is not the best of treatment, and therefore there will be a lot of side effects on that. So that also we have to see whether it is truly type 1, which is, I don't think we have seen any type 1 which is less than 1 year. In fact, throughout the world, maybe there are only 2 reports where they have seen. So we always consider to be more neonatal than this, but then we don't stop there. What we do is, we do the genetic panel testing, and then only we give the treatment. It is never given without treating. But what sort of treatment do we give? I'll give that to you. So the next is about the syndromic forms, the other one. So we have a number of syndromes of neonatal. Even this comes in the early age, and it also comes along with diabetes. So like the Digmard syndrome or the Wilfreman syndrome, and then the Wolfgang-Allison syndrome. And thiamine responsive megaloblastic anemia. In most cases, in thiamine responsive, it responds to thiamine and it doesn't respond to insulin. So that also had to be tested. And then only thiamine can be given. So all these supplements are totally dependent on genetics. The next, as I said, is CHA, which is exactly the opposite condition here. So moving on to the genetics of monogenic diabetes. So studies have been very successful in finding certain genes that are implicated in its pathogenesis. And there are 2 main components. One is the insulin secretory pathway, where the ABCC8 and KCNG11 genes come into play. The ABCC8 and KCNG11 genes are actually KATP channel genes, the potassium channel genes which are present, potassium channel which is present in the beta cell membrane. And the other pathway is the important is the transcription factors pathway. These transcription factors obviously will target the insulin gene or various other target genes through which they work. So they have a different way of working, and the insulin secretory pathway has a different mechanism of working. So the missense mutation in these pancreatic beta cell genes, these are all called as beta cell genes, pancreatic beta cell genes. It is not restricted only to these. I'm just giving you the main examples where we find more mutations than in the other genes. There are about a panel of 75 genes that have so far been implicated in monogenic diabetes and CHI. So missense mutation in these pancreatic beta cell genes, they cause a wide range of protein dysfunction that results in monogenic forms of diabetes and CHI. And what the mechanism really is behind this is that rare loss of function mutations in HNF 1 alpha, 4 alpha, 1 beta, and PDX1, that is the transcription factors, they cause highly penetrant Mendelian disorder. That's the MODY. And rare gain of function mutation in ABCC8 and KCNG11 cause a spectrum of disorder. One is the neonatal diabetes, another is the developmental delay, epilepsy, and neonatal diabetes. That is called as a GEND. To adult onset type 2 diabetes. So that clearly is dependent on the nature of the mutation of the penetrance of the gene.
Radha,
Yeah.
Kangaraj here. So I think time is up. So try to summarize in 2 minutes.
Hello.
Could you hear me, Radha?
Yeah, I can.
Yeah. So summarize in 2 minutes.
The whole thing.
Whatever you wanted to say now.
Hello.
We have only 2 minutes because the other speaker.
I have only 2 minutes left here.
Yes.
Hello.
Yes, Radha.
I have only a few minutes left.
Yes.
Oh, okay. So what we have done here is we have moved from clinic to laboratory, and then we have also moved from bench to bedside. Both these things have taken place in the case of precision medicine of monogenic diabetes and CHI. So what we really do here is to assess the variant pathogenicity, because the variants come in various shapes: pathogenic, likely pathogenic, VUS, likely benign, and benign. And what we have seen in our own clinic is that we have a monogenic diabetes and CHI patient of about the registry is about 2,164, and which is divided into monogenic, as you can see in this. So we see a number of cases how we have separated them into pathogenic variants, likely pathogenic variants, and VUS. That is because we have done a lot of work on bioinformatics, more on functional genomics also. So we are able to, based on that, we are able to give the clinical actionability of genetic variants. So I will skip one of this. I will show you a 16-year-old girl presented with a severe diabetes, and she was originally treated with insulin. But then after going through the pedigree and finding out that she has a specific mutation, arginine 263 histidine, we also went through a number of, we wanted to see whether it is a MODY, because based on the segregation analysis on the functional aspect and structural aspect, as well as the follow-up, we could clearly see how the blood glucose has come down after 2-3 years. So we were able to, when we shifted them from insulin to sulfonylurea, these children do not require insulin, but they require sulfonylurea. So that is exactly what I would like to submit to state in this case. And then the same thing, see, for example, after shifting them from insulin and giving them the sulfonylurea, the follow-up is that the blood sugar levels have come down drastically in the case of MODY. More so in the case of neonatal diabetes, which I can quickly show. See the way originally it was even 1,000 milligrams, and we have seen some patients with 1,000 milligrams of diabetes, DL of blood glucose, which has come down to 200. And less than 200 and 150, and then it continues to be so. And that is because we have shifted them from insulin to sulfonylurea, because sulfonylurea corrects the mechanical problem that is taking place in the beta cell, and then that continues to secrete insulin that is already, the insulin has already secreted, release the insulin that has been secreted. So that is the trick here. That is what, but not all mutations are analogous to this. They don't react to that. Some of them do. So that is the reason why we have to do the functional tests, as well as we have to follow up the patients. That is what we have done in the case of CHI, which is very MDM or MODY. But in the case of CHI, it is exactly the opposite, where we give diazoxide or octreotide from low sugar, how the high sugar and the normal sugar has come about, and how we maintain. It is not enough if one month or 2 months it comes, it has to be maintained over a period of years and much more. So we continue to maintain and we continue to be in touch with them. So we have seen the representation of a number of mutations in KCNJ11 and ABCC8. So we have moved from genetic type, clinical features and treatment response based completely on genetic subtypes. So that is very important, which is also important in the case of genetic counseling. So we have a number of things that are known and a number of things that are unknown. As you can see, we have the monogenic diabetes and CHI, pathogenic variants and unknown genetic etiology. And we have the VUSs and how we do the molecular characterization. And we are able to deliver precision medicine in monogenic diabetes and CHI because based purely on the genetic defect. and some of the genetic defects are totally new, and some of them are variant of unknown significance. So we have to do a number of multiple tests, and then only we have to come about. In our MDRF, we have the functional genomics lab where continuously work is being taking place to characterize the mutations regularly. So molecular genetics has truly revolutionized diabetes clinics from bedside to bench, where the reference from a clinical collaborator based on clinical assessment, and from bench to bedside, delivering on precision medicine based on molecular genetic workup. A change from conventional approach to the precision medicine approach, and this change has become very transformative in the case of genetics and also genetic counseling. Thank you very much for your attention.
Radha, thank you very much for the wonderful talk. Please stay back, otherwise we'll give you talk. Could you hear me, Radha?
Yes, I can hear you.
So we are going to have discussion after all the speakers finish their talk. So please stay back, otherwise I'll give you a call when there is a discussion. You can join.
Thank you.
Thank you. Bye.
Thank you very much, Dr. Radha. So then we'll move into the second speaker, Dr. Vinod Sikaria. He's a senior consultant in genome information at Vishwanath Cancer Care Foundation. And he was an undergraduate of University of Calicut and did the PhD in computational biology from the University of Pune. And he's the co-founder of the Genomics for the Understanding Rare Diseases India Alliance Network. And he's an adjunct professor, Indian Institute of Technology, Kanpur, and Dr. Y.D. Patali Medical College, Hospital and Research Center. And he's a distinguished visitor in Asok University and visiting professor, North Bengal University. And he has a very enlightened career highlights with more than 200 research publications in international peer-reviewed journals. And he had published 2 reputed books, and is a member of editorial board of many editorial boards and journals, and is a member of, he's a recipient of CSIR Young Scientist Award for the Biological Science in 2012, and also a Frontiers of Science Fellow of the US National Academy of Science in 2015, and an elected fellow of the Royal Society of Biology in 2017 and the Royal Society of Public Health 2022. So I invite Dr. Vinod Sikaria to take over the presentation. Over the next 20 odd minutes, I'll try to emphasize on how we can take from personal genomes to populations, and from population genomes, how we can impact individuals with their genetic diseases. Now, when we talk about genomics and genomic testing or genetics for all, what we need to realize is that genomics is not equitable across the world. And this is a slide from way back in 2008, when I started my lab at the CSI Institute of Genomics and Integrative Biology. There were just 6 genomes at that point in time, and what you see in the top panel is essentially the admixture plot of the world. And you quickly realize that the most admixed populations across the world are not really covered in whole genome sequences. And to put numbers, these populations in the middle part of the slide is approximately half of the global population, right? And let that sink in. Practically all the populations that were widely sequenced at that point of time and still continues to be sequenced at that, even today, are from less admixed populations across the world. So we are really missing out the diversity of human population as we talk about genomics. So in 2009, we planned to change this one at a time. So we did the first genome in 2009 with my colleague, Dr. Sridhar, who is not here. And we went ahead to collaborate quite extensively across this region, right from Middle East to Central and Southeast Asia, to sequence many of the first genomes of its kind in these regions, including the first Sri Lankan genome with Professor Vajira, the first Malaysian genome with UITM, and we also created one of the premier databases for South Asian, Indian, as well as Middle Eastern population genomes. To essentially understand how does this genomic diversity impact clinical care.
Now, from there, we can, of course, step back to India and ask a very interesting question, because India is very, very interesting in many aspects. The first aspect is it's a large country. And to put numbers, we are 1/6 of the global humanity. And also to put in perspective, the next 1/6 of our global humanity, which is our neighbor China, is much less admixed, much less stratified than what we see in India. And to put numbers, India has around 4,000 odd endogamous populations. So if India was a European continent, we would have 4,000 countries. Put it very simple. Much more important than all of this, compared to the rest of the world, which has gone through 2 rounds of world wars, multiple rounds of famine, multiple rounds of displacement, India has largely been undisplaced in the last 400, 500 years. So that necessarily means that everybody in this room can trace their ancestry by minimum of 3 generations. You know where their grandparents are. Many of us can trace their ancestry by 10 generations or more. Right? And all in genetics and genomic counseling starts with the pedigree. Right? And therefore, we are at a much advantageous position compared to the rest of the world. Many of the rest of the world cannot even trace their ancestry more than 2 generations. Now, how does it impact families and patients in India? This is our first family. We call him the Bai, of course, from Gujarat. He came to us with a very interesting condition. As you see here, that's the skin of this individual. It breaks up as if it was blistered, and it's a very painful condition. It's a very common genetic disorder, common in the sense a very prevalent rare genetic disorder called epidermolysis bullosa. Now, there is not just one individual. We followed up this entire family, 28 odd affected members in 5 generations, staying within 10 square kilometers of where he lives. Now, this is something that is impossible to recreate anywhere else in the world, because 1, you never have 5 generation pedigrees. 2nd, they're undisplaced, so they all live within the same locality. And 3rd, identification of the mutation is not the end of the story for one individual, it's the end of the story for the entire community. Right? And from 2009, this sequence was done in 2009. Today we have eliminated the disease in the family. It was supposed to be a curse, because all 50% of the members would have this disease. And today, by prenatal testing, genetic counseling, we could eliminate the disease from recurring in the family. So you can actually put an end to the diagnostic odyssey for families. So that prompted us to actually create this program with my colleague Sridhar for many years at IGIV in 2015 called Guardian. I'm sure a lot of you have heard about it. The idea was that we will collaborate with clinicians to work towards understanding what are the rare diseases. We were told that, of course, rare diseases are so rare, so you will never find them. But in around 7 years, we had around 10,000 patients with rare genetic diseases. We didn't have funds, therefore we had to stop taking more samples. The idea is very simple. You'll work with clinicians, we'll do a genomic analysis. We will get back to the clinician whenever we have a pathogenic genetic mutation, which happens approximately around 30% of the cases. And whenever we require more evidence to prove that a pathogenic variant is indeed pathogenic, we step back into the lab, create that evidence, get back to the clinician so that the clinician can act upon it. I'll just delve with probably one or couple of stories on how this really works and how we should really work beyond that one single patient. Now, this family was in news because they petitioned the President of India to euthanize 6 out of their 8 children. Right? Because they have a progressive neurological disorder in the clockwise direction from the youngest to the oldest. They're perfectly normal at birth. At around 3 years, they start losing their milestones, eventually end up in a vegetative state. They have no diagnosis, they have no cure. And therefore, they petitioned the president that please euthanize 6 out of their 8 children. But think of the plight of the family. So, of course, euthanasia is not permitted in India, so it got referred to us through the official channels. We did genetic studies for multiple members of this family. We did identify the mutation, making the diagnosis of cystic megalencephaly or megalencephalic leukoencephalopathy with subcortical cyst type 1, an extremely rare genetic disorder. With a prevalence of approximately 1 in a million, this variant was only reported once previously in global literature, and that too in an abstract. So is that the end for it? For most of you, probably most of clinicians, including me, that should have been the end of it. We did not put an end to it. We really went back to the community. And what we realized is that these individuals come from a very peculiar population called the Nalband. Nalband do have an ancestry, recorded ancestry for 54 generations. in paper that they can trace where they came from. They came from Middle East. They came with one of the invading armies. They used to make horseshoes and they settled in the ramparts of Agra, Aligarh and Jaipur. And the entire population in the Nalband is approximately around 5 lakh individuals. Quite endogamous, largely consanguineous community. We did a house-to-house, door-to-door screen. And what we found was even more remarkable. Around 28% of this community are carriers for this extremely rare genetic disorder. And what this number would approximately mean is 1 in around 400 odd children born to this community will have this genetic disorder. So it might be an extremely rare genetic disorder elsewhere in the world, elsewhere across the state, elsewhere in the country. But for this community, it is indeed a very prevalent genetic disorder. And in fact, by just house-to-house survey, we identify another 3 families with children having the same genetic disorder. Now, how do we really make a difference for them? They just need a simple test, right? It is just one mutation. It is a founder event. You can sequence it, genotype it, do whatever. They don't need an exome or a genome. They just need a simple genetic test and more importantly, genetic counseling so that they can be counseled premarital, postmarital, and they can take the decisions accordingly. So we put this from 2016 to 2018 from the publication to the diagnosis. And 2019 today we have a diagnostic in the market. It's on the catalog of Dr. Lal PathLabs. In fact, in India, we have 2 founder mutations for this particular gene. One in the Bansal Agrawal community called the Wandernack mutation. And of course, the second one is a Nalband mutation. So think of it that every mutation that you discover in India, especially when you're in a founder population, could impact far more number of people than you can actually think. And that can really make a difference to the family and the community. Now that comes to a very interesting question on what is really prevalent. I'm sure you would have come across this, and yesterday I had a lot of discussions on diseases which are seemingly prevalent. Now, what you need to keep understanding is that in clinical settings, whether you're a clinician or a genetic counselor, what you see is only what comes to you, right? And if you tend to diagnose a condition better, you also tend to see more of those conditions. And therefore, practically all prevalence estimates for any disease in this country is skewed because it's skewed by one or the other individual who sees it better. You ask a cardiologist would say the prevalent genetic disease would be cardiomyopathy, for example. You ask a pediatric neurologist would say something else. You ask an adult neurologist, you would say, for example, spinocerebellar ataxia, because that's what he or she sees. There's nothing wrong with that. But now, how do you really build in policies when you really do not know what the prevalence is, and especially when you have a national rare disease policy? Now, to me, the only way you can solve this problem is to actually sequence all of you, right? Apparently normal individuals, and ask a very simple question, what are you carrier for? Right? If your carrier frequency is much larger, I can go back to the community, look at the marriage practices in the community, and have a fairly good estimate of what is prevalent in the community. Right? I hope you understand what I'm trying to say. Right? You can sequence normal individuals to actually figure out what is really wrong with the community. And by doing that, you can actually go back to the clinicians, teach them a few tricks, and tell them you're probably missing out this disorder. Not a good thing to say though. But of course, enable them to diagnose the conditions better. Right? You think this is impossible to do? It's not really. So in 2019, with funding from the government, we piloted 1,000 odd genomes from India. Because 1,000 people from 1.4 billion is nothing. But nevertheless, it's a good data set to really look at the low-hanging fruits, to really look at what is really, really prevalent in the community. So we did this, and I'll just put in a couple of examples. We landed upon one such condition. This is familial hypercholesterolemia. I'm sure, have you heard about this condition? What is the prevalence of this condition? Rare. The number is there. 1 in 140 odd individuals would have familial hypercholesterolemia. And each of these individuals, including people in this room, would have, assume there are 300 people in here, 2 people would have. You will lose 2 decades of your life because you will die out of a cardiac event very early on. And the unfortunate part of it, it is a treatable genetic condition. You can take statins and you can have a perfectly normal life. So it is not an expensive 70 lakh rupees test, 70 lakh rupees drug. It is a few thousand rupees test and a few thousand rupees treatment. And you can save clearly 2 decades of your life. People have shown this, and therefore in the Europe, this year onwards, there's a newborn screening program for family hypercholesterolemia. Of course, when you get diagnosed with family hypercholesterolemia, you take statins. Now there's a problem with statins, that statins also cause adverse events. One of the commonest adverse events is myopathy, and that's also unfortunately genetically determined, or fortunately genetically determined. So you can also test for that additional genetic event. And what we showed in our paper is that approximately around a third of individuals would require a dosage modification. So you can do these 2 tests together, and you can save, when it's a 1 in 140, just multiply it by 1.4 billion and you can get the number, right? That is how huge is the problem of genetic diseases, and that is the opportunity that we are today missing. We might be worried about the rarest of the rare condition, while we are missing out the commonest of the common conditions. We can think about even much rarer conditions that you might have come across, right? And this is of course bias set, because you will never come across this condition because these kids don't live enough to come to you, right? When you don't have an immune system, which is called primary immunodeficiency, you don't have an immune system, you succumb to infections. The slightest of the slightest infection end up in sepsis, and the kid succumbs to it. So they will never come to a clinician, they will never come to a genetic counselor saying that I have a disease, right? They are largely in ICUs battling for their life. So very unlikely that you would have ever seen a patient with primary immunodeficiency condition. But this is prevalent. You can actually go back to the genomes, you can understand the prevalence. We went back to one center, my alma mater in Calicut, try to teach my own teachers that this is something that we miss out. But of course, more important is to enable them with the right kind of test that they can test it. So they have a test that runs in the hospital with hundreds of patients today. In 7 years, we have created one of the largest cohorts of primary immunodeficiency disorders in the entire Asia. Just one single center. So you can imagine what the problem is. And fortunately, these patients can access treatments today. With, of course, the national and the state policies, they can take a one-time treatment option, which is a bone marrow transplant, or they can get IVIG for their entire lives. So there is a treatment opportunity available, provided you can diagnose them very early on. And to diagnose them very early on, you have to diagnose them in the first months of life. And that's really, really important. Many countries have a newborn screening program for primary immunodeficiency disorders, but of course we don't have, because in no national document you have ever mentioned primary immunodeficiency disorder, because no clinicians have seen it. I mean, these documents, of course, are made by people like you and me. Right? If you have not seen it, very unlikely that will be in the document. A third and of course more important aspect is the real opportunity in genetics and genomics is not seeing patients and advising them. Is to actually prevent patients from coming to your clinic. Of course, that is counterintuitive, but that's essentially the principle of newborn screening. The principle of newborn screening is that you have a accessible, affordable test for a treatable condition, and therefore you can save the child and have a normal life. US today does around 37 genetic tests. But of course, a broader question to ask is what is really prevalent, what is really treatable, and therefore can I create a list of genes and tests that I should do in a country or in a state? It might be different for different states, I completely agree to it. It can be different for different populations, but at least let us get to know what is really prevalent. Today, if you look at newborn screening programs, they're largely because some country created a newborn screening program, that kit is available, therefore I will also run it. That's probably not the way to do. So you can go back to the carrier frequency and ask a very simple question. What are the treatable genetic disorders? Intellectual disability being one of it, because that is something that if you treat, the outcomes are really, really great. Any guess what is the most prevalent treatable intellectual disability? Phenylketonuria, no. Congenital hyperthyroidism, yes. Second, G6PD, no. It is Wilson's disease. The carrier frequency in the population is less than 1 in 100, right? That means that there are 3 carriers in this room, right? There are 1 in around 30,000 children born with Wilson's disease. And unfortunately, our last patient was a medical student. A medical student, think of it. A medical student who collapsed in the clinic, was taken to the ICU. After a lot of investigations, around a week later, was diagnosed to have Wilson's disease. So if the medical student cannot diagnose his or her condition, I think we have a big problem here. Right? And the problem is largely because Wilson's disease in textbooks is a pediatric condition. Right? But Wilson's disease in literature is not a pediatric condition. The first report of Wilson's literature in literature is a patient with a neuropsychiatric manifestation, a 24-year-old adult. You can ask anybody in this room, you would have never seen a 24-year-old adult with Wilson's disease. So we need to really go back and ask this question, are we really looking at the right phenotypes? Are we really looking at the right things to teach the next generation of people? And before I close, just 3 more slides. We are in the midst of a revolution. We have seen how Sanger sequencing has got converted to WES. I'm sure everybody knows WES because everybody is doing WES. We will probably do one more step, and that is probably going to change the way we do genetics and genomics. And that's largely because that is possible to change the way we diagnose and treat patients. Today we diagnose and treat patients because a patient comes to the clinic. We teach doctors to look at patients. We set up hospitals so that patients will come to them so that they can be treated. The real paradigm is, can we diagnose them before they can get to the hospital? There's a lot of opportunity there because this widens the reach of genetics and genomics to a common set of people like you and me, apparently normal. We can prevent diseases, means we can have a healthy, normal life. And that is going to save a lot of money for the society and for the country. And if you think large programs across the world are not thinking that exactly what they're doing. The populations are sequencing everybody in the country, largely because there is enough economic evidence to suggest by doing this, you can save a lot of healthcare cost. Number 2 is, I'm sure you would have known all these genetic tests. The multiple kinds of genetic tests that we do for multiple kinds of genetic disorders. For chromosomal abnormalities, you can do karyotyping. For very specific abnormalities, you can do FISH. Then you do repeat PCRs for repeat expansions. You do panel sequencing or exome sequencing or Sanger sequencing for very specific disorders. And of course, you have arrays and other things in between. Each of them have a different turnaround time. It requires a different kind of expertise, different kinds of false positives, false negatives, different accuracies, and of course, different costs. What will happen in the future, probably half a decade from now, that this is all going to get converged into one single test. And that one single test will sequence your entire genome from end to end. And you can look at all of these genetic variations in one single test, much faster, much cheaper, and much more comprehensive in the way you can look at genetic testing. And of course, cost of sequencing is dwindling very rapidly. That's not a linear scale, that's a logarithm scale. Now put in perspective, that would necessarily mean that today if you're doing exome sequencing, probably you should start thinking about doing genome sequencing. I think I'll stop there and take questions, but before that, just a few points to go home with. Genomic sequencing is ripe for clinical implementation because the costs are coming down. It's just exome sequencing cost 4 years ago. Genomes can enable disease diagnosis because that's what you're interested in. But more importantly, it can contribute to real life evidence for public health decision making in terms of what should be a newborn screening program that I should run. Genetic diseases are extremely prevalent. If I give this number, of course, I'm sure you will all be surprised. 1 in 10 in this room have a genetic disease or a genetic disease trait that would impact your healthcare decisions. That means you can even be a carrier, you can have a pharmacogenomic variant, you can have an HLA allele, you can have a disease which is dominant or recessive, and some of them are actually treatable. And therefore, genomes or genetic testing is not just for patients, it is probably for everybody. And more importantly, this evidence is for a lifetime because a genome that you sequence today is going to be constant from the day you're born to the day you are dead. Thank you very much.
Thank you very much, Dr. Avnath.
Next speaker, I think Dr. Deepak Modi from National Institute of Research on Reproductive and child health. Deepak has been working on male infertility for a very long time, and I invite him without spending much time. Thank you.
Am I audible? Good afternoon, everyone, and thank you, organizers, for inviting me here. My job is one easy because precision medicine has already been introduced, but speaking after Vinod is always overwhelming because the way he puts up the entire perspective of genetics, I'm feeling humbled that I'm a part of this session. I'm not going to talk about any inherited genetic diseases or prevention of genetic diseases, but I'm going to tell you something about how genetic information can be used for improving clinical practices, more specifically in my case, infertility practices, IVF to be more precise. So when we talk about infertility, obviously you'll tell me that India is a populous country and we need not worry about infertility. But I must remind you that the total fertility rates in India have fallen below the alarming levels. We are at, chicken is 1.1. Rest of India is at 1.8. So 2 is what is expected. We are actually there in India falling below very rapidly. The last NFHS also tells us about 27.5 million couples in the country who are actively trying to conceive and are unable to conceive. About 1 in 5 couples experience difficulties in conception. And the infertility rate in India range from 6.4 to 16.8. And I must remind you, this is still an underestimate because it's from just NFHS surveys, and these are not something which are population surveys. And considering the taboo of infertility, specifically in the rural segments, these numbers are way higher than what we are talking about. So it's a matter which we must worry about. And therefore, when we talk about infertility, these couples obviously opt for some forms of treatments. They go to gynecologists, they go to obstetrician, and the medical managements and surgical managements. Most of these do take care of it, but that's only in the 25% of cases. A whopping 75% of couples who are infertile have to opt for in vitro fertilization or assisted reproduction, to be more specific. And that's a number. If you really count, if you translate the numbers, I'm sure all of you in the urban metros agree with me that IVF clinics are mushrooming. I mean, you would walk up to another area in about a month and you will see a new IVF clinic setting up there. And if the clinic's mushrooming, I'm sure there are enough patients for them to survive. And therefore, it's a problem. It's a global problem. It's an Indian problem. And it's important to worry about it. And typically, you would all know about the IVF segment where you would have a couple who is evaluated. It starts with a hormonal hyperstimulation to make enough oocytes in the ovaries. You would collect those oocytes surgically, have a sperm preparation, allow them to fertilize in a dish. You could either do a IVF or a ICSI, which is most commonly done. You take a sperm, put it in the egg, allow the embryos to grow up to a certain stage, transfer into uterus, and have a pregnancy. If we talk about its success rates, as of today, the success rate of IVF is estimated to be around 37% in general, but if the woman is above 40 years, the success rates drop by almost to half. Only 2 to 12% of women above 40 years are able to conceive of IVF. So the success rates are not very great. And the problem is that this 37% looks good, but it's about 1/4 of the total population, and remaining still needs multiple attempts to become successful. So is there a way of increasing these numbers here? And is there a way of helping these women so that we can help them better to have babies? So how do we improve the success rates of assisted reproduction is the primary question. And can genetics help us in this aspect? So obviously you all know about personalized medicines, where we are not talking about one size fits all. We are talking about figuring out what the patient is going through, what is the genome of the patient, and can this genomic information help to optimize the treatments and IVF so the success rate becomes better. I'll give you 4 examples, which is as of today in practice or getting into practice for improvising success rates, specifically using genetics. Personalized medicine is not just about genetics. Genetics is definitely a part of it, a good significant part of it, but in these 4 aspects, personalized medicine is getting into clinical practice very rapidly. And I must remind you, none of these are genetic conditions. I'm not talking about infertile couples with a genetic problem. So the first one of them is optimizing dosage of gonadotropins. As of today, So typically in IVF lab, women are injected with gonadotropins and their ovary hyperstimulates. And there's a very specific protocol, but if I'm really talking about numbers, this is not a protocol which is so well optimized. It's highly variable. The amount of gonadotropin to be injected for the woman so that she optimally hyperstimulates is something which is the biggest challenge in IVF practice. And from what, when the patient have a baby or not, when she starts her IVF cycle, it changes to will she make oocytes or not on the first hand. But she has sufficient numbers of oocytes in her ovaries. If I give her gonadotropins, will she hyperstimulate them? How many oocytes am I going to get at the end of the stimulation? Am I going to get 2, 5? Currently, by regulatory means, you can only have 7 oocytes which we can retrieve from a woman after in the IVF cycle. Will I get 7? Will I get too many? Will I get too few? The patient as well as the clinician is constantly in a dilemma, and therefore the patient constantly undergoes repeated ultrasounds while taking injections to figure out, and the dosage is optimized for them. However, this is not the best way of doing it, because that means a woman has to go to the clinic almost nearly every day, and it's very, very difficult to decide how much of dose to give at the beginning of the cycle. And essentially by the end of these treatments, women fall into 3 major categories. Many of them make reasonable 6 to 9 eggs, which is expected, but there are a sizable number of women who don't just make more than 3 eggs. This is called as hypostimulation. They have not got stimulated enough to make these eggs, and there's also a consequence, because if you just had 3 eggs, almost 2 of them, 1 will not fertilize, you'll be left with 2, you're left with 1 or no embryos, leading to cycle cancellation. On the other extreme, some women are hypersensitive to gonadotropins and they make just way too many oocytes, and this syndrome is called as OHHS, which is approximately 1 to 5% of all IVF cycles, and this often is life-threatening. And this is again a problem in the IVF clinic. The 2 extremes, and predicting these 2 extremes becomes very crucial before the start of the ovulation cycle. So the question is, can we predict these responses way before the ovulation cycle is started, so the woman doesn't get into this or that? If we believe that, okay, this woman is more likely to hyperstimulate, the initial dosage of gonadotropins can be low, and if a woman is less likely to hyperstimulate, the gonadotropins can be jacked up way at the beginning of the cycle or optimized by that. So towards that, we did some work which has been published largely, where we looked at multiple receptors which are known to be associated with ovarian stimulation. To name a few, we had looked at FSH receptor, LH receptor, AMH and AMHR receptor. Then there are others who have looked at way many numbers of receptors, but often the most promising one has been FSH receptors and AMH receptors. Physiologically, they make a lot of sense because FSH is the one which is produced by the pituitary, whose receptors are there on the granulosa cells, and these granulosa cells will eventually make the follicles. So what we found was that there are certain polymorphisms. Now, if you look at the omens, or if you look at most of the databases, these are synonymous polymorphisms, not pathogenic variants, non-pathogenic conditions. But one of the synonymous variations, and we took a cohort of women who had, this is I think about 150 cases to begin with. We had a cohort of women where we genotyped them and looked at their ovarian hyperstimulation outcomes. And what we find here is that if the woman carries a AA genotype, she is likely to have initially low basal FSH. If she is having a AA genotype, the amount of FSH required is going to be low to hyperstimulate her. And despite of a low hyperstimulation, almost 80% of these women will land up in an ovarian hyperstimulation syndrome. So if we knew these women with an AA genotype before, we could have avoided the entire hyperstimulation profile very early. And it's conversely, when we look at the TT genotype, it's conversely reversed, that if they are carriers of TT genotype, they will require more FSH and they're going to produce less numbers of oocytes. So if we use this test to begin with, could be able to predict the responses to FSH treatment. We looked at another polymorphism. Now this is in the promoter region. This was way better work than the previous one because synonymous variants, it's difficult to even do lab experiments there. But here there's the polymorphism at minus 29 in the 29th position on the promoter. And what the student had shown was that Patients who have AA genotype, in fact, in the -29 position now, innately have low amounts of FSH receptors on the granulosa cells. And almost all of these women required way higher amount of FSH to be required for stimulation, and they produce way few numbers of oocytes than what a normal stimulation protocol would do. And this is the actual data. The amount of exogenous FSH required to these women to hyperstimulate was way higher. The numbers of pre-ovulatory follicles were significantly lower. The numbers of oocytes which were retrieved were significantly lower, and almost 60% of these patients now on the converse spectrum landed up having a poor response. So FSH receptor polymorphism strongly correlate with the prediction to response to FSH way before a patient is even taken up into the cycle. Extending this further, because with the spectrum of mutation and spectrum of variants and polymorphisms which we looked at it, we figured out which of these combinations is able to predict this way better than just one or two of them. We did a lot of correlation analysis and found that -29 is the one which is the strongest predictor. In the clinics today, the clinicians use antral follicle count, AMH levels, FSH levels to predict all this. We use the machine learning algorithm where we took all this metadata along with our polymorphisms and found that all of the others, the age, the AMH, AFCs, basal, LH, FSH, predict the possibility of a good response. But in addition to that, if we add the -29 and the ASM polymorphism, which I spoke first, you could predict hyperstimulation in about 80 to 90% accuracy. The cohort is small, it's just 400 patients right now, but we are now running a direct cohort where we are genotyping first and then asking the clinicians what is the outcome there. And conversely, the clinicians are now optimizing doses based on these profiles and giving personalized hyperstimulation responses. So the end of the product is now possibly we will also have an algorithm for the clinicians to predict that if we add in the genotype and the basal metadata, what is the response going to be? Because just having a genotype alone or just having this information alone is too overwhelming for a clinician, and we will use counselors to make it into our actual practice. So we are making use of artificial intelligence, machine learning algorithms to say that, okay, you don't need to worry about it. You will get a number as soon as you put these values for it. So this is how we are progressing. So we hope in time to come, the FSH receptor polymorphisms of this metadata with FSHRs should change the way approaches in IVF, where it is just about giving random doses of gonadotropins to optimize doses based on the genotype. The second I'm going to talk about is pre-implantation genetic testing, which I see a session happening, so I'm not going to spend too much of time over it. But essentially, when we look at IVF, or even when we look at natural conceptions, about more than 50% of blastocysts naturally will have some chromosomal aneuploidies. The aneuploidies rate in pre-implantation blastocysts is way higher, and if we look at the rates of pregnancy in these cases, when we transfer obviously aneuploid blastocysts, the likelihood of pregnancy is going to be very low. The idea here is that if you diagnose these embryos if you know the aneuploidy status way before the time, before you do the implantation, you can now select amongst the 5 blastocysts, a genetically healthy one, to allow pregnancies. Lots of studies have been done globally, including India, and it has been shown that as compared to the non-PGTA cycles, with the PGTA cycles, the likelihood of getting pregnancy increases by 2-folds nearly, and the risk of miscarriage reduces by 25%. So you're not just increasing the chance of IVF success, but you're also reducing the chance of miscarriage post-IVF by introducing PGTA. The third is something which I'm very passionate about, is detecting the receptivity status of the endometrium. Now, the endometrium is a cyclic tissue, and it's only during a very narrow window of 4 days that the endometrium is receptive. And it's only during that time the embryos to be transferred. Obviously, based on hormonal status, you can predict it well, but it's not that it's going to be accurate. So you know that, okay, ovulation is done about 6 days, but 7 days post-ovulation, the endometrium is going to be receptive. The problem is in a bunch of IVF women, the receptivity window is shifted either to the left or to the right. So you predict that, okay, she's going to be receptive on about, let's say, day 20th of the cycle. She might have become receptive on day 17, or she might become receptive on day 22 based on her innate genetic profile. And this becomes problematic on IVF labs because now you don't know when the embryo to transfer. You may either transfer it too late, in this case, or you may transfer it too early. So the idea is, can we identify this window of receptivity appropriately for that woman? So my lab has been looking at trying to figure out what are the genetic factors, what are the molecular factors required to acquire receptivity. We have done some work and a lot of others have done it. And eventually a signature has been identified for the receptor endometrium. And translationally now it's been converted to something called as a endometrial receptivity testing. So now you take a biopsy of this endometrium, push it onto this receptivity array, which is now RNA-based transcriptome profiling, and you can predict when is she going to be receptive. So is she going to be receptive on the right day, or has her receptivity been prepone or postpone? And now we do a personalized embryo transfer. You don't do the embryo transfer just by the hope that she is receptive. You actually figure out when she is likely to be receptive and then do the embryo transfer. And if she is not receptive, then change her gonadotropin requirements, do the therapies about progesterone treatments so that she gets receptivity appropriately, exactly on the day you would want, so the transfers can be optimized. This is called as personalized embryo transfers. And studies from India, this is from Dr. Banker's clinic in Ahmedabad, they're very nicely shown that if you use endometrial receptivity array, the chances of pregnancy rates improve significantly, and the cumulative pregnancy rate reach up to 72%. By just simply applying a receptivity test. And imagine now the situation, now here they're not looked at the ploidy of the embryos. They're still added embryos whose ploidy status was not known. Imagine combining the two. Personalized embryo transfers of a genetically healthy embryo. And what would you get? This is a study again from Ahmedabad by Dr. Jaishamin's lab. And they're very nicely shown that the overall clinical pregnancy rates in the control group, which is 23% as expected, you don't worry too much about it, shoots almost to double when you apply ERA with PID testing. The overall implantation rates improve, and the abortion rates, which is 17%, drops down to just 2%. So personalizing the embryos, personalizing the time of implantation improves IVF pregnancy rates. And this, they have shown, is definitely a combination of both. Both of them together help way better than using either of one alone. So to summarize, in IVF practice, definitely personalized medicine is at great infancy, unlike oncology where a lot of things have become very, very personalized. In IVF, it's still at infancy, but at least definitely the IVF community is getting increasingly accepting to the idea that one size is not going to fit all. We will probably need to optimize things for each patient. And as of today, it's been rapidly getting personalized in IVF segments, especially in the metros where these access to these tests are available. Patients are using it very actively, and we believe that FSH receptor polymorphisms, if translated eventually, will help the clinicians to optimize the doses. Pre-implantation genetic testing, especially in women above 35 years, significantly helps them to improve the pregnancy rates, and personalized embryo transfers based on endometrial profiling is a promising approach to increase the rate. So we hope that probably will never reach 100%, but probably from that 20 to 30%, we should at least reach up to 60 to 70%. So more and more couples are able to benefit from IVF rather than what we have today. Thank you, and I'll be happy to take questions. I love this statement of how medical practice is more about personalized medicine and not just about letting the patients have babies. Thank you so much.
Thank you very much, Dr. Deepak Modi. Due to the time constraint, so I would like to invite Dr. Renuka Jain. She is the Senior Vice President for the Pharma and Nutrition of Anesthesian. So you can see her short CV. So I will give the stage to her to continue his speech.
I would like to thank BGCI for giving me the opportunity to present my work today here, and also most important topic on the unlocking personalized nutrition, the future of health and well-being through nutrigenomics. Next slide, please. So nutrigenomics, the gene and nutrient interaction it is. The pursuit of the knowledge about healthy well-being and all is I'm not able to is among the global population. It is the thing that we have to be healthy, but the genome is already set in. Once the zygote is formed, the nutrition is required until from womb to tomb. We need a healthy nutrition for us to continue and be healthy. But the genome advancement after the Human Genome Project, it is like the kind of food and bioactive compounds that we are that we are taking also influence. Okay. Yeah. Okay. Fine. Okay. Yeah. Sorry. So taking the bioactive food compounds might influence your overall health, and the exploration has led to the creation of nutrigenomics and gene-nutrient interaction. Nutrigenomics is not only about the genomics, it is also includes the biochemistry, the metabolome, the proteome, the epigenetics, and even the microbiome. We should have to look at a multiple approach or multi-omics approach instead of rather only genomics, because genomics gives you the prediction, but what is happening now is being by the protein analysis, and also the metabolome tells us, and even the biochemistry tests tell us. Next slide. So the nutrigenomics, the key concepts are the gene expression, how genes are turned on or off, or what are the epigenetic changes, and also the polymorphism that is being taken out, which is in the form of the genetic variants, which is also affected by the nutrients. What are the benefits of nutrigenomics? The personalized nutrition, tailoring the diets to individual genetic profile, disease prevention, reducing the risk of chronic disease through target nutrition, and optimal health, enhancing overall health and well-being. Nutrigenomics, it can be applied into the clinical practice. The research is still a long way to go. We have to have more data which could help us in the dietary guidelines, and also important for public health, designing the population-specific nutrition program based on genetic data. At Avastagen, we develop the blend of innovation and nutrition-based product. We conduct a robust R&D, ingredient-based pipeline, which have the proprietary technology MetaGrade. All the botanical actives that we use are natural, derived from plants, and also the coloring agent. We take care that the products are sugar-free, nut-free, dairy-free, and these are all scientifically validated bioactives, and which can go into various food formats like capsules, gummies, powders, and also into the nutrition bars. At Avastagen, we have a discovery platform, the Adapt Avastagen Discovery Platform, which has the information derived from the traditional wisdom of Ayurveda, Siddha, and Unani. And we have a compendium of medicinal plant information, and also the active and bioactive compound, and the research work that we have carried out on various health conditions like diabetes, bone health, cardiovascular health. All these products that we make goes through the stringent preclinical cell-based assay, preclinical and clinical efficacy test, and also we take care of the scientific validation and also organoleptic optimization. The other proprietary technology that we have is the MetaGrid. It's a metabolite fingerprinting technology. This is the snapshot of the database that we have for the plants, and this is the metabolite barcode platform. Every dot you see on this metabolite fingerprint represent a specific metabolite. And in the plant-based nutrition, you have multiple metabolites present in the food, and they work synergistically for the prevention of any diseases. We have, based on our criteria, we have developed the various bioactive, the diabetes, we have T-star for sugar and calorie management, Simcata for sugar management. They work differently. The mode of action for diabetes, it limits carbohydrate absorption. Simcata works on insulin, mimetic, and sensitization. We have bone health product Bonafide and Phytoshia, which is for increasing the bone, healthy bones formation, and also prevent osteoporosis condition. Phytoshia is the good product. And SmartCall and Prova, they work for cholesterol management. SmartCall works at HMG coenzyme A, and Prova works on the increase in HDL by ApoA1 enzyme. We have other products related to the brain health, the Think Well, respiratory and immune health, the Wint Well and Amla Pure. Other health conditions like skincare and weight management, we have product Hibix. For immune health, Gojimax, anti-inflammation, Xenomax. For aging, Boswell, and for mental health, again, we have good product Bacopamex, digestive health, Tifla-X, Shatavari, and Tribulus for women and men health. This, as you know, the genetic testing, this is the normal procedure where you take the swab and have the information of the variants present in it going through the sequencing data, and we can have that on the phone or a device where you can have the information of the variants that you have and can customize or design the food to specific markers that are there. This is the summary result of one of the test in skin aging where the variants are shown. I'm not going because of the time. I have been requested to put it first. So I'm just taking you through the skin aging biomarkers and variants that are there. And this is the eating habits and nutrient metabolism. Thing with the kind of genomic testing data that we get, we can recommend the products that we have for diabetes, bone health, cardiovascular health, brain health, and also for the immune health. And these are the genetic markers for diabetes. So we have the 2 products. We have 8 products in pipeline. So according to the genetic information that we have, we could suggest the specific products to the patient. This is the T-star just to showing the data, which is the clinical trial data, and we have launched this product in the form of crackers, in the form of capsule, and also into the form of gummies, and we got a finalist. The gummies were selected in the Nutrient Ingredient Asia Award. Our products are there at Amazon. And this is the Nutrient Ingredient Asia Award slide. For bone health, we have Bonafide and Phytoshia, and these are the different genetic markers associated. Another one is the heart health. The products are SmartCall and Prova for HDL cholesterol and triglyceride or increase in good cholesterol. We can suggest the product. These are the immunity and anti-inflammatory genetic markers. I have put biochemical markers as well over here because that are also important, not only the genetic markers as well. The products we have here for immune health. This is the whole bioactives and dietary supplement products for various health conditions that we have already customized, and we recommend for a specific health condition based on the genetic marker. I would like to show you, these are the 80 colored varieties of rice that we have procured from worldwide, and we have done the analysis, the nutrition profiling of these varieties of rice. We find around 5 to 6 varieties having the high ORAC value, which is the antioxidant property in this rice varieties. So we took this forward for further analysis based on the carbohydrate, nutrition, vitamins and minerals, and we have these branded in the different form based on the metabolite present in it. We have done the metabolome study of these rice varieties, and they have specific terpenoids, polyphenols, anthocyanins, and based on the metabolome data, we are designing the products for child health, women's health, and elderly health also. So it can be incorporated in various formats. So this is the whole functional food products with bioactive, which we have in the cereals, in the form of gummies, in the form of nutrition bar, in the form of powder. And today we are launching the Hibix, Goji Max and Amla Pure post this session, which is the beverage content for skincare and also as an antioxidant, and also for immune health. So concluding the future of genomics, it is the personalizing wellness through the digital health and testing ecosystem driven by data and diagnostic will be the way forward in the coming years. Thank you so much.
I invite the next speaker, Dr. Prashant Kumar Verma from AIMS, Delhi. Rishikesh? Oh, Rishikesh.
So, after taking very highly reputed lectures from different speakers, I am really thankful for the committee, madam, and organizing all supporting people for giving me opportunity to give me these presentations. My topic of presentation is pre and post. Yes, yes. Can you make like this? Yes. So today's presentation is more theoretical, like research. So specifically, I consider all wide range of knowledge of the audience to include this from basic to higher level. It is related to what are the things you keep in mind when you counsel a patient for genetic counseling. So only the genetic counseling is very, very essential part to include both pre and post counseling in the same setting. Except the other part of the medicines usually don't consider the post-testing concept in the pre-testing, but in this you need to keep a wide range of aspect of the test when you plan a test for medical genetic testing. So disclosure, I didn't get any fund support from any private or industry in this presentation. So objective of this lecture is I would like to discuss about the PPMI counseling concept, how to illustrate construct a proper PPI counseling, what are the key concept for components of PPI counseling, outline the stabilized protocols for PPMI like checklist. When you start a counseling, you should have pre-planned checklist, so you will include each component, otherwise you can miss them. Summarize the step of degree drawing and generation of HPO terms in the same setting, which is the essential part if you got a very fruitful medical report, especially for molecular team. Strategy for prioritizing the molecular test to the birth defects, how you plan for which test is the most useful, which you give priority by step-by-step manner. In India, in the developing stage, the patient's fund is very important part. So whole genome sequencing might be future the first, even they're not the answer for each disease. Like thalassemia, alpha thalassemia component is whole genome is no value. DMD, whole genome sequencing no value doing first. Even hemophilia, like lot of common disease, you can't jump to whole exome sequencing. When I noticed my resident even now the planning for DNA case, in the first year they didn't recognize their plan for whole exome. So this is very important to think which test you want to plan first. Apply cohesive approach. This is personalizations for even pre and post medical genetic counseling in each case. This is not being applied for each case the same thing. Summarize a few points in molecular testing regarding the detection abilities or genomic variation detections. At last, just summarize point because it's not the part of the presentation. So, what is the PPMI counseling? It is a scientific way to deliver the all information in a layman language, their patients, their own language and level of understanding. So they can take individual discussions about the testing and their future planning. That is the main aim of this study or counseling. The counseling preparation protocol have the two parts, psychosocial data and academic case preparations. Psychosocial data, you must get their backgrounds, educations, their pre-illness or during they find out some of their family have the issues, their psychological state. Unfortunately, Indian patients don't like to recognize their or don't want to open or don't want to know exactly psychosocial issues. So it is a very tough part, and the counselor itself should have knowledge to judge little bit about the psychosocial problem they have. Physical status of the family, some of the family have the cardiovascular disease or something, because when you open the diagnosis or open the counseling for further, it may impact the family. So you should have prepared for that. The financial support for resource for the families, pedigree analysis, you should have generated the pedigree at the time or reanalyze with the families why you are planning for testing. HPO annotation, human phenotype ontology annotation is very essential part for further analysis. Without knowing this HPO, to know the importance why we get the report, the phenotypically match report is not there, that is the most essential part. Anomaly scan diagnosis. The academic case, second is self-study for a condition. They have never complete a pre and post medical genetic counseling in one setting. We always do 2 and 3 settings. First setting is mostly the basic information regarding the family or the psychosocial data. Second, we prepare for ourselves for academic knowledge, the disease, everything that can be the literature review for each case. Genetic is unique. Study material may be prepared in the local language for during the counseling time. And is again the family able to decide and discuss about the molecular investigations. The objective beside the main aim is understand and its indication why you are ordering the test. All possible test method for the pros and cons is precision or I would say personalization of this test with regarding the families. Positive negative impact of the molecular test should be discussed in the same setting. Limitations, next investigation plan would be included or must be the part of your counseling, because otherwise you will face what the next step in the next meeting. So this is the main cycle of prepare of this counseling, data gathering. You will collect the data, analyze the data and plan for what the literature review and how we will start the counseling. It's usable for the family and they very simplified language and you can use smart tool, videos, maybe charts for overall and provide the information to the family so they can discuss, understand. You can cross check by indirectly what you understood, what the meaning of that, what you understood from that. So you can make cross check question also in the same setting. It should be very clear and with the confidence, because you are developing the confidence in the family, how they can ask you and also what the future. This is all parts should be can video recorded in a standard situation with pre-informed consent. In abroad, most in the Western country usually follow that to better avoiding any legal circumstances. So this is the part which we already discussed, the family, disease, test, impact. There are the 4 parts of PPMI. The family's education, mental health, and physical. This is little summarized form. Test, you should be planned in the 3 ways. It is a target test, it is a panel, or heterogeneous disease or whole exome sequencing or microarray high throughput techniques. And that could be the disease is you need to generate mode of inheritance for pedigree analysis and few other additional findings. If you analyze well pedigree, you can get many other advantages. HPO is the main key feature for nowadays after you get the diagnosis. So never ever try to make a clinical diagnosis like it for post-most. Try to get as much as possible phenotypic data, which is quite helpful for analysis for molecular team in future. What is the clinical impression if you get? It will help in the, again, the test planning. Impact, what the impact? There are two divisions, reanalysis of data, there are inconclusive or whole exome sequencing and other new techniques. So that is the checklist which we use in our OPD for any case we plan for pre and post genetic testing. So we don't try to miss any points. So literacy, this is upper, higher educated, middle is intermediate, low is below, lower is standard, below high schools. Psychological illness they have in the family, no have, don't have chronic disease in the family, or there any birth history which is very helpful to rule out what is the mechanism of the dysmorphologies, the birth defects, which is very, very important because up to 5 to 10% families they have the non-genetic mechanisms for dysmorphology, which you should keep in your mind. Otherwise, you will unnecessarily make testing and you will not get any results. So birth history is very essential part. So it is a part in the your history, yes or no, because when you plan further, it is if the birth history is significantly deformations or sequence or other mechanism of genetic deformation, this birth defect you should be keeping considerations. Socioeconomic is also helping the planning or molecular testing, the fund, what the family is getting, provide the fund or research or the fund, if people are able to, you get the another way. Projects, you related to the project in the research or not, it is a diagnostic yielding, what is the diagnostic yielding of this diagnosis of this test is there or not. So second checklist is disease. So after the families, the disease is familial or sporadic. The mode of inheritance is very clear or it is non-Mendelian. It's very clear which kind of that is. System, which system is involving, we usually make the tick marks. So again, they help with the molecular team and final analysis. HPO mass terms 1, 2, 3 you will get from the source. HPO websites. Clinical impression is clinically confirmed. It is clinically confirmed or clinically suspect or undiagnosed. Family pedigree analysis is consanguinity, pleiotropy, anticipations, the age-dependent penetrations, many advantages of making that very good pedigree, which is basic route in the all counseling. Followed by psychology, families, this is after that I think next is test, the cost, turnover time, is reliability, it is target test. If you plan, then it should be based on the what is the exact diagnosis you make. So you will go proceed with the target testing, not for like a specific whole exome sequencing. The target testing either you plan for basic cytogenetic test or the target molecular testing. If feasible, what is available in the institutes. MLPA and microarray for again, this is again you are what is the planning for target testings like ML for DMD. If you are thinking microarray, you are planning for particular trisomies for a antenatal testing panel, Sanger on NG best, which is again 3. Microarray again low resolution type, high resolution type, exome sequencing clinically or holograms. And what you will do in case of which is undefined, means which is the case is usually is not come. Impact, what could be the impact in the family? Impact on the patients, impact on the parents, impact on the family members. If the impact is positive, which I made the green is very good, but we need to consider the red ones, psychological stress, the prognostic point of views. This should be discussed at the same time. If you not discuss, it is the impact of non-carrier in the family as we reported. There are 2-3 members all carrier, one is not carrier. It may be because of the thing he is not similar with the other family members. Parents should be carrier protocols. Should be plans, the family planning for future, not a carrier, then it will again lead to psychological impact. Should be discussed with the family members, but they can plan for other next relatives and then psychological stress.
2 minutes.
Okay. So this is This is the written consent format. It is a family plannings for primary findings, primary finding which you are targeted, and secondary finding which you are not per directed, the review guidelines. So this is a pedigree design which is steps very well known. So that we use for specifically anomalies detected in the major, minor, anthropometric, and that is the dysmorphology checklist. You can check, like this is also we have the very well designed all the body parts and whatever involved. And Maelstrom's HPO, you can use that websites which can you help you to well define the dysmorphologies. First, focus on the dysmorphology, not on the making a syndromic diagnosis. So that is again where I will use that to particular phenotype. You will get a different Maelstrom's. As you more finer and finer in dysmorphology, you will get better outcome. So this is the mechanism of the dysmorphology. This is more theoretical, but I discuss is very, very useful to mention in the planning, because you should include the intrinsically abnormal, then you will get more positive results. So what are the factors helping the genetic counts overall investigation planning? Clinical impression and clinical diagnosis first. And what is the other secondary? Before doing this, you need to have the basic baseline test. Without the baseline test, which is in a pipeline planning, never proceed in hurry. You should have the basic test, then proceed to second molecular test. Because majority of the clinician is run toward the molecular disturbs without having the basic test. So without having the basic test, if you run to the secondary, it may lead to a lot of problem with the molecular team and exact HPO. So strategy of priority molecular tests, I usually use, or the recommendation you use the NGS gene review based database, where you can decide what is the disease exactly, what are the recommendations. If they are not there, you can use the genome, which is give the resource which test is based on the mutation database. It is a deletion, this then you will proceed with the MLP or particular one or study of it is point mutation, you can go for whole exome sequencing. So final decision is decided by the various factors: clinical impressions, cost, diagnostic yielding, family preference. This is the most personalized way. You should not advise, you are just informing them. Let them decide what they want to choose. What is the first, second line early versus late or availability of the test? So this is a combined approach which is basically personalizing for testing for each family. So this is just like summarized form, what is the overall genetic testing plan for the families. So low through output, high through output. And the last, second last slide is related to what is the resolution of particular tests we use. Again, the more theoretical, what their resolution capacity to identify the copy number variations. So the whole exome sequencing can detect point mutations, single base CLV up to the smaller size 10 kb or 10 exons. What are the limitations which can be overcome by the whole exome sequencing? But proceed to whole exome sequencing. I always recommended to reanalyze your data, which give additional 50% yielding, and reanalyze the patient phenotype, which give additional 10% yielding. So cumulatively, up to 40-45 to you will reach to 65 with reanalyze the whole exome data. So whole exome data give additional 50 to 20%. So before jump to whole exome, which is a very cost restricted things, you will need to proceed with reanalysis of the data, and a lot of things you can additionally. This is the resource. So thank you. I'm happy to any question now.
I thank all the speakers. You all agree that they brought different perspectives. So I think they are now invited for a discussion.
I would now like to request all the speakers to join us for the panel discussion. Request the chairs to initiate the panel discussion.
Yes. Yeah, Ada also here. So as I mentioned that it's very diverse but very useful talk. You all agree with me, and maybe I'll just initiate first question to Vinod. If somebody want to do the whole genome sequence, who's supposed to be apparently normal, so what do you advise?
Yeah, so if there is a apparently normal individual who would want to do a whole genome sequence, I would advise yes, because of two reasons. The first reason is that you will uncover a significant number of unknowns that you don't know yet. And I can put a number to it. 80% of individuals could have a pharmacogenomic testing implication as of evidence today. Not yesterday, not tomorrow, today. 3% of individuals would have an incidental finding that is actionable. That means there is a clinical path forward. 10% of individuals would have a carrier frequency which would implicate in your family or decisions in your reproductive life. So put together, almost 90% of individuals would benefit today from a whole genome sequencing. If you can afford it, I think you should take it.
I differ from this. Right. So purely based on the report which I have seen, some of the organizations, we are apparently normal. Apparently normal by looking at the phenotype. But we all carry all the mutations. Every gene will have some mutation, but probably they are not expressing. We are not getting the disease. Maybe nature of the mutation and age of onset, and also there are several factors which probably not allowing that mutation to express and cause the disease. I've seen the report saying that categorizing, you have 20% of having diabetes, you have 40% of having cardiovascular disease. So that, in that kind of scenario,
Mendelian disorders,
correct.
Yes. I'm talking about Mendelian disorders.
Yeah, I agree. So I wanted the audience to know that this kind of things also exist. So they should be very careful in interpreting the result. Yeah.
In COP, the post-test all the consequences, even the medically actionable testing, and already in recommendations. Or mainly, if you don't have the guideline, better to don't do it. More better.
Yeah.
If you already have well-established guidelines for something and they follow the ethics, the four pillar of ethics, negligency, no, never, beneficence, justice, autonomy, then you can proceed with fully discussion. It is nothing wrong to do with the family. The person is individually want to whole genome sequencing after understanding all the circumstances, but the testers should be testing for selected genes, not
so
for all.
Ultimately, the information what we have to convey is that we have to do little more research to find out what is the impact of some of the mutations. As Vinod pointed out, that if it is involved in, for example, pharmacogenomics, and somebody is taking drugs, so what is the implications of that? So that kind of thing needs to be conveyed to the patients. Of course, that's all of our.
Before we become a patient. Correct? Because once you are taking a drug,
Already well-established guideline by American College of Medical Genetics.
forget American College, we will talk about here. And I wanted from you to tell me about Rishikesh. In AIMS there, how are you going about this whole genetic counseling thing? Are you again doing it patient-based, or are you looking at areas? That's what I wanted to understand from you.
Patient-based.
You do it.
We do in patient-based. Whatever I get the referral from different specialties or in my OPD, I do myself most of the genetic counseling.
Can I just ask? You want to pass?
So
can we start a proper genetic counseling unit? And once any one aim starts with it, then it becomes easy for other aims to start with. And as we had incorporated yoga trainer for students and everyone, so why not to patient we can add a genetic counselor as a permanent post. So
I am not the right person to answer.
I know, but it is just my because sir,
in the patient load there, I am doing myself. This patient load. If there, I request usually is 30 patients per week. So it is. So I do myself, and actually for opening any new position is not in my hand. We don't do the outreach program at present time.
And Annie ma'am, I just wanted to add on to this. Ministry has given us proposal to start new courses also. So we can start with the.
I already started the plan is submitted for MSc Medical Genetics course.
Okay, sir. So why not for
Counseling. Medical genetic counseling course.
Okay, so in future, if you can share the curriculum, so we can also follow, sir.
Yeah, sure.
Yeah, thank you.
Yeah. Hi, Tangaraj, you opened the can of worms. And as a geneticist, people like, who've been in the domain for such a long time, things have moved very ahead in the world. And today, almost all major countries are sequencing their entire genomes as actually an economic tool so that they know what their people are carrying and therefore they can plan in the future for their healthcare costs, for how much they will invest and how much has to go towards healthcare. So it's much more than, one is, of course, it's individually driven and it will be individually led. So you're making a choice to do the test, but ultimately it's not about how much you found today. You know that once the genome is released, you will discover for times to come because technology is going to improve and reveal much, much more every time. So we cannot say, let's learn a bit more about the genomes and the outcomes of it, because today it drives further discovery. It will drive us to be healthier. So collective genomes will add to improvement of healthcare. So I would encourage that.
Yeah.
Somebody wants to know. No, somebody is paying for that exclusively. They want to know about their variants. So that is the problem. So otherwise, sequencing and making use of it for research and finding more and more in the future, there's no issue.
Hello, there is going to be a little bit of difficulty when you are going to have a number of genomes, whole genomes released, which is very good. It is going to be a discovery tool, and we are also going to interpret a number of variants, and that is going to be very tough, and that's not very easy. It is not just bioinformatics applied and putting the databases, etc. Because I'm also a part of that curation and annotation, consortium, which is done by this Bangalore College of Medicine. But there again, there is always a question that what is the experimental evidence? Do we have, in addition to the experimental evidence, what is the treatment response? So it is going to take us a little time, but somewhere we have to start. And somewhere we have to also see how the data can be managed, and it shouldn't become too big for us, too unruly for us to handle. So that has to be also seen. I think that might be faster than getting the experiments and following up and getting the responses of the drug response in the patients. That is why I'm saying that the building the cohort and keeping them and following them up is also very important aspect of this, the whole job here in this genetic counseling.
Yeah, just one point which you mentioned.
Adding to these people's earlier and even Annie, I think now maybe some of you are aware that already in USA, a NIH project for newborn screening the whole genome sequencing NGS started. They will be coming out, and within few years they are going to put it on all Asian countries and India newborn screening. And that time we are going to say yes, we should do. Because we always take it from the West.
No, it is not that. We should do even now. We absolutely agree on that point. We should do, but who's going to fund us is the big thing. We should do it now, but we must know how to go about it in a careful manner.
Yeah, I'll come to that. So I completely agree with Dr. Patel's observation that it's actually an economic tool. There is enough evidence today to suggest that you actually recover every single penny out of that in your entire lifetime. There is evidence to recover. Question number 2, when should you do genomes? To me, the best economic outcome, not on a scientific standpoint, just a pure economic standpoint, the best time to do it as a newborn. What we need to think is that we all were born in an era in India which was very different from what we are living today. And we should also anticipate the child that is born today is going to live in a different India than what we see today. So we should not base our assumption about what our past experiences are. Yeah. I was born in an era when Indian per capita GDP was less than $1,000. Today we live in an India which per capita is around $2,000. Our children will be born when they will become 24 or 25 years, where the per capita GDP is around $4,000, $5,000. Right?
Can I make a comment at the back?
Yes, yeah.
On the same conversation that Thangaraj and Vinod is having. So yes, as Animam said, US is doing newborn screening. UK is now doing 100,000 genome newborn screening after doing 100,000 adult screening. And I think Vinod and I probably are on the same page on this, that whether we like it or not, genomics will become a consumer item. We are only thinking about genomics from disease point of view. People are curious who we are, where we come from, how can we find out. If they can afford it, they will get it done. You can't stop it. It will become a consumer item. So that's what we have to understand. As Vinod rightly said, that we are looking at a different world. And how do we use it? How do we make sure people are not misguided? That is how we as genetic counselors can play a very, very important role. You can't stop people from having their own genomes done. You cannot stop it.
Sorry to interrupt, we can take one more quick question from the audience. I just want to add to Vinod's and the diagnosing earlier with the genetic test, if it is done with the prenatal babies, then you have a chance of preventing it further, both providing proper nutrition to it and proper medication that is required. So that would be the best approach. So rather coming to know very late when there are no further prevention or the cure possibility. So we can take that very well in the beginning itself. And also I want that the genetic counselor should also look at the nutrition aspect as well, because it's not only the genetics, it is also the what it is feed, because it is the interaction between the nutrient and the genes. Thank you.
Sorry, could I just make one last comment and one question, right? As a geneticist, I completely agree with whole genome sequencing, the comments you're making. At a personal level, I've had my own whole genome done, by the way. However, if you are in a counseling session whereby a couple has come by, they're about to get married, there's a lot of family pressure to know whether they are carrier or not for some sort of a disease. Now, if you have had your whole genome done, and let's assume girl is a carrier for a pathogenic BRCA1 variant. Now, what would you do at a social level? How would you counsel that? Because the boy's family is not willing to accept that risk. Now, how would you go about it?
Yeah, it's actually, sorry. Actually, it's the wrong question to ask. Right? You will identify carriers, and there are countries where premarital counseling and genetic testing is mandatory.
Okay.
No, just a minute. Let me complete. What you do carrier screening and what you want to prevent is a non-treatable genetic condition. Right? If it is compatible with normal adulthood, you would not want to intervene upon it. Right? That is the basic principles of counseling and action in genetics. But for example, there are countries, the best example I can say is the state of Dubai, which implemented a premarital mandatory genetic counseling and genetic testing for thalassemia. And in a decade's time, right, Maldives also has cards, yeah. But in a decade's time, they have eliminated children born out of thalassemia. Just think about it. Right? They've eliminated people born out.
I'm saying they're about to break off the marriage.
No, 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 no. No. Why should, my point is, why should you reveal the career status of your prospective spouse to your prospective, right? That should not happen, right? How do you, can I?
Very enthusiastic audience, I must say, but in the interest of time, we have to complete the session, sir.
There is, wait. There's a mixed opinion, mixed discussion. I think as a counselor, let us.
So, see, as counselors, we think differently. So what he said is right and what Dr. Vinod you are telling. So whether you are counseling a couple who have come to you as a pre-marital or preconceptional, you have to follow the ethics. There are guidelines, SOPs, and even the companies, whether when you find a BRCA, not many companies, not all of them give a BRCA mutation. It's not supposed to be told unless you talk to the family. No, you should not. It's an incidental finding. You ask them whether you want to know, and it is the it's the doctor's responsibility, the responsibility of the counselor to put it in the report. And without pre-test counseling, first of all, carrier screening test should not be done. Okay. So if okay.
Yeah, I think. Who has a question?
Yes, we have Parimal online who has joined just to ask a question. So, can we take that question?
Oh, Parimal, you have any question? Hello.
So we spoke about the timing of genetic testing, right? And I think everyone agrees that earlier is better. So my question is, the world is moving forward, right? You have Silicon Valley where you've gone beyond early stage. Now you're talking about longevity, right? So when do we start thinking about accumulated mutations, right? So that is something that plays a big role, whether it is telomere shortening and so on and so forth, right? So when in fact we need to go beyond that. So how are we looking at extending life, improving life later?
Yeah. So really seen that recent paper that came out of, I think, Iceland, the large population genome, right? Just buried in the last paragraph of that, what they've shown is people who have got the testing and have acted upon it have extended their life by 1 full year. Right? No, just not joke about it. Their per capita is $20,000. So that means that every individual has actually got $20,000 contributed to the economy. And that's huge.
Yeah, Parimal, you have any question?
So meanwhile, sir.
Parimal, could you hear me? Parimal?
Hello?
Yeah. Please ask if you have any question.
Can you hear me?
Yes.
Yeah, I have two questions. Our question went very well, and all the people went very well. I have the same question probably which have already been discussed, but it's pretty obvious when we know all the theories and that we are not supposed to influence the patient to take a decision. But in reality, you will find that patient is confused from starting from karyotyping to whole genome sequencing, and they need a physician guiding. I know that law doesn't allow you. What is your opinion that what something should we try to modify to help really pull the patient to take a decision?
So you are addressing to whom? This question? The question is to whom?
Parimal, we are there to do counseling, correct? And the counseling tells you the implications of the test, the relevance of the test, and also the incidental findings, giving them the options to do it. If like Harish was saying about breaking a marriage can be broken for 10 million things. This could be one of them. But we never see to it that it's broken. I always ask the couple, first of all, do you want to marry? That's the question. Do they want to marry? Otherwise, they will find 10 million ways of, they can break up for dowry for all I know.
Highly engaging panel discussion we had. So I want to ask questions for Deepak Modi.
One question, please.
Deepak. Deepak, it's like you were talking about IVF and thing, and I was telling you we are working on Duchenne's in huge numbers, and we have at least 3 cases of Duchenne after IVF. So more to say regulations, anything we might do, but unless you have a proper process and regulations and a structure to do all this, it will go haywire. So more, if you can do something about the regulation, it's going to be really helpful.
So regulation-wise, the ART bill has come into practice, and it's normalized a lot of genetic-related testing amongst couple with undergoing IVF. The problem being carrier screening is still not a part of it. And the problem is precisely the thing which we have been discussing all the time.
See, again, the mother didn't even know which egg they have used and what happened, nothing else known. At least at the level of before implanting and things, if there's testing being done, at least for these lethal genetic disorders.
Absolutely, that will require an entire program of carrier screening. The problem is, do we know the top 20 disorders or top 10 disorders which you want to do a carrier screening in India?
We should.
1. 2, the problem is, do we have enough people doing PGTA in the country? Because doing a carrier screening and telling the couple I don't want a baby doesn't work.
Yes.
We have to give them an option of doing a PGTA. Do we have labs doing PGTA in the country?
So that is the next step. There are
one in the country. One is not going to suffice the total number of people. That's precisely what the IVF clinicians ask me. That okay, we will do the carrier screening. We'll tell the couples. The problem is, do we have a solution? Can we tell them not to have a baby? That's not an option.
That's not an option.
So then, do we have labs offering PGTMs?
How many?
That the numbers just don't match.
More in this crowd, I would want to say we should also think about regulation and process where we need to contribute.
Now the regulation comes when the solutions are available. Do we have solutions for the problem? I am in no phase to tell the clinicians do a carrier screening because this is what you're getting into, because they ask me where is the solution to this. If there is a couple, both of them are carriers, what do we tell them? Do a PGTM? And then they ask me, Dr. Modi, PGTM kahan hota hai? I say this lab. And that lab is not geared to even have done PGTM for 5 of the disorders.
After the thing, because I'll talk to you before thing.
Thank you everyone for such an engaging panel discussion. We thank all the speakers and all the chairpersons and the audience also for keeping the panel discussion such an engaging one. Thank you.
Now I would request you all to stay for a group photo. Yes, please. Group photo 1 minute.
I request all the board members and certified genetic counselors to please stay back for our AGM. And all the delegates and participants, I request you all to please exit from the left side of the auditorium for lunch, because the right side is blocked. So I request everybody to exit from the auditorium towards the left side. Thank you.
Dr. Thangraj, I would request you to give the mementos to our speakers, please.
The speakers, yeah. Give it to Thangraj, sir. Come. Yeah, le lo.
to Dr. Deepak. Dr. Vinod. Annie ma'am. Sir, Tangraj sir. So, group photo please. Dr. Vinod, please stay on the stage. All the board members to remain in the hall for the AGM and all the rest to proceed for the lunch towards the left of the conference hall. All members, yes. All BGCI members should remain back in the hall for the annual general board meeting. Ma'am, Annie ma'am, to Dr. Jain. Yeah. All speakers on the stage. Sir, group photograph. Ma'am, please stay. So हम एक फोटो ले लो. This session group photograph. अच्छा.
Check. May I please request all the members of the Board of Generic Counseling to stay back? And the remaining delegates can proceed towards lunch. Towards left. May I please request all the members to settle down fast? May I please request all the members to settle down fast? Take the others to the
polyol pathways, exaggerated oxidative stress, and overactivity of protein kinase C, as well as increased formation of advanced glycation end products. So initial, you know that it's a cascade. So initial point is increased amount of glycated end products, which leads to a lot of other alterations in this cascade. So what are the consequences? You know that it is, yes, it is a debilitating condition where that severe pain, neurogenic pain, where by using medicine, it's very difficult to maintain that severe pain. So it is a cumbersome pain. And also they develop lack of coordination with the voluntary muscles other than this sensory involvement. So because of all this, so they are susceptible to ulceration and ultimate toe and foot amputations. So this is usually 15 times more when compared to the non-diabetic patient. So we have identified more than 80 loci that is associated with the diabetes mellitus, but on that, there are a lot of other susceptible genetic loci associated with the diabetic peripheral neuropathy, which gives you a nice network of association. I will be explaining it later. And according to the literature, so it was identified that there are differences with some diabetic patient with high level of glycated products. They are not prone to diabetic peripheral neuropathy. But even with the short standing uncontrolled diabetic mellitus, they develop severe form of diabetic peripheral neuropathy. Why? So that is the point that we need to address. So there will be a genetic predisposition on these people. Right. So this shows clearly the mechanism, the pathophysiology of the diabetic peripheral neuropathy. You can see that all the hyperglycemia with the increased glucose level, with the increased protein and advanced glycated end products. And also, you know that with the dyslipidemia, it's another important fact that it's associated with the diabetics and the complications of the diabetes. And same time, insulin receptor as well as insulin resistance. And these are the more common mechanisms that involve with the diabetic peripheral neuropathy. What will happen ultimately? So there will be DNA damage, endoplasmic reticulum stress, mitochondrial dysfunction, apoptosis, and loss of neuropathic signals, which leads to hyperactivation of macrophages. Ultimately, the cell damage. Especially, we know that the Schwann cells of the neurons are more prone to get damaged with these hyperactive macrophages, will lead to neurosensory impairment in these patients. So, this is very important. Why we need to understand this diabetic peripheral neuropathy? Because understanding the mechanism is very important, not only for the clinical geneticist, not only for the endocrinologist. So it is important to the genetic counselor as well, because my belief is the genetic counselor should be a person who can understand the genetic, the etiology of the disease, as well as the mechanism of the disease progression. Then only they can link whatever the genetic variants, and they can interpret these variants related to the mechanisms and the prognosis as well as the risk of these disease conditions. Right, so there are several studies that shown there are different genes that are associated with the diabetic peripheral neuropathy. I'm not going to discuss one by one because I know that the time is precious, so I will let few minutes for the discussion. So I will just rush this because there are studies they have done and they have identified key genes that are associated with this inflammatory response, cell damage, and other mechanisms involved with the diabetic peripheral neuropathy. So on that, most of them are associated with the polyol pathways as well as increased inflammatory response and cell apoptosis associated with the advanced glycated end products. So I'm rushing these different variants, right? So anyhow, my presentation is there, so we can share the presentation. You can go through those all the variants. But I want to go to the next session where, yeah, this one. So this shows a network of genes that associated with common variants of the diabetic peripheral neuropathy. So this is what most of the genetic counselors need to focus on, right? So it shows that glucose, lipid, and lipid metabolism, cell death with the inflammatory response, and the cellular homeostasis with the cellular response. These three are linked with these key genes, and that is associated with subsets of other common genes that leads to enhance the responses of these the inflammation as well as the cell damage. So by understanding this, you need to link that with the mechanism that I have shown you earlier and how these variants of these genes are associated with, especially whether it is increased risk or prevent disease progression, as well as it also indicates it's part of the pharmacogenomics that the response to the drugs. So that is also very important to understand whether, because the main idea of controlling diabetes is to controlling the blood glucose level throughout the 24 hours. It is not the fasting, not the postprandial. It should be 24-hour control. So that has to be done according to the particular individual's genome and its variants that are associated with that. Right. So what is the role of genetic counselor? So as I mentioned earlier, so genetic counselor, it should be a person who connect the patient with the clinician. Therefore, that particular genetic counselor need to be a patient side as well as need to be a clinician side. So therefore, for the purpose of having a better knowledge on the disease progression, as well as its mechanism, as well as the response to the drugs, they need to know the mechanism, the pathophysiology of the diseases, especially these common disorders which are polygenic, associated with a lot of other environmental factors. So with that, there has to be a pediatrician, there has to be a physician, there has to be a clinical geneticist, as well as there has to be a genetic counselor, as well as this has to be another psychologist to convey all this into that to the patient. Right. So, in perspective of the genetic counselor, so they should know what are the different variants associated with the diabetic peripheral neuropathy related to this network of genes. So here I have included few the common susceptible genetic loci and its variants and how they are associated with the genetic peripheral, the diabetic peripheral neuropathy, and so this list includes that variants associated with the defense response and inflammatory response in the pathogenesis of diabetic peripheral neuropathy. We all know that the inflammation and the defense mechanism is very important, very crucial in case of increasing the risk as well as the preventing the disease progression. So if there's a inflammation, the risk is high, with associated variants, it means the patient is more prone to get diabetic peripheral neuropathy rather than in others. Same time, if their defense mechanism is more enriched and the variants are more towards that, so that means they are not that much prone to get this inflammatory response. So by identifying these variants are very important in that sense. And the variants associated with glucose metabolic response, metabolic pathway and PPR signaling pathway in the pathogenesis of TPN. So this is other important thing, that is PPR pathway, because this also involved with the intake of glucose into the cell as well as metabolizing the glucose within the cell. And this PPR pathway is very important to prevent, it goes towards the polyol pathways where it causes a lot of free radicals and cellular damage. So the gene variants associated with that, we need to understand how that involved with this diabetic peripheral neuropathy. Right, so you know that even in the diabetic peripheral neuropathy, there are different phenotypes. Right, so it's a range of clinical symptoms and signs where it's starting from mild to severe, where most of the patients are presented with glove and stocking type neurosensory defects, where it leads to ulcerations and infections in these patients. Other than that, some patients will develop some autonomic dysfunctions, leads to autonomic misbalance and abnormalities, especially involved with the bladder functions. And you may have seen that patients with the sweating, even during gestational setting, during the eating. So mainly those are associated with these autonomic dysfunctions. And also, we have identified that there's an important gender dysmorphism. So that in male and being a male and being a female, and there's an important variance associated with the increased risk and the prevention of the progression of the diabetic peripheral neuropathy. So that is again another aspect that we need to understand, and these are the important variants that we need to identify in the either whole genome exome or the exome analysis. Right. So other than that, there are in the literature there are a lot of other pathogenic susceptible genes are associated with the progression of the diabetic peripheral neuropathy. And other than that, what I want to express here is, yes, diabetes is growing. So according to the WHO information that they have mentioned, that the pre-diabetic level is now grown up to the 60% of the population. So as it is a polygenic associated with a lot of environmental factors, there are a lot of environmental factors that we can modify, and for that we need to understand whether that particular individual is more prone to that particular complication or not. So therefore, there's a very good and very high chance of identifying these important variants related to the diabetic peripheral neuropathy. And there's a important area that a genetic counselor that can explore related to the diabetes and its complications. So here again, I'll show some other loci and variants that associated with that. So especially when we talk about diabetes, so we know that it is an increased glucose level, as well as we know that abnormalities of the lipid metabolism also will be a cause. So that ApoE gene variants are also associated with the DPN. Yeah, this is also some other variants that associated with that. So I'm just rushing all these things, and we all know that about the AC gene. So angiotensin converting enzyme is the one of another important one, where that DI is more susceptible with, more protective with, and with the DT it's reported to be more
2 minutes, if you can start to wrap up.
okay, right, more protective. So therefore, the common genes, so those are the actually the common genes that are associated with the diabetes and diabetic peripheral neuropathy and its variants. So what I just wanted to stress in this session is, yeah, I'm just rushing to the conclusion. Yeah, so next thing is the background and the different population. So for the variants that identified in different populations, they have shown that different populations are prone to get DPN with the susceptible genetic factors. So it is another point that we need to understand. So we have these ancestors are from where and what are the ethnic background, so on. In conclusion, right, so we all know that it is that the targeting gene panels, array technology, as well as the NGS are the common platform that we are using for these diagnosis purposes and screening purposes. And where we are, though we are more towards the monogenic disorders and identify the monogenic disorders, and the prevention of polygenic disorders and prevention of its complications are also very important. So therefore, it is important to look for new panels, new criteria, guidelines to identify these risk patients and to prevent these complications. Thank you.
Thank you. We now have a third speaker for the session. That's going to be delivered online, right? Third speaker is Tania Noel Abil. I hope I'm pronouncing it correctly. She is currently holds a faculty position as BCM as an assistant professor in Department of Molecular Human Genetics from 2021. She has done her BSc and MSc from Penn State and University of Pittsburgh, USA. She is board certified by American Board of Genetic Counselors. And as you can imagine, she has various accolades, including multiple peer-reviewed journal publications and membership in prestigious societies. Can we have the talk?
Hello, I'm Tanya Edley, and I'm a certified genetic counselor practicing at Baylor College of Medicine. I was so happy to be invited back to speak with you again this year. Today, I'll be talking about familial hypercholesterolemia in adults. As you may know, familial hypercholesterolemia is very common, affecting 1 in 250 to 500 individuals across the world. As you likely know, FH is characterized by very high LDL cholesterol, which is strongly associated with premature coronary artery disease. Genetic testing is available for FH, and when affected individuals are identified early, there's management that can reduce morbidity and mortality. For the purpose of this talk, I want to zoom in for just a moment on what is known regarding familial hypercholesterolemia in India. Although a considerable portion of the world's population are Asian Indians, we have limited knowledge of the pathogenic variants of NFH in this group. This paper from the Journal of Clinical Lipidology describes a study that identified 38 pathogenic variants associated with FH, 10 of which were novel. They also reported a pathogenic variant in LDLR, which was identified in 6 North Indian families and was hypothesized to be a likely founder mutation. Of note, the variants observed were different from those described in Western populations. Gathering more information about the variants in this population may improve our classification of the variants and improve our ability to diagnose. Data from this paper was also used in the review paper that I'll discuss in the next slide. In this paper, the authors looked at 6 studies. The authors report on the heterogeneity of pathogenic variants in the LDLR, ApoB, and PCSK9 genes. In the United States, prevalence of FH in young adults with myocardial infarction is nearly 10%, which is about 20 times higher than that observed in the general population. The authors of this paper state that only prevalence study shows FH in 15% of patients with premature CAD in North Indians. What we don't know is the prevalence of FH in India overall. The authors of this paper argue that establishing an Indian FH registry can help to better understand the prevalence, provide data to better classify variants, and lead to earlier diagnosis. Earlier diagnosis is important because early detection, followed by aggressive therapy such as with statins or PCSK9 inhibitors, can reduce the morbidity and mortality of affected individuals. Also, cascade screening of extended family members can lead to improved diagnosis rates. Hopefully, with the previous 2 slides, we're all on the same page regarding the importance of genetic testing for FH. Now, I want to zoom in even more to a specific patient. Here is an example of a 37-year-old woman who presents prepared due to chest. Her echocardiogram is normal, and you can see here on her lipid profile that her LDL is high. She shares that her father died from a myocardial infarction in his 40s, and this leads the cardiologist to recommend the patient talk with a genetic counselor. So our patient goes to the genetic counselor. Our knowledgeable genetic counselor knows that familial hypercholesterolemia is an autosomal codon inherited condition. There is the somewhat less severe form that an individual has if they have only one pathogenic variant. This is the more common form, and individuals with the heterozygous form typically develop cardiovascular disease in middle age. With these individuals, you can expect to see LDL levels greater than 190. We also have the homozygous form, which is much rarer and leads to development of CAD in the teens or sometimes even earlier. With these individuals, you might expect to see LDL levels greater than 500. So when should the genetic counselor or other healthcare providers suspect FH? The first thing to consider is lipid profile. When the LDL is greater than 190, a diagnosis of FH should be considered. Another factor to consider is a history of coronary artery disease. Coronary atherosclerosis develops at an early age, and myocardial infarction and angina may occur around 30 to 50 years of age in untreated men, and around 50 to 70 years in untreated women. Looking specifically at patients who have a history of coronary artery disease, about 1 in 30 will have FH. Of those patients who have premature CAD, meaning CAD developed at 55 years or younger in men or at 65 in women, then we will see that 1 in 15 have FH. The next thing to consider is whether there's family history of premature coronary artery disease or familial hypercholesterolemia. As genetics providers, we're likely to ask when taking the family history if any family members have a known pathogenic variant for clinical diagnosis. However, even if there isn't a known diagnosis of FH in the family, if we see a history of premature CAD, early MI, or elevated cholesterol levels, then further work at this needed. We can also look for physical features. Here you see corneal arcus, which is a light colored ring around the iris of the eye picture on the upper left of the screen. We might also see xanthomas, which are yellowish plaques that develop on the eyelids, Achilles tendon, and elbows. Here you see it on a person's hand. Another feature that can be seen are xanthorrhosis, which are yellow bumps on or near the eye that are caused by cholesterol deposits. Here are some additional photos of xanthomas. Keep in mind that these photos might be more severe presentations of what you may typically see when a patient has xanthomas. The point I want you to recall is that the xanthomas show up on skin in areas that are subject to mechanical stimulation. Also, not having xanthomas does not mean that a person does not have FH. 20% to 30% of patients with FH do not have xanthomas. However, do see interdigital xanthomata, then this is virtually pathognomonic for homozygous FH. Also, the corneal arcus that was seen on the previous slide can only be seen in about 30% of patients with FH who are younger than 50 years of age. One of the challenges with evaluating adults is that they may have a variety of other comorbidities. Keeping this in mind, we should also consider that there may be secondary causes for hyperlipidemia, such as diabetes, hypothyroidism, drug-induced high cholesterol, nephrotic syndrome, static liver disease, and familial combined hyperlipidemia. At the bottom of this slide, you can see a graphic of the Fredrickson classification of hyperlipoproteinemia. As you can see, there are a variety of hyperlipoproteinemias. The next slide gets into how we can differentiate between these. This table lays out the subtypes of hyperlipidemias in more detail. Familial hypercholesterolemia falls within type 2A in the original Fredrickson classification system. In type 2A, you would expect to see an increased level of LDL, but normal VLDL concentrations. Plasma cholesterol levels are usually increased, while plasma triglyceride levels are normal. And if you observe the standing plasma, it will usually appear. So you can see that there are many factors to consider, and the diagnosis can get fairly complex. There are three main sets of criteria that are commonly used for diagnosis of FH in Western countries. These are the Dutch Lipid Network, Simon-Broom, and MedPen criteria. Though we'll briefly touch on these three criteria, I want to spend more time on the guidelines presented in this paper. A working group of the Japan Atherosclerosis Society published guidelines for diagnosis that are more commonly used in non-Western countries. Those diagnostic criteria are listed here. The diagnosis of FH should be made after excluding other primary and secondary dyslipidemias. The criteria are having untreated LDL of more than 180, having tendon xanthomas, cutaneous nodular xanthomas, and having a family history of FH or premature CAD in a first-degree relative. A clinical diagnosis is made if the individual has at least 2 out of the 3 of these criteria. A diagnosis can also be made if an individual has a pathogenic variant in an FH-related gene. The authors included a helpful flowchart to better understand the diagnostic criteria. You can see that if you can rule out other primary and secondary dyslipidemias, and the patient has 2 out of the 3 criteria, then the patient has a clinical diagnosis of FH. If the patient does not meet at least 2 out of 3 criteria, but they do have an LDL of 250 or higher, then they have probable FH. If they don't meet the criteria, but they do meet 1 criteria, and they have a first-degree relative who has an LDL of 180 or higher, or the patient has a history of premature coronary artery disease, they can be said to have possible FH. Some individuals with possible FH should be followed with lifestyle guidance, and aggressive drug therapy should be considered for those high-risk cases. If none of these criteria are met, then the patient is unlikely to have FH. Now I want to briefly touch on the other diagnostic criteria. The Dutch criteria assigns points to risk factors and stratifies the likelihood of diagnosis into one of 4 categories. These categories are definite, probable, possible, and unlikely. The Dutch criteria requires DNA testing for completeness. The European Atherosclerosis Society recommends use of these criteria to diagnose FH. Here you can see the scoring system for the Dutch criteria. A score is achieved by sum of the individual scores for family history, clinical history, physical exam, and LDL cholesterol. Individuals who have a score of greater than 8 are said to have definite FH, while those with 6 to 8 points have probable FH, and those with 3 to 5 points have possible FH. Here we have the Simon Broom diagnostic criteria. There's an online calculator that a provider can use when their patient is suspected to have FH. This is the most widely used criteria for FH diagnosis in the UK. However, some drawbacks of this diagnostic criteria is that it has limited validity in non-British populations, and it requires DNA testing for complete data. We also have MedPed criteria. Of the 3 Western-focused criteria, this is the only one that does not require genetic testing for diagnosis. Okay, now let's switch gears and talk a little bit about how FH occurs. As established earlier, there are several genes associated with FH. The main genes are LDLR, ApoB, and PCSK9. Normally, the LDL is bound to the LDL receptor cell membrane and taken into the cell. It then ends up in the lysosomes where the protein is degraded and cholesterol is made available for repression of the HMG coenzyme A enzyme, which is the rate-limiting step in cholesterol synthesis. When there's a pathogenic variant in the LDLR, then the LDL does not bind to the mutated LDL receptor, and instead, the LDL accumulates in the circulation. Now, looking at our diagram in part C, you can see similarly, some individuals have a pathogenic variant in ApoB. ApoB is required for normal receptor binding of LDL. When there is a pathogenic variant in ApoB, it prevents binding of LDL to its receptor, again leading to accumulation of LDL in circulation. With pathogenic variants in PCSK9, the LDL receptor degrades due to increased PCSK9 activity, and again, the ultimate result is the accumulation of LDL in circulation. I'll talk about this one more in a few slides when we talk about treatment with PCSK9 inhibitors. As I mentioned earlier, FH can be said to be a codon variant condition. Individuals who have just one pathogenic variant have heterozygous. There's also homozygous. Individuals may have 2 pathogenic variants in the LDLR gene. It's also possible to have a pathogenic variant in 2 different genes, such as one pathogenic variant in LDLR and another in PCSK9. Both homozygous and compound heterozygous are expected to have a more severe presentation with earlier onset than those who are heterozygous. This diagram further shows that individuals who are homozygous for an LDLR pathogenic variant or who are compound heterozygous have higher LDL levels than those who have one heterozygous pathogenic variant. There are also individuals who have polygenic variants, such as multiple LDL raising SNPs, who may have some overlapping levels of LDL with those who are heterozygous. Similar to the LDL levels, you can also see that the CAD risk increases with compound heterozygotes or homozygous pathogenic variants. Many laboratories offer panel testing for the most common genes known to be associated with FH. These labs are just some examples of those that offer panels that include ApoB, LDLR, LDL-RHP1 and PCSK9, but they are not the only labs to offer this testing. Genetic testing can be used to confirm a suspected diagnosis, provide information about CAD risk, and to make cascade testing early diagnosis of at-risk family members possible. The Familial Hypercholesterol Foundation recommends that genetic testing become standard of care for patients with suspected FH, as well as their close family members. Familial hypercholesterolemia is one of 3 genetic conditions that have been labeled as a Tier 1 condition by the CDC. These are relatively common disorders, significant morbidity, and for which screening and treatment options are available to reduce adverse outcomes. Of note, the American College of Medical Genetics and Genomics recommends that FH genes be included in the secondary findings when full exome or whole genome testing is performed. So, who gets testing? Individuals who themselves have an FH phenotype, as well as patients of increased risk based on their family history of FH, can seek testing. If they test positive but they do not yet have a phenotype of FH, then their LDL cholesterol levels should continue to be monitored. If the individual tests positive and they have a phenotype of FH, then treatment should be initiated. For those who test negative but have a phenotype of FH, then it's possible that there's an altered etiology or that technology can't detect their variant. Phenome copies also exist. The patient should be treated for their elevated LDL level, and for those with a known phenocopy, they should follow specific treatment recommendations for that diagnosis.
I
As you can
think
see in this graph,
due to time, sorry to interrupt. This is a recorded talk, so due to time, we'll have to stop the talk here, I think. Yeah, thank you. We'll move on to the next speaker.
The next speaker is also going to join us online, so perhaps IT can have the person prepared. This is Dr. Vamsi Krishna Thantam. He is the National Head of Genomics and Clinical Cytogenetics at Dr. Lal Path Labs, based out of Delhi. He is an MBBS and MD in Pathology from NIMS, Hyderabad. He also has a postgraduate diploma in Public Health from University of Auckland, New Zealand. He's been at Johns Hopkins, at Tata Medical Center, Kolkata, and also did molecular genetics at Royal College of Pathologists in UK, and is part of the CAP accreditation inspection team. He's had more than 20 years of clinical experience, along with 7 years in molecular diagnostics, with many publications. So with that is Dr. Vamsi Krishna online.
Very good afternoon to everyone. Can you hear me?
We can hear you.
Wonderful.
To mention, you have 10 minutes.
All right. Thank you so much. Are the slides visible?
We can see the slides. We can hear you.
Okay. So at the outset, thank you so much to the organizers and especially to Dr. Hassan for having me as a speaker at this really good conference wherein there's lots of beautiful topics and wonderful speakers that we've been listening to. My topic that I'm going to cover today is cardiogenomics, from genetics, looking at the genes, to genome, looking at the entire genome, genomics, or vice versa, where I've been moving to. Next slide, please. This is basically, these are the topics that I'm going to cover. As you can see, it's the cardiovascular disorders, cardiogenetics, then what is the testing that we do, genomics, and then a few case studies. When you're looking at the problem statement, cardiovascular disorders are the leading cause of death globally. 17.7 million each year that we lose to cardiovascular disorders. And WHO basically talks about India having 1/5 of these cases. And the global burden of cardiovascular disorder death is 235. In India, it is 274. I think it's a typo there. 274 in India is more than the global average of 235. What are the common cardiovascular disorders that are seen, especially these are the monogenic ones that we look at in the form of cardiac genetics, cardiomyopathy, channelopathies, congenital heart disorders, and the cardiovascular disease, including the dyslipidemias that the previous speaker was talking about. Cardiomyopathy is basically, we are looking at the monogenic disorders for dilated, hypertrophic, as well as restricted cardiomyopathy. There are various genes which are associated with cardiomyopathy, and these are obviously from different proteins that we see, including the sarcomeres, the V lines, etc., including the myosin heavy chains which are involved in the cardiovascular muscles. How about cardiomyopathy in pediatric age group? Basically, what happens in the pediatric age group is that we do not see the same symptomatology being presented. We have diverse phenotypes. These obviously are life-threatening in the pediatric population. They do not do well, which is the reason why we need to understand the disorders more in depth, looking at the molecular level. Next is the channelopathy that we look at. The channels of calcium, potassium, and sodium which are involved in cardiac action potentials, and as you can see, the genes which are involved or rather associated with these channelopathies are seen with the ionic current responsible with these ionic channels. The most common ones, the long QT syndrome. If it is a syndromic manifestation, as you can see on the left, the Gerville-Lange-Nielsen syndrome, the Anderson-Tawil syndrome, Timothy, Romanov-Ward, which obviously are autism dominant, which we see. The short QT syndrome basically is more of the potassium channel which are involved, the QC and H2, J2, and the Q1 genes. We got a syndrome basically is type 1, type 2, and it is involved in the sodium and the calcium channelopathies. Congenital heart disorders, obviously, the ASD, VSD, the top, or the coarctation of aorta. These are the most common human congenital disorders, and they are leading cause of death in infants. The prevalence is quite high, somewhere from 4 to 50 in 1,000 births, and so obviously, this is something which we have to address. These are clinically divided into cyanotic and non-cyanotic, but then based on the symptomatology, the phenotype that the kids present with, they can be syndromic or non-syndromic. If looking at these syndromic disorders, most of these that we do in and day out, using different platforms, different technologies, we kind of look at these syndromes in the form of aneuploidies, in the form of microdeletions and duplications, looked at by microarray or the next generation sequencing, as well as the important ones which are the single gene defects, which are well documented, and these are the small little gene panels that we look at. So the cardiogenetics look at these gene panels to identify the phenotype with the genotype that is documented in literature. So this is the proposed clinical genetic testing by AHA. Obviously, when you are present with certain symptoms and then we have a clinical suspicion, we go in for genetic testing, find a pathogenic or a non-pathogenic variant. That's good. At least we are able to delineate a specific monogenic disorder. But what do we do when we have a variant of uncertain significance? So that is where we kind of have to categorize them. These gene variants well. We have to look at the hotspot mutations. We'll have to see whether the protein changes and how was it in the parents of the proband. How is it in the siblings? That's how we characterize the variant of uncertain significance. We're giving it a significance in the form of pathogenic, likely pathogenic, or maybe falling back into the basket of benign disorder. Congenital heart diseases obviously have one of the aortopathies, the thoracic aortic aneurysm as well as the abdominal aortic aneurysm have a very strong genetic component. As you can see, those are syndromic aortopathies which are described in the list. The previous speakers spoke about hypercholesterolemia, dyslipidemias, obviously are a major risk factor, especially in the young adults, and the monogenic dyslipidemias account for about 5%. And obviously there is a lot of epigenetics and the environmental changes that we look at apart from the pathogenic gene mutation that we see. And each of these disorders, as you see in blue, those are the genes which are involved or are associated with each and every condition which obviously leads to a cardiovascular heart disease. It's not just only the disorder, but also the treatment, which has a bearing on what gene or what gene mutation that the patient or rather the program has. Variants in AMD1, COQ2, CPD2, CYP26, these are associated intolerance with a specific drug class of statins. So this is something which we need to know about the patients when we're studying those medications for them. Various cardiology associations, the ESC, AHA, and the Cardiology Society of India has recommendations towards genetic testing for various cardiac disorders, and this is similar to what I have shown before. The flow chart obviously goes in the form of phenogenetic correlation, and once the correlation is very well set up, we kind of understand that this disorder would be a monogenic one, or there would be multiple other epigenetics involved into it, especially when it's a variant of uncertain significance, and then it's up to us to kind of characterize that. So what are the various advantages of genetic testing in cardiovascular disorders? Basically, it clarifies the diagnosis to us in a big list of differential diagnoses, order syndromic versus non-syndromic. We're at least able to get to bottom line to at least diagnose that as a single disease that we know. We are able to identify the cause in a family. We are able to stratify the risk and accordingly manage the patient. Basically, the prediction of whether this autism will dominant or recessive variant, which is being found in the proband, how does it affect extended family members is what a prediction that we are able to do, which is the reason why we are able to counsel the patients for family planning, especially during pre-implantation genetic diagnosis, and obviously, we're able to help out not in this generation, but the next generation as well. And obviously, the genetic testing gives you a pathogenesis. It gives you an idea as to how this entire pathogenesis has happened from a mutant protein causing symptoms in the patients. Having known that, clinical management becomes very much easy, or rather very much improved for us. Because there are no ambiguities regarding the diagnosis. Obviously, there are emerging therapies which obviously are looking towards targeted therapy for such disorders, and cost effectiveness is something which we have to really understand.
Dr. Vamsi, this is 1 year, 1 minute warning. 1 minute. Please plan.
Okay, thank you. Basically, we kind of understand that there are these young adults who kind of go through these various cardiac events. Even after the cardiac event has happened, we obviously know something is called as the molecular autopsy, and so we can understand the genetic sort of cause in the post-mortem blood as well. HLD classification of variants everybody understands, the supporting criteria as well. So the CAL module in 1, 2, and 3. Are the slides below? Right. The CAL module in 1, 2, and 3 are the ones which have been included into the secondary findings, which obviously we need to report for any patient. If you understand, these are the 3 different, young adults. When I say young, less than 50 years, who suffered cardiac deaths. It's not just the risk factors that we all know about, the junk food, the stress and etc., but also the genetics, which plays a very important role. The cardiogenomics is something which obviously we have to really, cater to, and obviously that's the need of the hour. For the sake of time, I'm going to skip these cases.
Yeah, I think you need to wrap up, Dr. Vamsi.
Right. So if we could come to the last slide, wherein I'm talking about the holistic approach, it's not just one specialty or super specialty, other patients, general physician. It's everybody put together a holistic approach towards solving patients' problem. So clinical genetic testing basically has facilitated diagnosis, identification of at-risk individuals, as well as application of these genetic testing to prevent transmission in the next generation as well. So from gene panels to exome or genome sequencing, and then back to gene panels. This is the circle of life that we're kind of following especially in the cardiac, in the cardiac field. So having said that, that's my last slide. Thank you so much for your patience with me.
Thank you.
Thank you so much, sir, for your valuable talks. Now we would move to the panel discussion and would like to call all the speakers for the session on stage. Now the house is open to questions. No, sir. She's not joining.
Open for discussion or comments or questions.
Suk, your talk really was very interesting, but it was unfortunately the time when people were coming back from lunch and after AGM. But the most important thing, this whole session talks about diseases where there are multiple genes involved. And as genetic counselors, we know that as of now, for a long time we talked about body type. Anybody who looks at me will say I have diabetes, but I don't. Okay, so the anthropometry has gone out. Now we are coming to polygenic scores, which includes anthropometry, your personal history, and your genes. But how many genes do we take in? When do we say that we are ready to counsel the patient?
Yeah, so that's where the difficulty is when it comes to polygenic risk score. So there, I think the technology has run very fast. So there are many labs that are already selling risk estimates, so risk-based tests using polygenic scores. So if you are in that space, it's very important to see the source of that report. So you can actually ask for raw data. You can actually look at what variants they have included. It would have been pulled out of all of the GWASs that was done. So you just need to make sure that their cohort that was used for their, not validation, for their curation, at least includes the Asians GWASs, which we know that there are not many, which is why a low polygenic risk score in current tests may not necessarily mean that it is actually low risk. It's just that there isn't enough known about the SNPs that are from Asian cohorts. So you haven't found them. So you are testing against variants that have been found in mainly a West Eurasian-based cohort. So it's more of that. So you have to be careful when you're doing these reports, the source of where they're making their, they curate based on which GWAS. So do have a look at that now.
Is Dr. Vamsi available for questions? Dr. Vamshi Krishna?
Yes, ma'am, he's online.
Okay, so I had a question for him. Dr. Vamshi, can you hear us?
Yes, ma'am, I can hear you now.
Okay, so I just wanted your comment on how much of these cardiovascular panel that you were talking about, how much data is there from Indian or South Asians or whatever groups? Because in the past it used to be panels that were licensed from the West and the risk scores were assigned. So have data from Indian CAD, CVD, genomics been incorporated for additional founder mutations or risk analysis?
That's a great question, ma'am. I think we are all working in silos at the moment. I do not see a comprehensive set of data which has come from the really major groups which are working, both public as well as private. And we will require the public-private partnership to actually, get back on a database level wherein we're able to give those risk, stratification to our own Indian population. I think we at Dr. LighTouch Lab, we kind of do somewhere about 20 to 30 these cardiogenomic panels per month. Having said that, we will have to collaborate with other, governmental organizations and private as well to kind of get back to that data.
Hello, ma'am. I want to add here that I come from Vet Genome Labs. So the Vet Genome actually has a test called Cardiogen, where we offer a test that is polygenic risk score based. So it was introduced few years back, but as GCs, because it's not a diagnostic test, it's a direct to consumer testing. So the GC team does not actually, promote it earlier. It's the company's, this. But now, as this, this question of wellness that we are seeing for a few years, it's growing. And there are individuals where the genetic testing for diagnostic variants comes negative, meaning the doctor is telling that there is a history. Then it may not be as a severe history, but there is a history of cardiac events in the family. And it does not fit with a monogenic or a Mendelian factor, but there is some genetic factor, meaning it is not monogenic, it could be a polygenic one. So how do we do it? So there is a study that was done in Harvard based on this PRS, and a similar model was taken by our scientific team, and I forget the number, but in the South Asian population, affected individuals and normal individuals were taken. A PRS risk score was first studied. I'm sorry, I don't have the paper, because if anybody asks us, we first share the paper to the doctor. If they want, then we would say that only contact the lab and do it. So, but it is based on around 5 million, variants. There are lots of genes and there are millions of variants. So put together, they give a risk. And we have counsel patients who have done the test. But again, as Dr. Suki said, low risk does not mean that there is no risk. We have to be very clear about that. And yes. No. It is based upon because the PRS actually differs for each population. So what variants work for the Caucasian population is not going to work for us. So just because the PRS is available somewhere, we can't directly use it, we should not. So for the first a project was taken up, and in collaboration with multiple doctors. I think they have presented in different forums and all. But the test was introduced later into the market, and based upon the study, and those variants are taken, and based upon that they give the risk. So it's called a cardiogen, and for South Asian population it has been studied, and there are millions of variants that are actually used in the study. But still, we
What she said was right. We have, who's here? Gulfiza is here. Yeah, we have got patients who have had a MI below the age of 40. We have taken those, done the whole exome, correct? If you do the polycystic scores, they just don't come under it. So there is something novel which we are missing, and that has to be very population specific. Now with this sequence data, that's the question she asked. Have you incorporated, when you have done clinical, one person says person has a history. You are not getting any of this. Then what are you going to do? Are you removing it, calling it skewed?
Have not been sequenced in an unbiased population or unbiased pool to incorporate additional mutations that may have an impact in our country but are not part of the standard panel, which is how originally the cardio and the whatever was launched by other companies. So there might still be missing ones. There must be missing ones.
I'm more interested to hear from the back of the room. Any questions from you guys? We can keep talking hours and hours. Questions from the back of the room, I'm very interested to hear.
Yeah.
Sir, I have question for diabetic peripheral neuropathy. The variants which you have found in your presentation, is it associated with the onset or is it associated with the severity?
Actually, there's a spectrum of variants. There are variants that are associated with the progression of the disease, and also there are variants that associate with the initiation of the condition, as well as there are variants who are protective
against the diabetic peripheral neuropathy, and as well as there are variants which are shown that the drug response, whether it is responded well or not. So considering all this, we need to counsel the patient.
Okay, sir. Thank you.
Just for information, because you are doing studies on diabetic neuropathy, can you throw some more light? Of course, we are all genomic, but let's not forget metabolomics also. And in the neuropathy and all, I think we know all of us doctors, when the neuropathy starts in the diabetes, they are chronic. Some may start in 3 years, 4 years, 5 years. Maybe that is individual susceptibility. But I believe there are metabolomic studies which are coming, which before clinically establishing neuropathy, you can detect that. This is something like a prevention, or say maybe similar to newborn screening, I don't know. Prevention before the symptoms come. So is there anything, something like that in your diabetic?
Actually, so that's why I mentioned that pre-diabetic population, where that you can screen the pre-diabetic population, where you can detect whether this population is more prone or not. So then it's going for the preventive aspect, so preventive medicine.
You know how you predict then, based on?
So by looking at the different variants, because there are
variants only, genomic.
Yeah, and also it's you have to correlate with the pedigrees, the family history, how they have progressed and everything. The clinical progression of the disease with the family also you need to consider.
Because myoinositol is again an important biomarker which is coming in detection of diabetic neuropathy. And being a metabolomic person, I thought I would ask you whether you have, so that the people can just do that simple test metabolic rather than genomic.
Yes, definitely.
Last one.
Thank you for accommodating this question. I have a question for the cardiogenomics experts. Online, Dr. Ramshi, can you hear me? Hello?
Yes, I can hear you.
Yeah. So this question is regarding cardiogenomics. Cardiogenomic testing, it's sort of a gray area still. We do not have clear-cut guidelines like in cancers. We're still talking about testing genes related to cardiology. Like we do not have clear-cut recommendations for these genes. What would be your comment on that, and where are we heading? Will we have clear guidelines if we screen set of cardiac gene panels? And followed by, if LPA-related cholesterol, how is it treated, and would diet play a role? So these are my questions.
That's a great question. As I was mentioning earlier, the monogenic disorders account for about 5% of these, which are causing the cardiac event. Most of the 95% are something which is, they're not just one sort of protein forming or monogenic sort of conditions that we see. There's lots of epigenetics also, which plays a role. Having said that, it's not easy to delineate or rather associate having just one causative factor, just the gene. Pathogenic or likely pathogenic, which is described in the literature, or maybe something which strong immune population has been submitted to the NCBI. It's not that very easy. We have to obviously characterize these variants not only just on the population basis, but also on the familial segregation basis as well. Only then we will be able to sort of formulate onto the next level. So if you understand the diagnosis, like for example, as we just mentioned, the somatic level or on the disorders level, if you're able to have a definitive diagnosis, only then can you move towards precision therapy or a targeted therapy. So to be able to go towards the precision therapy, we would have to rule out all the other factors which are confounding towards monogenic or a specific gene-related mutation, which is pathogenic or likely pathogenic that we're looking at in that particular program, who's presenting a symptom or whose family has presented that symptom, and I'm sure he or she is predisposed to be causing that particular cardiovascular disorder.
Thank you so much for this engaging panel discussion. Thank you so much for the engaging panel discussion. Now we'd like to invite Dr. Arijit to felicitate Ms. Yeon Sung and Dr. Jugnu to please felicitate Dr. Lahiru. I would like to call Ms. Reena Tripati ma'am on stage and Dr. Vijayalakshmi Kodate ma'am on stage to felicitate the chairpersons, Dr. Arijit and Dr. Jugnu. I would request the panelists to please stay back on the stage for a group picture. A volunteer. ठीक है. The participants are requested to please move for a quick tea break of 10 minutes. Thank you.
Sorry, a quick announcement. This is a book on genetic counseling, clinical and laboratory approach, edited by our own Dr. Usha Dave. It is there available at the registration counter if any of you is interested to buy this. Thank you so much. Check. So this book is available at a discounted price on the line, especially for the students and young professionals. So feel free to check it out. Thank you.
That is how it goes. So pretty
Good afternoon, everybody. Welcome to session 4, Cancer, Somatic and Germline for Treatment and Prevention. My name is Shreya.
Yes. Yes, I'm ready.
My name is Shreya Satish and I will be co-moderating this session with Ms. Leisha James. Cancer, a complex group of diseases characterized by abnormal cell growth, presents distinct challenges in both treatment and prevention strategies. Understanding the differences between somatic mutations, which occur in non-inherited cells, and germline mutations, which are inherited and can predispose individuals to cancer, is crucial. This knowledge informs targeted therapies for somatic mutation and personalized prevention plans.
I can't hear you. Can you hear me?
For those at risk due to germline mutations. By leveraging these insights, medical researchers and practitioners aim to enhance treatment efficacy and reduce cancer incidence through tailored approaches.
We are honored to have a distinguished panel of speakers and chairperson and co-chairperson today for this session. Further ado, let me introduce our chairperson. It's my pleasure to introduce our chairperson, Dr. Parni Madasamy. He is a professor of genetic engineering in SRM University, and he has several career highlights with awards and honors, DBT Cutting Edge Research Enhancement and Scientific Training Award, and 12 awarded and 4 currently registered research guidance, and patents under plants expressing environmental stress tolerances having petunia,
and several publications, and there are numerous research grants received. I welcome Dr. Parni Matsami. Our second chairperson needs no introduction. She delivered an excellent talk in the previous session. Ms. Suki, I request you to please come onto the stage and co-share the session with us.
Thank you very much. Our first speaker is Professor Dr. Shirley Hodson. Prof. Shirley, are you able to hear us now? Let me just introduce her first. Professor Shirley is Emeritus Professor of Cancer Genetics at St. George's University of London and is a part-time consultant in cancer genetics in Leicester. She's got a wonderful set of qualifications, doing research in ICRF, now CRU UK, developing regional cancer genetic services from GUIDES, and later in St. George's Hospitals in London. She's also done work on Duchenne muscular dystrophy with Professor Dubowitz at Hammersmith Hospital, earning her DM. So she's had a very illustrious career highlight, but I think her interest in genetic counseling and her support for BGCI is it's wonderful. It's so lovely to have you back with us, Prof. Shirley. And without much further ado, I'd like to invite Professor Shirley to give her talk. Not very clearly at the moment. I think we're trying to sort out some of the technicalities. Just give us a second. Professor Shirley, can you try that again, please?
Can you put on the slide for my talk?
Yes, now we can hear you well, and we're just going to put on your slides right now.
Oh, thank you. So is that all right now?
Yes, we can hear you very well now, and you're able to hear us. Yep. Okay, so we're just putting on the slides. Just give us a moment.
Right. Okay, I'm sorry, this is the one that's got my face in it as well, which is rather depressing, but anyway, it's a great delight and a pleasure for me to be able to come and join you and to open this particular session, because it's what I'm mainly working on now, and it's very exciting, the changes that are happening with all the genomic information that is increasing our ability to understand cancer genetics and the clinical relevance. So, should I say, what is it? Sorry? The clinical relevance is that we are much better able to identify individuals at increased risk of cancer, screening and management, and also for testing their family. And of course, genetic counselors are absolute key in arranging all of this. And personalized therapy is another avenue where it's helped. All right, can you hear me still? Can you hear me? Yeah. So
Yes, we can.
So there are two main ways in which genomics is helping. One for the cancer. One is testing the tumor sample, which gives us many ideas, and I'll show you in this slide in a moment. Many ideas about causes of the cancer, information to help diagnose the particular cancer type, and may provide clues to the genetic susceptibility of that person to the tumor. And it may also provide information for personalized treatment, which I'll mention. But the other thing that we're doing with genomic testing that involves cancer genetics. So, sorry, you're going too fast for me. Can you go back, please, one slide? Can you go back one slide? Oh, that's better. Yeah, the other way that we're using this information is in screening, and I'm going to talk about that a little bit later on. But just to talk about genomic testing, as I say, it may help us identify why the tumor started to develop. And I'll show you in the next slide this is a tumor, a cancer genome, and you can see that you can learn an enormous amount. You may have had these pictures in previous talk, but it can show you whether there have been mutations or translocations and indels, loss of heterozygosity, gain of heterozygosity, and all sorts of things. You can learn about the tumor. You can also learn about how many mutations have developed in the tumor, because some cancers have enormous numbers of mutations, whereas others don't. But we now know the sorts of signature of mutations that you find for different cancer types. And this is a slide which shows, whoops, sorry, one back again. Back one, there you go. This is a slide which shows you the types of genomic changes that you see. For instance, if you do my blood, you'll probably find a lot of C to T changes because I'm very old, which would be characteristic of older people. Whereas if I'd been smoking a lot, you would find C to A changes, as you see in the environmental exposure section. And also near the bottom, you'll see that you can get very specific mutational patterns if people have got defects of DNA double-strand break repair, as you do in BRCA1 and BRCA2 mutation carriers. And the last one, you can see you may get defective DNA mismatch repair patterns, as in Lynch syndrome, with C-to-T transversions as well. So the types of causes this is a very complicated slide, but basically it shows you that each tumor type, as you see in the line at the top there, will have a very specific tumor signature, which you can use to identify the type of tumor. For instance, if you go down from breast cancer, there are quite a number of different signatures are associated with breast cancer. Whereas with bladder cancer, it's a lot fewer. And if you then were to look at different types of breast cancer, you'll find they have very different signatures, and this can help to show what specific type of breast cancer you're dealing with if you're uncertain. On the next slide, please. So this is a signature which was found in a colon tumor, which showed many variants in domain 1, 83 domain 1 variants, which were found in this particular tumor, and the signature was compatible with mismatch repair, as in Lynch syndrome, with a high tumor mutational burden. And we know that in Lynch syndrome there are lots of neoantigens being formed, and this will give a high tumor mutational burden. And it was also found that there was an MSH6 mutation in the tumor, and you can then go on to check the patient and find, in this case, they found an MSH6 mutation, which therefore meant that this individual had the tumor, was susceptible to this tumor because they had Lynch syndrome. Next slide, please. And so just to diverge a little bit with Lynch syndrome, it's very interesting because we know that there are many treatment advances in relation to germline mutations. For instance, cancers with antibodies to express oncogenes, and the genomic mutations in the tumor that we can find can identify particular antigens that then treat that tumor with, like Herceptin, for instance, BRAF and MET mutations in melanoma, tyrosine kinases that are mutated in lung cancer and that kind of thing, where you can then go on to specific targeted therapy. And if you know that somebody's got a BRCA1 or BRCA2 mutation, then of course you can offer them PARP inhibitors for the treatment for their tumours. And immunotherapy to be offered as well. And just vaccines is an interesting view. Then if you go to the next slide. With Lynch syndrome, we know that the mismatch repair, next slide, sorry, mismatch repair syndrome in Lynch syndrome enables germline mutations cause microsatellite instability in mutations in genes with short repetitive sequences like the growth promoter mutations. And what you have in two outcomes of this, one is that the mutations which are formed by this DNA repair defect are growth promoting in themselves. So tumors in people with Lynch syndrome should grow really, really fast. But also, we know that people even with Lynch syndrome, maybe even before they develop any cancer, they have these neoantigens, mutations with these short, small d, which are antigenic. And so you can then use that potentially for a vaccine, which is being thought about, that you might be able to give a vaccine to people with Lynch syndrome aimed at these neoantigens which are formed in their body to failure. Yeah. Next slide, please. Next one. Thank you. So I think, well, sorry, I just sort of mentioned all this. So next one along, please. Next slide. Yeah, I should press a button. So, yeah. So, germline genomic testing is another aspect of genomic testing in cancer genetics. And what we're now doing is finding screening people who may or may not have cancer. But people who are being screened by genomic screening may be found to have unexpected variants in cancer predisposing genes. And that is a big worry because we don't know whether those mutations in these unexpected variants are in fact high penetrance and likely to cause cancer, or perhaps are low penetrance. And so there are problems in determining the penetrance of variants diagnosed by screening, and they're obviously dependent on the mutation type, but also the familial context. So next slide, please. Most current estimates of mutation penetrance are based on families with multiple cases of the same or related kind of cancer. For instance, we know that BRCA1 and BRCA2 were identified by linkage analysis in the first instance because there were many affected individuals in the family. So they were high penetrance. But population screening detects mutations in non-familial cases, which is quite different. And what we're finding is that if you look at this is a good one. There have been some studies recently, and this was looking at biobank, in a UK biobank, where all these individuals had some genomic screening, and some of them were found to have mutations in BRCA1 or BRCA2 or Lynch syndrome genes. And interestingly, the ones who have a family history of breast cancer, the penetrance of those mutations was something like 45%. And 4% of BRCA2 up to the age of 60. But if they didn't have family history, the women with the same mutation had about half the chance of developing cancer as in the ones with a family history, and that's with even undetermined amount of family history. And there was the same sort of finding with people with colon cancer, in people who were found to have MSH1 and MSH2 Lynch syndrome genes, had mutations in those. Their risk of colon cancer by the age of 60 was about 40%. Whereas if they had a family history, but if they did not have a family history, again the risk of colon cancer up to the age of 60 was only about half that. So it's fascinating that there must be other factors within these families that make that dependent these genes. And so it's difficult for us when we find these mutations in the unselected population to know what to do about screening in people. And you may have heard about or thought about people, for instance, who've had CDH1 mutations identified in the screening area. And you don't know, if you find a CDH1 mutation in somebody who's got a family history of gastric cancer, because it predisposes strongly to gastric cancer, you might well offer them a gastrectomy, which of course is a huge imposition on their lives. And, oh gosh, right. And one wouldn't do that unless one knew that they had a very high risk of cancer. So, very important what genetic counsellors do in this situation. Well, obviously, the first thing is up to date all this research, which is quite a big ask, because there's a lot going on, as you know, in finding genes being identified and so on, and the importance of it. And so, of course, the genetic counsellor is to assess the genetic situation for the patient. What's their risk? Do we know the diagnosis for them, and what do we do about it? The counsellor must also understand the importance of the tumour genome information. What does it tell you about the patient? Can they be offered personalised treatment? And those things, which again are very important. And if you find mutations in the screening setting, what do you do about screening and treatment? So I think one of the take-home messages from all this, to my mind, are that it's very important not only for the genetic counsellor to be absolutely up to speed with all these things, but also to participate in the linked up meetings, MDTs as we call them, a multidisciplinary team meeting, because they need to liaise with the oncologists, surgeons, the pathologists, to try and get the best ideas of the importance of what they're finding. And sometimes the pathologist, for instance, or the oncologist may even find the mutation first. And so linking in with these other professionals in these different disciplines is one of the very, very important roles of genetic counselling. So I would like to thank you very much for listening, and sorry for the bumpy ride, but very good to talk to you, and I do hope that the rest of this part of the meeting goes well. Thank you.
Thank you very much, Prof. Shirley. If you like to stay on, there will be a panel discussion at the end of this session.
Yeah.
Next, we have Professor Dr. Managari Chetty, and she'll be joining us online as well. And let me introduce Professor Managari. She's the head of the Department of Craniofacial Biology. And she's the full professor in the Faculty of Dentistry and director of the African Society of Dental and Craniofacial Genetics. She's highly recognized and respected as a dental genetics and non-communicable disease area, particularly in rare diseases manifesting in the head and neck area. She promotes the development of dentist scientists within South Africa and Africa. And I think she's joining us from Africa. Right, okay. So let's have Professor Dr. Managari's talk.
Now it's only that there is fun. So, there are episode and word
In our countries, we also have limited healthcare resources for the management of these patients. And oral cancer also contributes largely to the socio-economic burden of Africa within Africa. Several studies have also reported that there is a 50% increase in the use of tobacco in Africa and other lower middle income countries, which certainly doesn't help the incidence of oral cancer. Looking at the global map, the world map, with an age standardized rate per 100,000 in terms of the incidence of oral cancer, it shows that the lower and middle income countries are vastly affected. Although if you look at Africa, there seems to be some disconnect almost And that is basically because of the underreporting of cases, and we don't have any or functioning cancer registries. Additionally, if we move on to now the global map showing the mortality rates, here there seems to be a more sort of accurate prediction in terms of mortality rate in our lower and social middle income countries, and Africa seems to be a little bit more accurately represented. Coming back to the focus of the talk with regards to liquid biopsy, if one has to describe liquid biopsy, it is a non-invasive diagnostic approach that involves the analysis of various biomolecules. Some of these molecules are circulating tumor DNA, for example, circulating tumor cells and exosomes. These biomarkers can be isolated and identified in various fluids such as blood, saliva, and other bodily fluids. You'll notice that I've actually put exosomes in red, and that is because most of our work that we do here at our faculty is centered around exosomes, and I will elaborate a little bit more on exosomes. Exosomes are described as extracellular vesicles, and they consist mainly of three subclasses. One of them being endosome origin exosomes, plasma membrane derived microvesicles, which we commonly refer to as ectosomes, and apoptotic bodies. Exosomes, as you can see in this diagram, are secreted by both normal and tumor cells. And that is why they can serve as an efficient means for transferring intracellular content. They also are in a position to enable cell-to-cell communication, either within a tumor microenvironment or at a distant site. I've just elaborated a little bit about exosome biogenesis. I'm not going to read through the slide or go through it, but I've put down a very simplified diagram which enables one to sort of understand more clearly how exosomes develop. I will share the PowerPoint with the organizers, and if they're okay with you receiving the PowerPoint, I have no problem with that. Briefly describing exosomes, they are microvesicles. They range in diameter between 30 to 150 nanometers, and like I said before, secreted by both normal and cancer. You can find them in several body fluids, like I said, blood, saliva, and at this point in time, in our lab, we're actually looking at isolating exosomes from saliva, which is less invasive than blood. And if you really think about it, you know, most patients are amenable to giving you a saliva sample as opposed to a blood sample. So during cancer development, you get the cells that secrete more exosomes. And these exosomes, particular the ones secreted by the cancer cells, tend to have significant changes in their size, in their composition, and so on. And these particular exosomes secreted by the cancer cells facilitate the communication within this tumor microenvironment. But more especially and more sort of ominously, they are the ones that are involved in metastases to distant organs. So isolating my exosomes is one thing, but the most important step of these exosomes is the identification and the characterization of them. There are several products on the market that enable you to actually isolate the exosomes, and you subsequently then have to store them, which we do in a -80 freezer. But like I said, the most important step with regards to exosomes, it's characterizing them. Because it's these characteristics that actually tell you which molecule is present, and you can then target the molecule or not with regards to your treatment approaches. With regards to characterization, we have two basic methods. One, you can look at the external characteristics, and that's mainly the morphology and the size of the particle. And secondly, you can look at what is included in the substance of the exosome. That is, for example, DNA, RNA, protein, lipids, and so on. In general research, the identification of these isolated exosomes, we can use three methods. The two that we use are the TEM, transmission electron microscope, where we look at the morphology of the exosomes, and we look at the nanoparticle tracking analysis, NTA, where we use a nanosite machine. Western blot is also another way of identifying the exosome surface, or looking at the protein markers that are found on the surface of the exosomes. Now, each method of identifying these exosomes are tabulated here, and the advantages and the disadvantages are also listed. So given the common advantages and disadvantages, you can now make the decision on which type of method you would like to use to look at particular aspects of the exosome. These are some TEM images of exosomes from our lab, and as you can see, they range in size. The normal exosomes tend to be smaller and more regular, whereas you can then get the tumor exosomes or malignant ones from malignant cells, which have a different characteristic altogether. This is a still life image of an exosome using the NTA process. If you had to look at it sort of visually using the machine, you will find that these particles tend to float around. I've put down a table here by these authors. This is, to the best of my knowledge, like I said, the present state of exosome research as biomarkers in Africa and other low and middle income countries. A review was done last year by a colleague and myself, and we found, just looking on the continent of Africa, there were no studies available in sub-Saharan Africa. Now, what is important here is that we know that our genetic diversity is enormous in Africa. So we can't take first world studies and translate them into ours, especially those studies involving genetics and genomics, for example. At our faculty, we still have ongoing research where we're looking at the role of exosomes as a diagnostic biomarker for oral squamous cell carcinoma. We've just registered a multinational sub-Saharan African study involving about, I think, 6 sub-Saharan African countries. Now, this slide is a little bit busy, but what I wanted to do is put down as much information as possible for what possibilities could arise should we be successful in identifying these exosomal biomarkers in our country for our populations. Firstly, it could enhance early diagnosis of oral cancer. The biomarker identification option via a liquid biopsy could serve as an alternative option in areas where we cannot easily get a histopathological diagnosis, for example. We could eventually begin to use exosomal biomarkers to determine the prognosis. At this point in our faculty, we have just started the process of looking at cell-free DNA in liquid biopsies. So I don't have much to report here, except to say that when tumors form, they release DNA into the circulation. And we have got to find ways of identifying the cell-free DNA which is floating around in our bloodstream, or maybe in the case of oral cancer, in our saliva. And the features of this cell-free DNA can be considered as important diagnostic and prognostic parameters for. Once again, I've just put down a whole lot of information, but I felt I needed to sort of provide as much information as possible for the advantages that could happen if we have worked out successfully the identification of cell-free DNA as a biomarker for head and neck or oral squamous cell carcinoma in Africa and other lower middle income countries. Another aspect that we are just beginning work in is circulating tumor cells. And circulating tumor cells are cancer cells that circulate within the bloodstream after being naturally shed from the original tumor. And we all know that, unfortunately, these are the source or the facilitators of. This is a review that a colleague and I did last year, where we looked at what was happening in Africa at the time. And much of what we found during the process of doing that review actually has informed our projects at this point in time. The conclusions that we saw were quite interesting in that within Africa, I've just taken the opportunity to list some of the research foci of our department, and I'm hoping that at some point we might be able to develop, collaborative projects and research relationships across our continents. I've already listed the focus of our research, but some of the factors which have contributed to the development of our projects in South Africa, for example, we had a multicultural. So given all of our thoughts and investigations with regards to liquid biopsies, trying to develop new programs to address the needs that we face in Africa and in other low income countries, some of the recommendations we have listed here is the training of a dentist scientist, for example, equip dentists to have PhDs, which then will increase their research or their focus in their academic career towards research. We recommend the need for more collaboration among dental researchers across continents. The development of cancer registries. There are cancer registries, but I do not believe that they're optimally managed or do not function in the way that they need to. Biobanks are just now developing across the continent, and clearly these are all very necessary to optimally be able to function.
I think this is actually quite a nice place to end, and we thank Professor Managari for her thoughts. Shall now move on to our next speaker.
I would like to introduce the next speaker, Dr. Harsheth, PhD. He is affiliated with the Translational Genetics, Newcastle University, UK, Assistant Professor and Head, Advanced Genomic Technologies Division, Fridge Institute for Human Genetics, Ahmedabad. He is also a honorary clinical geneticist in
Jivraj Mehta Hospital, Ahmedabad, and Civil Hospital, Asarwa, Ahmedabad. He is also a founder and CSO of Decipher DNA. He has additional qualifications like a panel member of South Africa's Open Genome Project, ICMR Subcommittee on Precision Oncology, and Indian Society of Medical and Pediatric Oncologists Rectal Cancer Guidelines. He has 39 odd peer-reviewed publications in international journals, 3 book chapters on cancer prevention, ring chromosomes, lysosomal storage disorders, diagnosis and management. He also holds a patent, US patent, and Indian patents. He's a visiting lecturer at Gujarat Technological Universities, and I welcome Hasmeta to present his talk.
Is that? Yeah. Thank you so much, sir, for the kind introduction, and good evening, everyone. Being the penultimate lecturer, I have the honor of going through this rather quickly. And on the outset, it's been a couple of hours since we had our food, so it's a good time to have a bit of an introspection as to when the food is going through our GI tract, what is happening, and the syndromes that are associated with it. So, to begin with some conflict of interest, well, as sir mentioned, I'm a named inventor on a patent describing microsatellite instability assay for colorectal cancer. I've co-led data analysis and interpretation of the CAP2 trial, something that I will be showing to you today, and I think Suki has already seen it a few times. I'm also India's lead submitter to the prospective Lynch syndrome database. Again, that is something I'll be talking to you about today. And I'm panel member on ICMR's Task Force for Precision Oncology and South Africa's Open Genome Project. Now, I have had the pleasure of doing all sorts of things, all the way from pharmacogenetics of orphan to rapid molecular diagnostics, working on male infertility. Of course, Dr. Deepak ji is one of the collaborators on this work, aspirin and colorectal cancer prevention, lysosomal storage disorder, diagnostic development, and so on and so forth. But one thing that has stayed with me is prevention of cancer. And the reason why that is from this one line from 1618 by Thomas Adams, that he is a better physician that keeps diseases off us than he that cures them being on us. Prevention is so much better than healing because it saves the labor of being sick in the first place. And that's exceptionally important when you are in genetics. And I begin my story with Angelina Jolie. Way back in 2013, she came to stardom, not because of her movies, but because she was diagnosed to be BRCA1 carrier and went for double mastectomy and oophorectomy, something rather radical. And after she did that, in the NHS in the UK, the rates of referral for BRCA testing doubled. And it just still can't keep up with that anymore. And essentially what she managed to demonstrate was that identification of at-risk individuals provide you an opportunity to reduce risk of cancer down the line. And before we do that, let's go back to class 6, Knudsen two-hit hypothesis. Essentially, we have 2 copies of DNA, right? So if one copy is knocked down, you just require one more copy to be knocked down later down the life, and therefore you are well down the carcinogenesis pathway. Therefore, majority of the hereditary cancer syndromes already have one copy hit, and you have an earlier age of onset. Now, since I'm not in Gujarat and this is not a dry state, I can give this example. This is one of the pubs right outside where I used to work in the UK. And these were 2 bouncers out there. So if one of them went on a loo break, there was still another guy keeping things in check. But if you just had one bouncer, now we had a problem and there could be a brawl. And there used to be from time to time, especially if Newcastle got knocked out in football. So on the left-hand side, you're talking about a condition where there's low lifetime risk of cancer, something that happens in the population. On the right-hand side, that's a scenario equivalent to hereditary cancer syndrome, high lifetime risk of cancer. And I give an example out here of Lynch syndrome. And this is how essentially your normal gut looks like. That's the one on the left. And as you have a little adenoma popping up, it then turns into carcinoma and it goes all the way into full-blown metastasis. And generally, there are 2 approaches as to how this can form. The one on the top is known as FAP, or familial adenomatous polyposis. Essentially, your DNA gets jumbled really badly and you're well down the pathway. That occurs in about 5 out of 6 patients walking into your clinic. The one at the bottom is known as mismatch repair deficiency. So this is where your mismatch repair mechanism is knocked out, and because of that, you start forming small errors in your DNA all over the place, what is known as microsatellite instability, and you end up down this pathway again. This is about 1 every 6 patients that would walk into your clinic with colorectal cancer. And it's the bottom pathway where you would find Lynch syndrome primarily. This is one of the first pedigrees where Lynch syndrome was diagnosed way back in Newcastle in 1993. In fact, this is the 30th year when the very first gene for Lynch syndrome was identified. In fact, the very first one was MSH2. It was in fact not identified in a human, but in in a bacteria, in fact, later on. And since then, a lot of work has come through in Lynch syndrome. Primarily, it's not one, but there are 4 key genes involved: MSH2, MLH1, MSH6, and PMS2. In fact, now there's a 5th one called EpCAM. And these are the patients who are at risk of not just one, but multiple different types of cancer during their lifetime. Colorectal cancer being, of course, one of them. You have endometrial cancer, and a whole host of upper GI and genitourinary cancers. In fact, WHO estimates that the worldwide incidence, population incidence, is 1 in 125. In fact, it's slightly exaggerated. If you take into account the low penetrance of PMS2, it comes to 1 in 300. It means in India, considering the population size, there are 5 million people who are Lynch syndrome carriers. That makes it way more common than HBOC. It's the lowest hanging fruit there ever is. And as they say, not all fingers in your hand are equal. Similarly, not all genes behave equally. Each gene confers slightly different lifetime risk of cancer. But in the one, which is the most common one, MLH1 gene, you can have sometimes lifetime risk of cancer going all the way up to 80%. So that's pretty substantial, come to think about it. Now, when it comes to diagnosis, there are 4 types of modalities that are available. Of course, you have your good old standard immunohistochemistry. You have PCR fragment length analysis that is also routinely available. Of course, it's the world of NGS. In fact, I was one of the co-inventors of the assay, and this is an example of that. It's right here. And of course, nowadays you have got TMB, or tumor mutation burden. However, I'm going to put my neck out, and I'm just digressing slightly out here. This is the CAP guideline when it comes to prescribing immunotherapy and how you can do that. So if it's colorectal cancer, you can have results from immunohistochemistry or fragment length analysis or NGS, and that's fine. You can still go ahead and give immunotherapy. But if it's endometrial cancer, your immunohistochemistry is the gold standard, not PCR by fragment length analysis. And when it comes to non-colorectal cancers, there is no gold standard even today. And this is data from 2022. And we are still talking about, and also one more thing, TMB is one test that cannot be used to give immunotherapy. Now, once you've identified Lynch syndrome patients, what the hell you can do? Well, you can start to think about the lifetime risk of developing cancer, right? And like I said, not all genes are equal. Here is an example. I'm sorry, it's slightly blurred out here, but on the top panel, you've got colon cancer. On the bottom column, you've got rectal cancer. In the colon cancer, you have the highest risk being conferred by MLH1, followed by MSH2, and the lowest is PMS2. But look into rectal cancer. Oh dear, what is this? I'm sorry, somebody. Okay, well, I'm sorry, this is not working. Can you just click next, please? This is one of the data from the prospective Lynch syndrome database that I'm a part of. And in fact, we managed to show that if you have Lynch syndrome, your risk of rectal cancer is actually pretty low. You generally find cancer in the right-hand side of your colon. Next slide, please. But here is the interesting part. Regardless of which gene is affected and you have had cancer, your risk of mortality is absolutely equivalent. There is no difference whatsoever. And in fact, your probability of survival is pretty high. Next slide, please. However, please go back. However, if you are thinking about mortality, half of the patients who die of Lynch syndrome are just colorectal. The other half are non-colorectal Lynch syndrome cancers. So you're talking about esophagus, stomach, liver, pancreas. And if you have MSH2 gene carriers, these upper GI cancers are the ones that are most prevalent in them. And that is where you find the highest risk of mortality. Next slide, please. Now, sorry. Oh, it works. Wonderful. Thank you. So, now that you have identified a patient with Lynch syndrome and you know what are the possible risks, well, how the hell can you prevent the cancer from coming in the first place? Because that's why we diagnose them. Well, this is a trial that started in 1990s when I was a wee boy. And it was a 2x2 factorial trial done in 1,000 patients who were Lynch syndrome, proven Lynch syndrome carriers, never had cancer before. Half of them were given aspirin and aspirin placebo. Half of them were given resistant starch and resistant starch placebo. Essentially, it's a 2x2 factorial trial, so you're doing 2 trials for the cost of 1. And since I'm a Gujarati, that's a wonderful thing to do. And these patients were given aspirin or resistant starch for 2 years. Then they were invited to take that intervention for further 2 years. And then they were followed up for up to 20 years of their life. And in 2020, we published this study in Lancet. This is Professor Sir John Byrne. He's the PI of the study. And this is me on my graduation ceremony. He's my teacher. And at 20 years of follow-up, we showed that people who are given 600 milligrams per day aspirin for more than 2 years, the risk of colorectal cancer was reduced by half. Not only that, if there are some number buffs here, the top slide is showing intention to treat analysis. And at 5 years, you can see a clear separation of risk. So blue are the people in aspirin, are placebo, the red is aspirin. And you can see that there is a beautiful separation and it's still keeping on going. Remember, these patients had only taken aspirin for more than 2 years. They've not taken anything since. And these lines are going apart. When you see per protocol analysis, it's even more astounding. And therefore, when we did some calculations, it turns out that the number needed to treat was just 24. Put it in simple words, you give 24 people aspirin, you save one of them with cancer. That's a substantial number. That's even better than colonoscopy. So because of that number, in 2020, the NICE in UK published guidelines saying that people who are Lynch syndrome carriers can be given low-dose aspirin to reduce the risk of cancer. Not only that, earlier this year in India for the first time, the guideline came out saying exactly that. Not only that, at what interval colonoscopy should be given was also incorporated in the guidelines. So now there are published guidelines to tell you what exactly to do and when. But 600 milligram dose is ridiculous. It's too high by modern standards. So there is a dose non-inferiority trial called CAP3. It's ongoing, but the data will come out next year. So we are all waiting with bated breath till then. However, again, not everyone is equal. This is data showing people who are either normal weight, overweight, or obese. And it's obese people who have additional risk of cancer on top of what they already have because of the genetic defect. So, what does aspirin do in those? Well, it ameliorates that excess risk. And that's substantial. So if you're thinking about giving your patients aspirin, or counseling them about aspirin, well, remember that low-dose aspirin doesn't work in reducing cardiovascular events in big people. In old people, if you are prescribing aspirin, they're at higher risk of GI bleeds, and that risk increases exponentially with age. In the first few years when you start aspirin, actually the risk of cancer rises slightly. And this is particularly true for old and obese. The working hypothesis for this is that big people need more than 100 milligrams to achieve the desired effect. And aspirin, whilst it's helping to destroy early neoplasms and reduce metastasis, but what it could be doing is it's allowing those advanced lesions to pop out. That is why early on you get these slightly higher risk, but later on they tend to dip down. And if you are thinking about taking aspirin, it's better to give to people who are young, not old. Now, that's just one part. The second part was resistant starch. So each of these people were put onto 30 grams of resistant starch. That's equivalent to 2 bananas, slightly unripe bananas a day. And when we looked into data, it turns out that people who had taken more than resistant starch for more than 2 years, their risk of upper GI cancer was reduced by half at 20 years. Not only that, for the first time on a journal cover, we managed to put bananas. Again, for the number buffs, the upper graph is intention to treat, the bottom graph is per protocol analysis. And just like aspirin, you're seeing these beautiful separations happening at 5-year intervals. And we were able to show that if you are at risk of cancer, if you are a Lynch syndrome carrier and you're taking resistant starch, your risk of upper GI cancer is reduced by half. Now, all of this work, of course, has to get into public domain. So since I'm a panel member of ICMR subcommittee on precision oncology, guidelines for this are being written up. I'm also India's only submitter for prospective Lynch syndrome database. So essentially every Lynch syndrome patient that's diagnosed at my center, they're prospectively followed up every year for every little detail. That is, have they gone for colonoscopy? Are they taking aspirin? Have they been diagnosed with cancer? Have they been diagnosed with adenoma in their colonoscopy? So on and so forth. And through all of that work, this is a collection of 8,500 patients that have been recruited globally, and we are the only center in India that are participating in it at the moment. And this, for the first time, including Indian data, the brand new database has come out. And by the way, this is accessible to all genetic counselors for free. Essentially, all you need to do is put down which organ for which you're interested in estimating risk, which gene is affected, what's the age of the patient, and it gives you profile of their lifetime risk, which is substantial. It helps you in providing accurate counseling in these patients. Not only that, remember, all of this work, majority of the cases are colorectal. We're missing out on non-colorectal cancers. So we received a substantial funding from the Gujarat government for the first time to study the prevalence of Lynch syndrome in non-colorectal cancers. Primarily, we are looking for esophagus, stomach, liver, pancreas, small intestine, and endometrium. And not only that, if they are diagnosed with Lynch, we are going to be testing all the first-degree relatives, and we'll be employing food frequency questionnaire to try and understand why did some of them got cancer and why some didn't. Is there some dietary modification which is also leading to something like this? And by the way, this study is open to recruitment. All the test that is done in this is done free of cost. So if you do come across a patient who cannot afford any of these tests, please do let me know. So, conclusion, it takes 2 to tango. Remember, you don't require just one, you require both alleles to be knocked out to begin the carcinogenesis pathway. There are multiple modalities to help you detect Lynch syndrome carrier, but you need to be careful of which cancer you're dealing with out here. When it comes to personalized risk and management, now there are wonderful sets of guidelines that helps you when to do colonoscopy, when to start, what interval you should be doing, when to do hysteroscopy, and so on and so forth. We are even coming up with surgical outcomes analysis later on, and you will be bowled over by the fact that whether you should do total colectomy or just partial colectomy, and what are the outcomes of those for metachronous cancer down the line. When it comes to chemo prevention, of course, there is aspirin, costs 10 rupees a month, absolute peanuts. But if that doesn't float your boat, go bananas. When it comes to guidelines, not only just UK, but for the first time in India, guidelines are recommending aspirin as a chemopreventive agent in Lynch syndrome carriers. When it comes to cancer risk prediction, of course, we have the wonderful PLSD database, and it's free for use for anyone for how many times you like to do. And when it comes to future direction, of course, colon and endometrium has been analyzed to the nth degree. It's time to go beyond that and look at non-colorectal or upper GI cancers. And with that, all these wonderful people from across the world who have been part of this team, our wonderful funders, GSBTM, Cancer Research UK, MRC, and all my teachers and mentors and team members. Couldn't do without them, and thank you so much.
Thank you, Dr. Smith.
I will take up the last speaker for this session, Dr. Jugnu Jain. She is a CEO of Sapiens Biosciences. She's a PhD in molecular genetics from Cambridge University, UK, on Nehru Trust Fellowship. She's a postdoc from Dana-Farber Cancer Institute, Harvard Medical School, Boston, USA. And she has 15 years of experience in drug discovery from Vertex Pharmaceuticals, USA. She has handled a lot of drug discovery projects. She forged collaborations with academia, industry, and NGS for advanced drug candidates in clinical trials. She returned to India in 2011, and she is contributing to the developments in India. She has published several papers on Indian works, and now I welcome Dr. Jain to present her talk.
So thank you for your patience. The penultimate speaker has spoken and now I am the last speaker. So I am between you and your freedom, and I do promise not to stretch beyond 20 minutes, maybe earlier. So I run a biobank, India's first commercial biobank. I won't speak much about that because this is a genetic conference. I'll talk more about some of the genetic work we have done and some of the insights we have in that. So, we are partnered with Apollo Hospitals, and just like Apollo is pan-India, so are we. We have people present in all these locations and samples from all these locations, so we have no particular bias on our samples. Our headquarters are in Hyderabad, but we have people in many other sites. The number of patients from whom we have samples available are more than 73 hospitals, which are the Apollo hospitals. And we have multiple formats of samples. So a large percentage of our samples are FFPE blocks that are remnant material given by pathology departments after ethical approvals. Those are used for a number of biomarkers and diagnostic work. We also have flash frozen tissue. We also have blood and derivatives such as plasma, serum, PBMCs, and we also have multiple different types of matched samples. So we have tissue plus blood or plasma plus flash frozen for a number of different studies. This is a busy slide. I just want to walk you through it briefly because I think many people ask the questions, what do you do? What do you use the samples for? And the reason the biobank was set up is because there is a dearth of ethically procured, high quality, well annotated samples for biomedical and pharma research in the world. So the things that we do, starting from the left, as I mentioned, we have FFPE blocks and flash frozen tissues that are used for drug target validation, for looking at diagnostic panels, for doing omics. In the second is OncoPrime, which is a panel of primary cells. So these are not cell lines, these are primary cells taken from breast cancer patients, glioma patients, oral cancer patients. These are cultured in our lab, and these are then established so that we can test new drug candidates. So there are companies who will send us their molecules to test alone, as well as in combination with existing drugs to see whether they can further enhance the activity of drugs, such as immuno-oncology drugs. So this is something rather unique we do. We are considered a living biobank because of the work we do in that. Then we have a growing amount of onco data, and that also comes in multiple flavors. So we have very detailed, structured patient data that is collated from medical records. We know the patient's demographics, we know their diagnosis, we know the treatments they were on when they came to the hospital for whatever procedure. In cancer, we are also collating their treatment after surgery and then their 3 to 5 year outcomes. So that is longitudinal journey of the patient. We also have a lot of pathology data which we are scanning, and we have probably the largest digital library of whole slide images, which is used for AI and ML algorithms for building predictive algorithms. We also have genetic data, which is what I'll talk about today. On the right-hand side are other applications. We do a number of immune inflammation assays to look at activity of compounds that could block or enhance antiviral, anti-inflammatory activities. And then we used to do some assays using discard skin for cosmetic companies that are looking to have better formulations without using rabbits and other animals. So there's so many uses of samples. So the type of data that we have accumulated comes again in different types of panels. And so one of the questions I was asking people is there a common database where one can put exome data and panel data and TSO100? So the types of data we have here, the pointer doesn't work, I guess. Okay, it was too far above. So we have Oncomine DX data for close to 1,000 solid cancer cases. This is what I'll show you. We also have growing data for an Oncomine comprehensive panel. We have whole exome data for 30 triple negative breast cancers for which we have treatment and outcomes data as well. We have the mismatch repair or the homologous recombination repair data for, this is wrong, 98 ovarian cancer cases. And we know the frequency of BRCA1, BRCA2 mutations in our ovarian cancer patients, and so on. As I said, in addition to that, we also have data on digital pathology. So recently we did a project on 1,000 breast cancers that were all uniformly stained with H&E, ER, PR, HER2, Ki67, all are digitized for building algorithms. Similarly, you were talking about MSI, Harsh. We have 675 cases of different solid tumors, 7 different solid tumors. These were all stained with H&E and the 4 MSI markers with IHC, and we have the scan slides. So talk about doing non-CRC, we can do that. And many more markers, many more things which I don't have time for today. So now this is a collaboration with Thermo Fisher. They have sequenced close to 1,000 of our cancers. This is done in their 46 gene Oncomine panel, both DNA and RNA. And the number of samples that were tested for various cancers are listed here: brain, thyroid, small cell lung cancer, non-small cell lung cancer, colorectal, and CRC. And what have we seen in these? So this is something interesting that we learned when we were working with Annie. One is that you can publish papers. These are the various things that you see in our tumors. But the reason we are doing it is to start to understand what are the actionable mutations, what are the actionable changes, what is the landscape of Indian cancers that can be targeted for precision medicine. So this is the foundation on which we are analyzing our data. And for those of you who may not be as familiar, although you are largely genetic counselors, you can probably teach this slide even better than I can, is that these are the levels of evidence based on which OncoKB classifies the mutations or changes. It can be other mutations, not just mutations. So if in green, for example, there is an FDA recognized biomarker, which can be a gene change, which is directly associated with an FDA approved drug and in that particular indication. So 3 things: the marker should be approved, linked with that drug, the drug should be approved, and it should be in the same cancer type. If all 3 match, you have the highest level of evidence, and that is actionable immediately. The next one is the biomarker is predictive but not yet approved. It is also for an FDA approved drug and in that indication, but the biomarker is not yet approved. The third is there is some clinical evidence, but it is still predictive, but it is not in that cancer. It could be that original cancer in which it is approved is lung, but now you are seeing this mutation in breast. So there is a bit of a gap there, and so on. The last 2 are interesting because they predict actually resistance to certain drugs. It is counter-prescribed. So we've looked at that data with this lens. And if you see here, a large number of our cases, Indian patients for which data is usually not available, you see a number of level 1 actionable alterations in breast cancer, CRC, thyroid, brain cancer, NSCLC, and very few in SCLC. So in terms of level 1, these are the common ones that are seen where the marker is approved, it is in the cancer type, and these drugs that I will show you in a minute. I'll come back to the previous one. So these are the drugs that are paired with this. So in thyroid cancer, BRAF V600E is approved, and the drugs that are associated are these. And it's a frequency that we see in our patients, 49%. That's a pretty high number. For breast cancer, PIK3CA, alpelisib, and fulvestrant, 33%. NSCLC, EGFR, 22. So this is not the complete list. I'm just showing you some of the top ones in each cancer. And if I was to go back, what is the frequency with which we see level 1 actionable alterations? You see pretty high in breast cancer, thyroid, NSCLC, but not as much in colorectal, very low in brain, and SCLC. I think, can you advance? It stopped working. Okay. Then we come to level 2. Level 2 is where the biomarker is predictive, but not yet approved for pairing with that drug. But it is predictive in this particular cancer itself. So I'll just show you 2 such examples. The rest of it we can discuss offline. So breast cancer, there are several level 2. Again, in colorectal, few. There are absolutely none in thyroid, brain, and SCLC. Quite a few in NSCLC. Can you please advance? Yeah. So compared to the level 1, the percentage of level 2 is quite lower, much lower, highest being 22% in NSCLC, followed by breast cancer. Go to the next. These are the drugs, some of the drugs. Again, there's not enough space to go through every single combination. For breast cancer, PIK3CA, other kinds mutations that are not level 1, different drug combinations, NSCLC, ERB2 mutations. This is a very new development. The targeting of HER2, ERB2 is no different than HER2. Oncogenic mutations happened just about a year ago. And colorectal cancer for KRAS G12C. Next. Now, so there are other level 3, level 4 resistance ones. Again, I just want to give you a flavor of that and move on to something else to remember. There are, no, go back. Yeah, just stay there. I just wanted to mention. Now, here are some other mutations or gene changes that are already designated as tissue agnostic. So the evidence for that mutation or that gene change to be driving that particular cancer is so high that it doesn't need to be validated for every tissue type. And currently there are NTRK, 1, 2, 3, RET, BRAF, and then tumor mutational burden that Harsh was talking about. So this means that the same drug that is approved for any of these can be used to treat various types of cancers as long as they share the same genetic mutation. Next, please. So now this is the percentage of cases in which we see these tissue agnostic mutations. So you see very high, green is thyroid cancer for BRAF. I showed you earlier also there was almost 50%. Lesser so for colorectal and then for brain and others. By the time you go from BRAF to the NTRK, which is really in fashion right now, I can't tell you how many requests we get for samples that have NTRK fusions. So when you get to NTRK, you see all of them are hovering around 1%, 2%, with maximum being in SCLC at 4%. And then the last one is RET fusions. Lo and behold, NSCLC has not as high a percentage of RET fusions compared to CRC, breast, and others. And it's not well appreciated that RET is not a domain of lung cancer. And on the right-hand side, bottom are the drugs that are mentioned for these various tissue agnostic markers. Next. Another point I want to make is that these evidences for OncoKB are not static. These are constantly evolving with more evidence coming in. So keep re-looking at your data. Don't stop. This is our own data just for breast cancer. This is the highest number of cases for which we have this data, 480. The data when we got initially in October, this is how it would pan out in the pie for level 1. Just look at the green, which is large. Now, in about 7 months, you can see it's gone from 45% to 53%. So keep looking at the data and keep up with it, as somebody was saying earlier also. Next. Again, apart from this being updated from time to time, the directionality of the change also can be different. So sometimes, like I mentioned, in breast cancer, there were several cases that were originally previous level 3A. That means that drug is not yet approved for breast cancer. So the marker may be predictive, but not yet in breast cancer. But since that time, FDA has approved capivasertib and fulvestrant combination for breast cancer. So that moved from level 3A further up, more significant, to level 1. Same thing for BRAF. It went from 3B to 1. But there are times it can be downgraded as well. So in the case of RET, for medullary thyroid cancer, pralsetinib was originally level 1, but because of the withdrawal of FDA for this drug for RET mutant MTC, it has dropped down. So please keep those things in mind. This is very recent in October. Next. So in conclusion, we have started to address the issue that a lot of genetic data does not come from Asia and Africa. So we are open to partnerships with people who are looking to annotate genetically data because we have a large number of samples. We find actionable alterations in a majority of breast, thyroid, and lung cancers, followed by colorectal cancer. We have very few level 1 or level 2 alterations in SCLC and brain tumors. And we know that the prognosis is very bad. So we need to continue to find other actionable mutations. As I mentioned, OncoKB levels of evidence keep changing, and they can be bidirectional. And as you see more and more gene alteration data coming out, as FDA continues to integrate this information into drug approvals, we hope that the number of tissue agnostic markers will get more, and presently approved drugs will get expanded for use, which means cost of drug development will not be as high. You are using the same drug for more indications. So it basically emphasizes the importance of identifying actionable genetic mutations, alterations, to aid the choice of targeted therapies to provide more personalized treatment options for Indian patients. Next, I think, is our team. If I can, okay, this is a very large number of samples we have, and we worked out a lot on FFPE blocks, even as old as 2005, to get good genetic data. Next is the team, because you're only as strong as your team. So this is our team. Thank you. We'll have questions later, right?
Thank you. Now we can probably start the panel.
Thank you all the speakers for your very insightful presentation and talks. I would now like to invite chairperson, co-chairperson, and all the speakers for the panel discussion.
Right, let's have the panel discussions. Are there any questions from the floor for any of our speakers? And Prof. Shirley, I can see that she's online. Is Professor Managari online as well? Professor Managari, can you hear us? And Prof. Shirley, can you hear us? Okay. Any questions from the floor? Yes. Can we have some of the mics?
Thank you so much for all the speakers. It was a very insightful session. I especially liked Dr. Shet's presentation. You had mentioned about a lot of factors, and then the banana thing especially. And I had to, no, on a serious note, I had to mention that in our Indian community, we have, even North Indians started, I mean, I have seen in our, culinary, we have a raw banana dish since, centuries. No, it's actually, so it's basically, I wanted to clarify, it's basically the raw starch that helps in colon. And so that makes a difference, right? Banana and a raw banana. So we cook raw bananas, and it's the raw starch that is. So I wanted to link that question to, do you see any pattern of diet to the colon cancer, and how has it influenced? And you have studied a lot of cases. So do you see any pattern?
Yeah,
changing food these times.
Yeah, there are some wonderful epidemiological studies depending upon certain countries, especially from the top of my head. Italy is an excellent example of that. You see difference in eating habits between North and South Italy, and you also see correlation with the colon cancer rates. The general rule of thumb is high fiber diet is good for you, low processed food is good for you. As little processed food you eat is best for you. If you have obesity, it's bad, especially in Lynch syndrome. As I showed you, it gives you an excess risk on top of the genetic risk that you carry. Exercise is also very, very helpful. But beyond that, if you are talking about any micro or macronutrients at scale, that sort of analysis has not been done to date, because it's very hard to prospectively follow up and ask your diet and diet history questionnaire. So that's exactly what we are now trying to recapitulate with our Lynch syndrome patients.
When we talk about having aspirins long term, it's better to talk about curcumin and peppers long term and things like that, which is part of our food and culture. Yeah. Thank you.
Hi. This is a question for Dr. Manogari Chetty. First of all, let me thank you for this wonderful talk which you presented. A very interesting topic about use of exosomes. So my question is, we know that exosomes are secreted by the normal cells and the cancer cells as well. So how do you differentiate the exosomes coming out from the cancer cells, from the normal cells in the test which you do?
Yes. So what we've got to do is we've got to always, what we are doing at the moment is we have the controls, which we do quite a comprehensive history to exclude many diseases, many autoimmune, whatever hereditary conditions that they know of. And then we looked at the exosomes in their blood. And then what we have done is we looked at individuals with known oral cancer and then compared the blood, and you could, there were many different characteristics in the cancer exosome compared to the normal exosome. And some of the molecules that we identified, I can't remember off the top of my head, but even the content of the exosomes were different, apart from the size, apart from the shape. So that's how we found out. So that was our baseline study, and based on that, we will obviously begin to develop diagnostic tools for our population here in South Africa. Is that answering your question?
Yes, please. Thank you so much.
So, as far as the risk by gender is concerned, nobody knows. But even at a population scale, if you remove Lynch syndrome equation out, even at a general population scale, males are generally at higher risk of having colorectal cancer than females. Again, the reason for that is unknown. But when it comes to therapy, we see that the response is equivalent in terms of reduction in risk of cancer. In fact, if you are an MLH1 gene carrier, females are at an exceptionally high risk. Even in MSH6 Lynch syndrome carriers, the risk of endometrial cancer is very, very high. And in those, aspirin actually is very, very good compared to resistant starch.
Yeah, that's a good news.
That's a good news, absolutely.
Any other questions from the floor? Eric, there's one more.
Yeah, just to add on to what Suraj had asked, and you said that there was an observation in RCC Trivandrum with scientists in Rajiv Gandhi Center for Biotechnology. So in CRCs that there was overwhelming youngsters. When you say young onset CRC, you would see 27, 35, 42. But they were seen in adolescents less than 25 years with no genetic predisposition tests. And generally in the state, we do know the food habits from North Kerala and Malabar region, North Kerala and South Kerala. And it was completely, told across in the general, I don't know if a paper was published, but it came out in the news across all channels, telling that food habits, especially because of the amount of reduction in fruits and vegetables and increase intake of non-vegetarian food, and how it plays a very important role, and because it's become part of their lifestyle. And they had shown numbers actually, because Kerala has only one RCC and all of them come for treatment there. And along with scientists at RGCB, they have shown that it does play a role. And they specifically ask questions to all these young onset individuals with no known significant family history. And in some of them, they have also done genetic testing and have not found any, it was all MSI negative itself. So, and some of them have also gone for germline testing. So that's some data that is available there, done by the doctors and scientists.
Thank you.
Ma'am, one last question.
This question is to Dr. Manogari. Is it possible to detect cancer even before the formation of tumor with the exosomes, marker testing? Kindly comment on this.
Yes, actually, the thing is, what we are trying to do is find out if there is, in our population, because it is such a diverse population, we are trying to, at this point in time, we haven't even done whole genomes in our population. We can now, let's say, oral cancer in our population has a mutation in this particular gene. So we haven't even gotten to that point yet. So the point that we are looking at now with the liquid biopsies is trying to find out, before you get already a developed tumor, when you start getting this mutation, already there should be some changes in the exosomes. And that is what we are trying to identify at this point in time. So that's going to tell you, okay, this patient most likely is going to go on to develop. We haven't, like I said, the initial thing about doing our whole genomes, identifying what the mutations are in terms of oral cancer in our country. And then we can say, okay, this is a marker, this is what we will look for in the exosomes. But that is what we're working towards. If that helps understand better. We're a little bit behind at this point in time. Thank you.
Yeah, this is to Shirley. Hello, Shirley. Thank you so much for your talk. You still inspire all of us so much. Huh? The youngsters, I think, here really should take a great inspiration from you. It's been, more than 10 years that we have been dealing with Shirley in the BGCI. And I don't find I find that her enthusiasm is increasing day by day. Anyway, it was a very good talk, but conveying this very sort of bad news, I should say, for the patients when they are having cancers, you have to make it sound as if it is not that bad, not to worry about it, and show them the bright future. I think that is the lesson that we have to take for the genetic counselors to cheer up these patients. Thank you, Shirley. It was very nice to see you here again. Thank you.
Well, thank you very much. It's a great, great pleasure to be here. And I quite agree with you about being cheerful. And one of the things, of course, about genomics, testing the tumors, as we've been hearing, is if you find some mutation that is going to be a target for treatment, which is one of these important mutations, a driver mutation that you can actually target. It has been very, very successful in some cases. And some recent studies in lung cancer, for instance, have been amazingly successful. So I think all this genomics is worth doing.
Thank you so much, Prof. Shirley. Just one last question for Dr. Jain. It was very nice, and you have done a lot of the genetic profiles for those who contributed to the biobank. Were any of the genetic profiles returned back to the patients, and did any of the patients use it for their treatment options?
This is a very important question which we addressed early on. We do not return results to the patients because we are very disjointed in time. So the samples that we have sequenced may be 10 years old, 20 years old. And a lot of our work is in research protocols, research panels that are not approved for diagnostic use. So when we come across something interesting that is of a patient that may be less than 5 years, this happened with Ross for one of our lung cancer patients, we normally go back to tell the treating doctor and leave it with them to decide whether to give it back to patients. But worldwide, biobanks often do not return the data back because the samples are tested much later. They are tested in tests that are more geared towards identifying new drug targets or new biomarkers, but not clinically validated tests. And so it can raise anxiety in patients if you say, I found this result, but I cannot say it's a validated test. I cannot say whether this is completely backed up. So, no.
But in instances, for example, like the ovarian cancer, then Hirsch can put in your comments about policy.
I think Dr. Jugnu already answered that question very aptly, that if there's a disjoint in time, especially these are somatic variants that we are dealing with, then the likelihood of the utility starts to diminish very, very quickly.
But what about certain cancers,
these are somatic
like ovarian
variants.
cancers?
These are somatic variants. If it is a germline variant, I completely agree. And because taking in view of that, especially the study that we are conducting at the moment, it's not just restricted to the patient. It gets expanded to first-degree relatives as well.
So for
because that's the
certain
benefit
cancers,
of doing it.
of certain cancers like ovarian cancer, where you did mention that you did find somatic BRCA, would there have been an opportunity to verify that in germline at patient's choice, so that they are aware that there might be an inherited risk in their families. This is just for discussion. It's not, I'm sure you have a policy in place, but perhaps, in terms of consenting and what patients consent to know and not what to know when they deposit in the biobank.
So most of these samples come under waiver of consent requirement as per our ICMR guidelines if the patient sample was taken for some medically necessary reason. So these are all removed surgically for treatment of patient. The original purpose for which they were taken should have been completed. So the pathology diagnosis and all of that was completed years ago. They have to be stored for patient use for X number of years. It used to be 5 years as per NABL. After 2019 February, it has become 10 years. So the hospitals that are NABL accredited and so on will keep them for 5 to 10 years. After that, we get these samples. So there is really no possibility almost of recontacting patients after 15 years, 20 years, and you may cause more trauma. There are many publications about how biomedical research results should or should not be given back to the patient because they may not want to know, and the doctors themselves may not want to be involved. Yeah. Correct. Correct. But germline, yes.
What do you build in there for the patient or the families who are giving their. Now, for you, you may just be doing with the NABL. NABL, you have 10 blocks. 2 they may give you earlier. If something has come out for the new
we cannot compromise diagnosis. So we never take blocks that were used for diagnosis.
But the question still remains, and the discussion for biobanks still remains, that how can we or should we contact patients if you think anyway it is of use? That's the question.
So wherever it is germline, we have been involved early on in several germline testing. There definitely. And the first case that I remember very, very well, I think I was telling you last night, was when one of the GI docs contacted us and said he was operating on a patient. He was doing colonoscopy on a patient and operating on a patient, operating on a colorectal cancer patient who had more polyps than he had ever seen in his life. And he just said, you have to get this patient tested. So we sent that sample for testing, and it had a pathogenic variant of APC gene, not surprising. And then the doctor counseled the patient. The patient had a 10-year younger sibling who was barely 19. Then that sibling was counseled. Her blood was tested only for that APC variant. Unfortunately, she carries that. So a preventive colonoscopy was done, and she had benign polyps. And the doctor is going to stay in touch with the patient, and hopefully the patient keeps coming back because she had a 6-year-old son. And obviously everyone will want to know whether the son has it. But as per the doctor's advice, they would wait till the son has turned 18. I don't know.
It's early onset, but the likelihood of having large polyps at the age of, or cancerous polyps at the age of 18 is very, very low.
Prof. Shirley, would you like to comment on that? Screening for FAP, for carriers of APC, at what age? And what age can we do genetic testing?
Absolutely. Yes, if we know, well, it was absolutely the right thing to do to test that patient for polyposis, for APC mutations.
And obviously the discovery of that would probably lead to that patient having their large bowel removed at some point, because that's the definitive treatment once they have multiple polyps in the colon. And we tend to, some people want to test their children very young, but we suggest that they shouldn't do that. That if they're at risk, they should be tested at about 12, 14, something like that. Because if they had polyposis, they wouldn't be likely to run into any trouble from polyps before that age. And we normally would start, if they had polyposis, we would start screening from about the age of 14, something like that. Because in affected people, the polyps really don't get going until after that age. But certainly the older ones definitely should have the genetic test offered to them, because they can have multiple polyps by the time they're 20, and it's important to make sure they're diagnosed by that age. Because if they have prolific polyposis by 25 or something, they may well be the age at which they need to have a colectomy.
Thank you very much. I think we've had a very interesting session, and I'll pass it back to our MCs. Are we going to close the session?
I'd like to request Ms. Suki to please present our speakers with a memento.
I would now invite Dr. Usha Dave to felicitate Chairperson Dr. Parani and Co-Chairperson Ms. Sukinu. Also would request you to stay back to click a picture.
Thank you to all our speakers. Thank you, Dr. Harsh. Thank you, Dr. Jugnu, for joining us in person. Thank you, Dr. Manogari, and thank you, Dr. Shirley, for joining us virtually. I request all of you to be present at 9 a.m. tomorrow.
Please, all of you join us for dinner at 7:30.